So first we are going to start with microbes in human welfare, the simplest chapter of whole biology. Okay, this is the most simplest chapter of whole biology in which we are going to study about different microbes and their products which we use in daily life. Okay, so in this chapter, what points we are going to cover? First, we have to see that microbes in household product, microbes in industrial product, microbes in sewage treatment, microbes in biogas production, microbes as biocontrol agent, and microbes as biofertilizers. So these all are the different values, different views of microbes which we use in our daily life. Okay, so first we have normal introduction about microbes. Microbes are present everywhere. Okay, in sand, in soil, in air, in water, in fire, in the water, that hydro water vents. Okay, hot water springs, atmospheric air. It also contains microbes, and some microbes are also found in our outer space also. Okay, so microbes are that organism on earth which present all over the world. Okay, there is no place on earth where you cannot find microbes. Okay, so that thing is written here. Microbes are diverse protozoa, bacteria, fungi, microscopic plant, virus, viroids, pyrons. All these come under the category of microorganisms. Okay, next we have microbes like bacteria and fungi can be grown for the human welfare. See, microbes have two types of properties. One, it is positive, another one is negative. Positive way we use for our better purpose, which we are going to study in this chapter, which is your microbes in human welfare. And some microbes are dangerous. They are very pathogenic for human race. Okay, that cause diseases, different pandemics they will cause. Okay, so in this chapter particularly we are going to study about the good features of our microbes. Okay, so next, microbes are present everywhere. As I said, example, thermal vents in geyser temperature above 1000 degrees Celsius, you can find microbes there. Thermus aquaticus. Okay, these type of bacterium, archibacteria, which are present inside the thermohydroventral vents, that can easily survive above the temperature of 1000 degrees Celsius. Okay, deep in the soil, under snow, diverse protozoa, bacteria, fungi, virus, viroids, pyrons present everywhere on earth. Okay, useful antibiotics. The usefulness of your microbes is to produce antibiotics. Different enzymes, different food supplements are produced by our microbes. That is the positive role of microbes in our daily life. Okay, while the harmful is cause disease. Okay, you know that disease are caused by many microbes, different which are we are going to study in human health and disease chapter. Okay, so first, microbes in household. Okay, what we do microbes with in our house. Okay, the household activities where microbes are used daily. For example, your curd. Okay, it contains a bacterium, a rod bacterium, which is called bacillus, lactobacillus acidophilus, or lab bacteria, which is present inside your curd. Okay, so a common example in the production of curd from milk, that is the most common example of microbes in our household. Okay, microorganisms such as lactobacillus and other commonly called as lactobacillus bacteriums lab, they produce, they convert your milk into your curd. Okay, that digest the protein which is present inside the milk and convert that milk into curd which we eat. Second thing, that curd, it is very high in concentration of vitamin B12. That vitamin B12 is good for our gut. Okay, so that is the good use of microorganisms. Okay, so this is the microscopic, ultra microscopic image of our lab. Okay, this is again an ultra microscopic image of lab. Next, microbes in household in the fermentation processes for formation of bread, yeast, Saccharomyces cerevisiae, that is the yeast, baker yeast, which is used to produce bread, dosa, idli, dhokla, all these breakfasts which we eat all over the India, either north or south, it is produced with the help of a fungus, a yeast, which is called Saccharomyces cerevisiae, which do fermentation of carbohydrate. It evolves CO2 gas in the reaction that come at the end of the process of the thing. Okay, so this is your bread which you eat in the breakfast, and this is your Saccharomyces cerevisiae and fungus. Okay, next, toddy, again it is an fermented drink which is drinken in South India. It is an alcoholic beverage which is produced with the help of your yeast. Okay, next, microbes are used to ferment fish, soybean, bamboo shoot, and provide cheese. Okay, in the next slide we are going to discuss about cheese, how a cheese is produced by the help of microbes. Different images. This is the toddy which is produced with the help of palm. Okay, this is your fermented fish, which is eaten in South Korea, North Korea, China, Japan. Then it is fermented bamboo shoot, again eaten in Japan, China. Okay, this is fermented soya bean, which is again eaten in North Asian and South Asian countries. And this is the most famous drink, soju, which you see in Korean drama. Korean drama. Okay. Next, we have three different forms of cheese. Okay. Roquefort cheese, Camembert cheese, Swiss cheese. Okay. Roquefort cheese, it is produced by a fungus, Penicillium roqueforti. Camembert cheese, produced by a fungus again, Penicillium camberti. And then Swiss by a bacterium, Propionibacterium shermani. Again, your Roquefort cheese produced by a fungus, Penicillium roqueforti. Then your Camembert cheese produced by Penicillium camberti. And at last we have Swiss cheese, which is produced by Propionibacterium shermani. Two are fungus, one is bacterium. Now I have the images of that. This is your Roquefort cheese and this is the fungus, Penicillium roqueforti. This is your Camembert cheese and this is the organism, Penicillium camberti. Next, this is your Swiss cheese. Okay, the cheese which you see in Tom and Jerry, eaten by Jerry. Okay, and this is the bacterium, Propionibacterium shermani, who produce this cheese. And this holes in this cheese is the result of fermentation which done by this bacterium. Okay, now next, microbes in industrial products. For example, your alcohol, beverage, drinks. Okay, so fermented beverages is again Saccharomyces cerevisiae. Now its name changes, brewer yeast, which is present inside all alcoholic drinks. Okay, which is the main source by which you can produce alcohol. That thing is your brewer yeast or Saccharomyces cerevisiae. Okay, so I have a table in which you will see that different concentration of alcohol in different drinks. Okay, brandy, it is produced by fruit juice, is contain 40 to 50 alcohol. Okay, then whiskey, it is produced by cereal grains and it contains 40 to 55% alcohol. Next, we have rum. It is made by molasses of sugarcane. When you cut the sugarcane, the remain, the root part, the stem root part of the sugarcane is used to produce alcohol. That is 40 to 55% concentration in your rum. Next is wine. It is produced by grapes and other fruit, and its value is 10 to 22%. While next is beer, which is produced by cereals, and it have concentration of alcohol 4 to 8%. And at last we have arrack, which is produced by paddy, rice or wheat, and its concentration is 50 to 60. Now beverage drinks is of two types. One is we have that in which yeast is present. Okay, that is called fermented drinks. Another one is distilled drinks. Fermented drinks are those drinks in which alcohol concentration is less, and distilled alcoholic beverages are those things in which alcohol concentration is high, and that alcohol concentration attained is in that drinks by the help of distillation process. By adding different chemical substances or doing distillation of that beverage, we add more alcoholic concentration and increase the concentration. Normally your yeast die after 13% production of alcohol. After that you have to increase the concentration of alcohol, so that thing is done by the process of distillation. So in this list, your beer and wine, they are non-distilled beverages. Okay, while brandy, whiskey, rum, arrack, these all are distilled alcoholic beverages. Okay, now next, this is your yeast which is present in your alcoholic drinks, which is used to do fermentation. Okay, now next is antibiotics. Okay, the major role of microorganisms, the antibiotics, the second main role of microorganism to produce a useful thing, that is your antibiotics. Okay, and the first antibiotic which is developed in the world is penicillin. Okay, and developed by an organism which is your Penicillium notatum. Okay, and this is your Alexander Fleming. He discovered penicillin, and this discovery of penicillin is an accident. It is not a real discovery, we can say. He do this thing accidentally while doing his research on some bacterium. He saw one day that some fungus is growing on that petri dish and it's killing the bacteria by secreting some substances, and he tell that that substance is antibiotic. The word antibiotic say itself its meaning. Anti means against, biotic means living, against the living. So after that he say that there is some substance which is producing by fungus which killing by that microbes, bacterium, and gave that thing name antibiotic. Okay, after that, Florey and Chain come in the play, and they modified that antibiotic power, increased the power of antibiotic, and due to which these three guys get Nobel Prize. Okay, now next, this is your Penicillium notatum. This is the fungus. Okay, and this is the petri dish in which your Penicillium notatum is grown. This disc-like structure is the colony of Penicillium notatum, and this yellow matter is your bacterium growth. Okay, now next, chemicals, enzyme, and other bioactive molecule which is produced by your microbes. Okay, so first we have Aspergillus niger, which produces citric acid. Next, Acetobacter aceti bacterium again produce an acid which is acetic acid. Next, Clostridium butyricum, which produce an acid which is your butyric acid. Next, Lactobacillus, which produce lactic acid. So these all are different acids produced by different organism. Again, we can say Aspergillus niger, a fungus which produces citric acid. Next, Acetobacter aceti, a bacterium which produces acetic acid. Next, Clostridium butyricum, and again bacterium which produces butyric acid. And at last, we have Lactobacillus, a bacterium again which produces lactic acid. Okay, this is the colony of Aspergillus niger. You can see it is also called sea urchin colony because its colony structure is just like a sea urchin as an echinodermata. Okay, and this is the ultra microscopic image of your Aspergillus niger. Next, this is the Acetobacter aceti. This is your Clostridium butyricum, and this is your Lactobacillus burgerius. Okay, now next we have chemical enzymes and other bioactive molecules which is used in hospitals. Okay, that is your streptokinase, which is an clot buster, and it is occupied from a bacterium streptococcus. Okay, next we have Monoscus purpurus, and species of yeast which is used to produce a medicine which is called statin. It is an blood cholesterol leveling agent. This is the medicine which we eat to decrease the cholesterol level. When we take this medicine, the cholesterol level inside our body get decreased. Okay, next we have cyclosporin A. It is produced by Trichoderma polyspermum, again a fungus which acts as an immunosuppressive agent. It suppresses the immunity of an organism. It is given to the particular patient after that organ transplant surgery. Okay, now we have some enzymes like lipase, which is used in laundry. Okay, which is present in our detergents. Okay, next, pectinase and protease. These are present in bottle juice. As you know, there is one bottle juice which you drink which is called paper boat. Okay, when you drink that paper boat, you feel some pulpy thing. So in that drink, there is no pectinase and protease. But when you drink maza or that fruity, you feel that there is a clear drink is coming inside your mouth. The reason of that clarity in the drink is pectinase and protease enzyme. Next, streptokinase, I said already. Okay, this is your streptokinase injection, and this is your streptokinase enzyme. This is the lipase which is present in your detergents. Okay, next, this is your cyclosporin injection. This is your statin drugs which is taken to decrease the blood cholesterol level. Okay, this is your Monoscus purpurus grown in a colony. Okay, and this is the medicine atrin which contain this concentration of Monoscus purpurus. Okay, now next, microbes as biocontrol agent means now we have to control some activities of other organism which come under microbes as biocontrol agent. So first we have biocontrol is used to biological method for controlling plant diseases and pests to kill the disease present in the plant. The pathogen which is present inside the plant has been killed by these agents. Okay, so first we have dragonfly. Okay, dragonfly, it eats mosquito. Okay, and larva of mosquito. Second thing is your ladybird. Okay, ladybird, it eats insects like aphids which destroy the crops. Okay, so it is the first example of biocontrol. Okay, second is Bacillus thuringiensis. This thing we are going to study in biotech also. Bt cotton, this cotton. Okay, Bt cotton. In this we have a gene of an organism which is called Bacillus thuringiensis, and toxin gene and of Bt toxin which we call that causes tumor inside the plant body. Okay, so the dried spores of Bt are mixed with water and sprays on the vulnerable plants which is easily get infected by different organisms, and these are eaten by caterpillars. Now when the caterpillar eat that Bt cotton which contain Bt toxin, the Bt cotton get inside the gut and it activate and start do perforation of the gut. Okay, example Bt cotton. Next we have trichoderma fungus. Okay, it is present in the roots of many plants which avoids the invasion of pathogens. Okay, Next, we have baculovirus, a species of virus which especially that nucleopolyhedrovirus, which also inhibits the entry of pathogen inside the plant body. Okay, this is the structure of nucleohedrovirus or your baculovirus. Okay, now next, microbes as a biofertilizer. Now we have the different microbes which is used as a biofertilizer, which increases the fertility of the soil, which increases the nitrogen content and phosphorus content in the soil. Okay, so we have some examples: Anabina, Nostoc, Oxytoloria, and Haplosipnoa. Okay, this is your Anabina, this is your Nostoc, this is your Oxytoloria, and this is your Haplosipnoa. Okay, these all BGA perform nitrogen fixation inside the roots or inside the body of that plant, due to which nitrogen level increases inside the soil and that nitrogen absorption by the plant increases. The efficiency of nitrogen which is present in the plant, it is fixed by these organism, and this organism when live inside the body of plant, they perform a symbiotic relationship, a mutualism relationship. Okay, so these are the symbiotic organism. Your Anabina azole, the most famous example of nitrogen fixer association with an organism, which is your Anabina azole. Anabina is an your BGA and Azolla is an fun, which has lived in a symbiotic relationship in which your Azolla, Anabina live inside the leaf cavity of the Azolla plant. Okay, where it do nitrogen fixation. We have some free living also which do not need any support. They are not in any type of relationship, but they perform nitrogen fixation freely in the soil. Example, your nostoc or Cytoloria. These all are your BGA, blue-green algae. Okay. Next have your rhizobium, which is in bacterium. Azospirillum, again a bacterium which do nitrogen fixation for plant and live as symbiotic relationship with leguminous plants. Okay, this is rhizobium, which live in the root nodules of leguminaceae family plants and do nitrogen fixation for them. Okay, next we have mycorrhizal relationship, in which we are going to study a relationship between a higher plant with a fungus, Glomus genus fungus, in which Glomus fungus fix phosphorus, mainly phosphorus. This time it is not nitrogen. This time it is phosphorus. So this Glomus fungus, this fix phosphorus for the higher plants, in which your plant gives shelter to that fungus to live in the soil. Okay, in the that roots. Okay, next, microbes in the production of biogas. Okay, now we are going to develop biogas with the help of microbes. For example, your most famous organism which do this phenomena of production of biogas, that is your that bacterium, methanogen. Okay, methanobacterium. Okay, which are we call archibacteria. So biogas is a mixture of gas. Mainly in this we have methane, okay, and other gases like CO2, H2S, and hydrogen in some level of water vapors. Okay, methanogen grow on anaerobically on cellulose material to produce methane along with CO2 and hydrogen gas. Example, methanobacterium. Methanobacterium an example of methanogen. Different methanobacteriums are there in the world, like pneumococcus, methanococcus. Okay, so these all are different species, strains of bacterium methanogens. Okay, so methanobacterium is found in the anaerobic sludge and rumen of the cattle for cellulose digestion. Okay, your cellulose, it is in a form of carbohydrate, a polysaccharide of carbohydrate. Okay, so these bacterium digest this thing in the anaerobic condition and produce a gas which we call methane, and methane with CO2 and H2S and and your hydrogen sulfide and hydrogen makes a some of this thing which we call biogas. Okay, next, cattle dung contain these bacteria, so dung is used to produce biogas, then it is called gobar gas. Okay, see in this image, these people pulling that gobar inside a tank to produce biogas. Okay, next, this is the diagram of biogas plant. Okay, in which we have a digester, in which we put these all sludge. Okay, from this way that sludge go inside the digester, and above that we have a gas holder. So after some month, when the person come and see that gas holder will go upside. Okay, because the gas is been accumulated inside, and when that level is increased, the sufficient level is filled, then there is a valve which is open, due to which this gas has been transmitted to the final area or final processing, for the final processing to fill in the cylinder. Okay, so this is your outlet pipe, this is your inlet pipe from which you put the sludge inside the digester, then your anaerobic bacterium, micro, that your methanogens, methanobacterium, rumenococcus. These bacterium perform the digestion of this material and produce methane gas, and that methane gas will go up and accumulate in the gas holder. From gas holder, now there is a pipe, outlet pipe from which this gas go out. Okay, now next, this is the some biogas plant which is present in the household. You can also make biogas plant in your house also. Okay, and this is a well developed biogas plant in America. Okay, in some countries biogas plant is used to lit the road lights. Okay, next, this is again the image of your normal biogas plant. Okay, and this is a big scheme, large scheme biogas plant present in a developed country like America and UK. Okay, next, microbes in sewage treatment. Okay, now is, as you know, we daily take bath, we clean the car, we give water to our plants, we use water to produce different vegetable food that in the household. So the water which we use, which go in our drainage system, where that water goes, it goes to your sewage treatment plant to become that water pure. Okay, so sewage treatment plant is a plant which is present outside of your city. Okay, in which all the water, all the polluted water, all the bad water has been accumulated and converted into a pure water, and then after that it will be transmitted in the water bodies. Okay, so your sewage treatment plant basically having three steps. First is primary treatment, second is secondary treatment, and third is tertiary treatment. Primary treatment is also called as physical treatment, secondary treatment is also called as biological treatment, and tertiary treatment is also called as your chemical treatment. In primary treatment, we use primary processes, physical processes like strainers, okay, different forms of strains, chalni, that things are used to strain the big parts of waste like polythene, pipes, okay, different plastics, stones, sand that had been filtered down in your primary treatment process. Okay, so that thing we are going to see this is your primary tank. Okay, primary treatment tank part. Okay, so in this you have a filter like appearance slit in which your large particle has been remain here. Now that water will transmit into grit chamber. In grit chamber, the stone and sand which is present inside the water has been set down. Okay, now that water contain small biological particles which is we send it to your secondary treatment plant. Okay, now your secondary or biological treatments. Okay, now substance which is coming from your primary treatment plant which is going inside the secondary treatment is called sludge. Okay, sludge, or the upper thing which is floating on that water, sludge water, okay, is called flocks, which is a mash of fungus and bacteria. Okay, this is your flock which contain mash of bacteria and fungus. Okay, they are present at the as like this. They present as a like of mesh in which your fungus, bacteria has been present. Okay, so this is present inside your flock which floats on the surface of your sludge. Okay, so primary effluent is passed to the large aeration tank and constantly aerated. This allows vigorous growth of aerobic microbes. Again, your secondary treatment plant, the secondary treatment chamber of your sewage treatment plant having aerobic bacteria. Okay, because oxygen is there. Okay, next, these microbes consume the organic matter in the effluent. This reduces the BOD, which is called biochemical oxygen demand of the effluent. The effluent which is coming from your primary treatment, it have very high BOD. Okay, so these aerobic bacteria decreases the BOD of that water by eating all the organic matter present inside the water. Okay, now next, that BOD, now the remaining part of that thing which is set down in the below, it will send it into a second chamber or other chamber where anaerobic bacteria start to do the work. Okay, and they start to digest that material, that biological material, and use to produce biogas. Okay, the remaining sludge is pumped into the large tank called anaerobic sludge digesters. Here some anaerobic bacteria digest the thing. Okay, and that thing has been converted and sent it to the biogas plant to produce biogas, or in some sewage treatment plant they have their own biogas production units. Okay, now we take some part of that activated sludge and add again inside the aeration tank to increase the number of bacteria which is present inside your secondary treatment. Okay, so this is the process. Okay, primary effluent which will go inside your aeration tank, then constant agitation means mixing of the thing will done constantly, then aerobic bacteria in the flow consume organic matter. When that mixing take place, at same time that bacterium which is present inside that water, it eat all the organic matter and your BOD is reduced. Okay, next effluent to the settling tank. The settling tank, the thing which is settled down in the tank, it will send for the biogas production, which called anaerobic slug digester. Now from anaerobic slug digester, that thing will go and digest by the help of anaerobic bacteria and send to produce biogas. So your sewage treatment plant is that type of plant in which there is no waste. Waste is used to produce biogas. This is the waste. Okay, these things are the waste of your primary and secondary treatment. So these waste are also used to produce biogas. Okay, next, this is the diagrammatic view or a blueprint of an biogas plant, in which you will see that different processes are taking place. In this step we have the primary treatment going on, in this we have secondary treatment, and after that we have other pipeline which is taking that remaining waste to the sludge digester, where we do that digesting of the sludge by the help of anaerobic bacteria. Okay, this is again on flow chart. In the form of flow chart we are discussing that primary treatment, secondary treatment, and at last we add some disinfectants which we are called tertiary treatment, in which we add chlorine, fluorine and ozone, chlorination, ozonation and fluorination. Okay, now last part of this chapter, that not last but last second part, we can say that some ministry of India have applied two plans. One is for Ganga river and second for Yamuna river. As you know, these both are holy rivers for India. Okay, but due to the day by day increasing population and use of factories, that waste product directly going inside these rivers, the BOD level of these rivers and increases, due to which these rivers become polluted. So for that, Indian government take an action, take a plan which we call Ganga action plan for Ganga river and Yamuna action plan for Yamuna river, to save these rivers, to make these rivers not to become degrade or polluted fully. Okay, now next thing, this is your microbes in human welfare. Now we will go for the second chapter. First chapter done, now the second chapter. Now the second chapter is your biotech. Okay, now your technology has been added with biology. Okay, the new technologies are being added with biology to produce new materials by the help of different machines and microbes. That thing is your biotechnology. In this 12th class, this chapter is divided into two parts. This unit is divided into two parts. First one is biotech principle and processes, and second one biotechnology and its application. In the first chapter we are going to discuss about what are the principles and what are the processes we are going to discover in this chapter. Secondly, application. Now we applied that processes inside different examples, and different examples we applied that processes. Okay, so let's get started with that. Okay, so first, genetic engineering. Okay, it involves the cutting of desirable segment of DNA and joining of desirable segment in a vector to produce recombinant DNA, which is called rDNA. Okay, recombinant DNA or hybrid DNA. Okay, which is accumulated in an organism to make that organism a transgenic animal or a transgenic plant or a hybrid organism. Okay, so the biological tool used in the synthesis of recombinant DNA includes enzymes, vehicles, vectors, desirable DNA, and host cells. Vehicle and vector DNA both are same things. Okay, so first we have tools for DNA, in which we have enzymes. Okay, different enzymes are present there to do biotechnological researches. Okay, endonucleases, exonucleases, DNAs, RNAs, proteinase, cellulase, okay, chitinase, okay, lysozymes. So these are different enzymes which is used to perform biotech practicals or biotechnological examples. Okay, so first we have enzymes. A number of specific kind of enzymes are employed in genetic engineering. This include lysing enzyme like your exonuclease, endonucleases. Okay, cleaving enzyme, again endonuclease and exonuclease, cutting type of enzyme. Synthesizing enzymes, your transcription enzymes, okay, and your joining enzyme like your ligase, okay. So first we have lysing enzymes. These enzymes used to lysis different materials, different products. So in plants, that enzyme is your cellulase, okay, or pectinase. In animal, that is your lysozyme. In bacterial cell, again it is lysozyme. And in the fungal cells or an organism which have very high stiff cell wall, it is your chitinase. Okay. Next we have cleaving enzyme. Cleaving enzyme means the enzyme which cut your DNA. Okay, and it is of two types: exonuclease and endonuclease. Exonuclease are those type of endonucleases which cut your DNA from outside. Okay, and endonucleases are those enzymes which cut your DNA from inside. Okay, inner side of the DNA, this is cutted by your endonucleases from a specific sites. Okay. Next, restriction enzyme recognize specific location in the DNA strand where it makes the cut. The sequence of which DNA strand recognize the specific site for cutting is not recognition site. Okay, see. If this is a DNA, circular DNA, or having this area, and this area is a restriction site. So your restriction endonuclease will come and cut this area, and this area. Okay, that's how your restriction endonuclease work. It will have specific sites. Specific endonucleases have their specific sites inside the genome. Okay, inside the DNA. So these recognized sequences are usually palindromic. Okay, palindromic like this. This name Naman. Okay. From first word is N, last word is N. When you read this word from back side, you will find the same thing. And when you read this word from the starting, you will see same thing. Same thing is called palindromic sequence like this. See, when you study from here, you will find that same thing will be present here. Okay, that thing is your palindromic sequence. Okay, the time of sequencing is called palindromic sequence. Now ahead we have some examples like this. Okay, GAATTC, and when you study this from here side, CTTAAG. Okay, this is called palindromic sequencing, which is present inside your DNA. Okay, in the DNA strand sequence of nucleotide, when read from fifth to third end in one strand and 3 to fifth end in the other strand, then it is same. Okay, same thing. From 5 to when you going to 3, GAATTC, and when you doing that thing in opposite direction, 5 to 3, then GAATTC. Okay, so this is called palindromic sequence reading. Okay, now next, how can we do formation of recombinant DNA? So simple, it is a simple step. Okay, you take a vector, okay, or a foreign DNA. Okay, now cut from here, okay, by the help of restriction endonuclease. Now you have a foreign DNA. Okay, put that foreign DNA on the place of this yellow band, and now this DNA will become your recombinant DNA. Okay, when this whole yellow circle is normal, then it is a normal DNA. When you incorporate this green strand in the middle of the some part of the DNA where we cut by the help of restriction endonucleases, then this thing will become your recombinant DNA. Okay, this example is also here. This is your cloning vector, okay, and this is your foreign DNA. Okay, you cut the cloning factor by the help of restriction enzyme, and you cut some part of the foreign DNA by the help of restriction enzyme. Now you will join this thing with the help of ligase enzyme, and at last you will get recombinant DNA. Okay, now next we have nomenclature of enzyme. Okay, there is enzyme, so you have to give the name to the enzyme. Okay, so first we have an example: eco R1. Okay, So as the nomenclature of enzyme says, the first letter of the name indicates the bacteria from the enzyme has been isolated. It is written in italics. Okay, so E, it is your Escherichia. Okay, next the second letter or second and third letter indicate the species of the bacteria. That is your coli. Okay, next the fourth letter indicate the strain of the bacteria. So it is your R strain. Okay, and the last one, the last letter is your Roman letter, which is a counting. Okay, so the discovery number, so it is ECOR1. So 1 means the category of this restriction enzyme, particularly this time it will be discovered. So we have second one also, ECOR2. Okay, so it's the second one, ECOR3, ECOR4, ECOR5. Okay, so this is discovery sequence, which is the noted as the last number. Okay, next, a restriction enzyme forms two types of end. Okay, your restriction enzymes forms two types of ends. One is sticky ends and one is your blunt end. Okay, see. When a restriction enzyme cuts your DNA fragment from the middle, then it forms blunt end. Okay, and when a restriction enzyme cut your DNA with the design of zigzag pattern, then it forms sticky ends. Okay, same thing is written here. Restriction enzyme cut the strand of DNA a little away from the center of the palindromic sequence. Okay, between the same two bases of the opposite strand, like this. So a restriction enzyme will cut like this. Okay. Now again a new DNA, foreign DNA will come of same coding and it will attach here easily by formation of hydrogen bond between these nucleotides. Okay. Next, blunt end or non-sticky ends. Some enzyme cleave both strand of DNA at the same nucleotide position, typically in the center of the recognition sequence, resulting in the blunt end or flush end, means like this. It will cut from here on the center of that sequence, then it forms blunt end. Okay, very blunt end, which can, in which we have to attach the that phosphodiester bond which is present here also, and the bond which is present in the between of them also. Okay, so this is your blunt end or your flush end. Okay, next, this is your factor DNA, this is your foreign DNA. Okay, it is cutted from here and it is cutted from here. Now it forms a sticky end like structure like this. Okay, it formed by these can stickily bond with each other. Okay, so this type of bonding is called sticky end bonding and it is created by your ECOR1. So there is some different types of restriction enzyme which forms sticky ends and some are there which forms your blunt end. Okay, so these are the examples of enzymes and these are the genus from that are produces. Okay, BAM H1 Bacillus amelia-lycophiasis. Okay, SAL1 Streptococcus albus. Next PST1 Provincium Suterius. Okay, next PVU1 Proteus vulgarius. Then CLAV1 Carophyra lactum. This is not coming in your CBSE exam, but still you have know these things. Okay, Hind3 Haemophilus influenza. Okay, and these all are strain does not cause any disease in human. Okay, next, these are the sequences which they cut in different areas and different phenomena. Okay, now next is synthesizing enzymes. Okay, these enzymes do synthesis of something. Okay, either they can produce any type of biomolecule, any type of protein, any type of carbohydrates. But in our in this chapter we are discussing related to the DNA. Okay, so we have two, reverse transcriptase and DNA polymerase. Reverse transcriptase क्या करता है? your DNA, that reverse transcripts convert your RNA into DNA. Okay, that is the work of reverse transcriptase. Okay, second we have DNA polymerase, means it will synthesize DNA from DNA. Okay, when an enzyme which perform to produce DNA from DNA, it is called DNA polymerase. And when enzyme perform formation of DNA from RNA, then it is called reverse transcriptase. It will going reversely. Okay, and this reverse transcriptase is present in your HIV virus. Okay, next we have joining enzyme. These are the natural welders. Okay, welding machines जो होती हैं वो natural. Okay, so this is your ligase enzyme, which is also called as molecular glue. Okay, so we have nucleus. This is nucleus, your molecular scissor type enzyme, which have two types: exonucleus and endonucleus. Exonucleus remove nucleotide from the ends, while your endonucleus, it remove the nucleotide from the center or from a specific sites. Okay, next we have what is the function to see. Normally, restriction enzyme does not see anything. Okay, it will cut DNA of everything. Okay, so your bacterial DNA is not safe from its own restriction enzyme. So how can that bacterial DNA survive in the presence of that restriction enzyme? The reason is methylation. Okay, methyl group is added inside the DNA of your bacteria, which make that make to do not cut the DNA of its own. Okay, see, when a bacteriophage come and inserts its viral DNA inside RNA or DNA inside the bacterium, so your restriction endonuclease will come and cut down the DNA inside the fragments. Okay, but simultaneously there is chance that restriction endonuclease can cut the DNA of your bacterium also, but that thing is inhibited by the help of methylation of its own DNA. Okay, which is not present inside the bacteria. Okay, that's how your bacteria survive with its own endonucleases. Okay, when this thing does not occur, then we can say that it is an autoimmune disorder type condition which happen inside the body of human. Your own immunity, this restriction and endonuclease in type of immunity present in the bacteria to kill, to cut down the DNA of that viral DNA. Okay, so this thing help to cut down the viral DNA and make your own DNA safe by the adding of methylations. Okay, next, which DNA? Vehicle DNA or your vector DNA. Okay, means the vehicle, the automobile from which you going to attach the DNA. Okay, the your foreign DNA, your desirable DNA, your alien DNA has been attached to some part of DNA, and that DNA from which you attach your alien DNA is called vehicle DNA or vector DNA. Okay, so the DNA used as a carrier for transferring fragment of foreign DNA into suitable host is called vehicle DNA. Plasmid and bacteriophage have the ability to replicate within the bacterial cell independent of the control of chromosomal DNA. Your bacterium contain two type of DNA. One is their main DNA, which is their chromosomal or nuclear DNA, which is present in the center of their cell, while there is other DNA also present inside the bacteria, which we call extra chromosomal DNA or plasmid. Okay, see, this is your bacterium, and this is the central DNA of bacterium, which we call nuclear DNA or main DNA. Okay, and some other DNA also present inside the bacterium, which we called extra chromosomal DNA or plasmid. Okay, So this plasmid, it used as a vehicle, as a vector, okay, in bacterium cases. So characteristic feature of a cloning vector, these are the some characteristic which is present inside your vector. First is ori, okay, origin of reason of replication, okay, that is your ori, ori. First, a desirable vector content origin of replication. Second, it should contain selectable markers. Third, high replicating rate. These are the some features which should be present inside your desirable vector. Okay, so ORI is that region particularly present inside a vector from where your process of that replication starts. So that thing is called ORI, or origin of replication. Okay, so it is a set of sequence which present on the vector from which replication of DNA start. Means the starting point of replication is called ORI. Okay, the desired foreign DNA which is isolated from the organism need to be integrated with the vector, and that integration is done on the ORI site. Okay, next thing is restriction sites or recognition sites. Okay, at the sites where your restriction enzyme work. In a plasmid, there can be 4 to 5 restriction sites like BAMH1, ECO-R1, ECO-R5, HIND2, BAS2. Okay, these all are different recognition sites which is present on your plasmids where your restriction enzyme come and cut that sites. Okay, next we have selectable markers. Okay, selectable markers are the particular sites which is present inside the plasmid which will you tell that DNA become recombinant or not. That's why it is called selectable marker. Okay, a marking type of scheme. Okay, so that thing is next. Okay, so this is your selectable marker. Okay, vector should have selected marker which help in identifying or eliminating non-recombinant from recombinants. Okay, selectable marker basically helps you to find that DNA become recombinant or not. Okay, that is the function of your selectable marker, and this is the plasmid which we call PBR322. Okay, in which this orange site is your tetracycline resistance site, in which if the gene is incorporated inside the tetracycline resistant site, then this plasmid does not survive inside the tetracycline medium. And this side is your ampicillin resistant site. If your foreign DNA has been incorporated in this side and the DNA become recombinant DNA, then this side will not survive in your ampicillin drug. Okay, so these both are your selectable markers which help to identify either the DNA is recombinant DNA or not a recombinant DNA. Okay, and these all are the properties of a good vector. First, replication autonomously means it itself get replicate, and then someone come and tell to them start to replicate, that system not there. Okay, so that is your replicator at autonomously. Next, should be able to carry the genome into the host cell. Okay, वेक्टर व्हीकल का मतलब क्या होता है, व्हाट इज द मीनिंग ऑफ व्हीकल, इट कैन कैरी समथिंग. ओके, so it should carry the genome into the host cell, then it is a good vector. Okay, next, less than 10 kb in the size. Okay, the nucleotide which is present inside that vector, it should be less than 10 kb. Okay, next, high copy rate. Okay, means the power of replication should be very high. Okay, easy to isolate and purify. Okay, you can easily isolate that vector from that original site, from that bacterium or from that organism, can do easily, then it a good vector. Next, should be able to integrate with DNA in which we are going to insert, means it does not start to do any activity over activity that foreign DNA, then it is a good vector. Next, easily introduced into the host. Okay, in the host it should be easily introducible, it can easily go inside the host body, then it is a good vector. Okay, next, suitable selectable marker, it should have suitable selectable markers which can easily perform its selections, which can easily tell you that DNA is recombinant or it is non-recombinant. And the last, unique target site for restriction enzyme, means it have unique sites where your endonucleases, restriction endonucleases should work. Okay, so next we have different types of cloning vector which is present in that biotechnology. Phage, okay, bacteriophage, lambda phage, different viruses, cosmids, transposons, plasmids, okay, is artificial chromosome that we going to study in molecular basis of inheritance. Bacterial artificial chromosome, it is again in molecular basis of inheritance. Shutter vector, okay, this is the special vector which is used in biotechnology. It will shuttering the different genomes simultaneously, okay, and this is the most common used vector in biotechnology, which is your plasmid PBR322, okay, and this is your E. coli vector PBR322, okay. Next, desired DNA, alien DNA, foreign DNA, or passenger DNA. Okay, the DNA which contains the phenotype, the genotype you want is called alien DNA, foreign DNA, passenger DNA, or desired DNA. These are the four different names of a single thing. Okay, so it is a DNA which is transferred from one organism into another by combining with the vehicle. Means it, you have a car, the car contains some passengers, okay, that car is your vector, and the passenger which is sitting inside the car is your alien DNA, foreign DNA, passenger DNA, or desired DNA, okay. So we have two types of passenger DNA. First one is your complementary DNA or cDNA, and second one is your synthetic DNA or sDNA. Complementary DNA is a DNA which is produced with the help of mRNA strand. Okay, template of mRNA is required to produce your complementary DNA or cDNA. It is synthesized on mRNA template with the help of reverse transcriptase. Okay, cDNA, it is produced with the help of reverse transcriptase enzyme. Okay, and necessary nucleotide cDNA formed through reverse transcription process is shorter than the actual or in vivo gene because of the absence of intron and non-coding regions. Okay, it does not contain any intron and non-coding region, it only content your exons. Okay, next is your synthetic DNA, artificial DNA which is made by human. Okay, so artificially synthesized with the help of DNA polymerase. So your cDNA, it is produced by reverse transcriptase, and your sDNA, artificial DNA, it is produced by your DNA polymerase. Okay, so Kornberg and Khurana, these two guys are the who synthesized this synthetic DNA. Okay, in 1961 Kornberg synthesized first synthetic DNA. Okay, and in 1968 Khurana synthesized first artificial gene or DNA without any template. They do not use template. Without template they produce DNA. Okay, next, host cell. Now the thing, that's the recombinant DNA you are going to insert in some kind of cell. That cell is your host cell. Okay, this cell is used for DNA cloning. Okay, host cell may be plant cell, animal cell, bacterial cell, or a fungal cell. Okay, so these are the different tools which is used in biotechnology. Okay, now we have the steps which is present in biotech. Okay, the major 6 steps which is used to produce for recombinant DNA. So first step is your isolation of DNA. Second step is your fragmentation of DNA by the help of and restriction endonuclease. Third is isolation of desired DNA fragment. Okay, then we have ligation of that DNA by the help of ligase enzyme. Then step 5, transferring the recombinant DNA into the host cell, and at last we have the culturing the host cell at large scale and extraction of desired protein, basically your downstream processing. Okay, so first we have isolation of DNA. Okay, it is a simple procedure in which we take different enzymes like your chitinase, okay, cellulase, and your amylase, lysozyme to isolate the DNA from a cell. Okay, so the DNA is enclosed within the membrane. We have to break the cell open and release the DNA with the help of different enzymes, like in animal tissue with the enzyme lysozyme, in plant it is cellulase, and in fungal cell it is your chitinase. Okay, other molecules can be removed by appropriate treatment as by purified DNA ultimate precipitate out after the addition of chilled ethanol. This can be seen as a collection of fine thread in the suspension. Okay, we have this experiment also in which we are going to isolate out the DNA of strawberry. Okay, next, DNA that separate out can be removed by spooling method. Okay, we have a wire like instrument in which we spool down that DNA fragment and take out that DNA fragment from that beaker. Okay, next we have fragmentation of DNA by restriction endonucleases. Okay, the molecular scissor, the restriction endonucleases are used to cut down the DNA we are isolated from the specimens. Okay, so restriction enzyme digest and performed by incubating purified DNA molecule with the help of restriction enzyme at optimal condition and with the specific enzymes. Okay, agarose gel electrophoresis is employed to check the progression of restriction enzyme digestion. Okay, there is method which is called electrophoresis, in which we see that DNA fragmentation take place or not. Okay, this is your apparatus for electrophoresis. Okay, there is one beaker which contains two different electrodes. Okay, positive side which is your anode and negative side which is your cathode. As you know, DNA is a negatively charged particle, so it moves from negative charge towards the positive charge. Now in that box we put a agarose gel sheet, okay, which is made up of gelidium glycineria secretion, that is your agarose gel, agar agar. Okay, now we put your DNA in the wells. Okay, DNA fragment which is done by your restriction endonucleases, and we put these DNA fragments inside the wells. Now we will start to give power supply in this, and when we get the power supply inside that wells, your DNA start to move from that negative side to the positive side. Okay, and when after that experiment done, your DNA reaches to that positive pole, we stain that thing by ethymid bromides. Okay, and that ethymid bromide give orange color in the presence of UV radiations. Okay, when we put UV radiation on the solution, we get orange colorations. Okay, this is the orange color which we get when we put ethymid bromide on that solution, on that box of the gel. Okay, so this is the steps, okay, that I tell. Well plus DNA plus restriction enzyme plus ethylene bromide a dye. Now next, DNA, that cut DNA fragment separate under the influence of electric field means a positive side and negative side. Positive side at that side where your DNA have to be reached, and negative side at that side where you put the DNA fragments. Okay, the negatively charged DNA move toward the anode means your positive charge, positive side. Large DNA fragments are present near the wells and smaller fragments move further away from the wells. Okay, as you know, in a race, if a fatty guy is there, he remain backside, while a thinner guy, it will run away. Okay, same thing will happen here. The big fragments of DNA, it remains at cathode side, while the smaller thinner fragments of DNA, it runs towards the positive sides. Okay, and that moving of DNA inside your agarose gel is called sieving movement. Okay, sieving movement, just like a needle crosses your stitching, when at the time of stitching when you use the needle, that type of movement is done by your DNA fragment inside the agarose gel. Okay, color, orange color bands of DNA can be seen when exposed to the UV lights. Okay, separate bands of DNA are cut out from the agarose gel and extracted for the gel. This is non elution, the process in which you separate out the DNA from that particular apparatus. That process is called elution. Okay, now the process of your electrophoresis is done. Now you will do PCR, okay, to increase the amount of the DNA. Okay, so next is your amplification of gene of interest by using PCR. This is your PCR machine. These both are PCR machine. The full form of PCR is your polymerase chain reaction. Okay, in which we use an enzyme which is your DNA polymerase, Taq DNA polymerase. Okay, which is extracted from a bacterium which is called Thermus aquaticus. Okay, now in the next slide we are going to tell the reason why we'll use that Taq polymerase, not DNA polymerase. The simple reason is that your DNA polymerase cannot withstand against high temperature, but Taq polymerase can easily withstand against high temperature. Okay, so also known as thermal cycle, your PCR is also called thermal cycle because in this process different temperature is used at different step. 3-step cycle it is, and every step you have different type of temperature. Okay, technique was invented by Carl Mullis in 1983, used for amplification of DNA and based on in vitro DNA replication and in vitro DNA synthesis. Mean the increase of DNA number, we will use this process. Okay, so requirements: apodor tube, next sample DNA, then 2 sets of DNA primer, small chemical synthesized oligopeptides, next all type of deoxyribonucleotide, DATP, DGTP, DCTP, DTTP for energy. Okay, next PCR machine, the most important equipment requirement for this process. If this thing is not there, then how can you perform the procedure? Okay, and its cost is 1 lakh or more than 1 lakh. Okay, a special type of DNA polymerase enzyme which is stable at high temperature, which we call Taq polymerase or thermostable enzyme. Okay, Taq polymerase, there are different polymerase which is used in PCR. Taq polymerase isolated from Thermus aquaticus bacterium. Next, you have Papilio polymerase isolated from Pyrococcus purifix bacterium. Next, we have Bent polymerase isolated from Thermococcus lithoris bacterium. Okay, these bacterium can withstand against 1000 degree of Celsius. That's why we are using thermostable enzymes. Again, I am saying that why we are using thermostable enzyme in PCR, because our DNA polymerase cannot withstand on high temperatures. These bacterium Taq polymerase, Vent polymerase, these are different polymerase can withstand against 1000 degrees Celsius of temperature. That's why we use thermostable enzymes. Okay, so this is the procedure of your DNA. So that amplification in DNA. Okay, so we have three steps for your DNA that PCR process. First we have denaturation, then we have annealing or cooling, and at last we have extension. This is the most high temperature process of the PCR, which runs on 94 degrees Celsius. Okay, it is the step of double strand DNA become a single strand. Means we are breaking down the hydrogen bond between the DNA and making a double stranded DNA into a single stranded DNA. Next, we have annealing, which is done on 54 degrees Celsius. In this step, two DNA primer are attached at three ends of the DNA which start doing replication. Okay, and that thing has been induced by your Taq polymerase in the process of extension. The simple process which we study in replication, same thing is happening here. The difference is that in the replication which we studied in molecular basis of inheritance, we use DNA polymerase, and here we use Taq polymerase. Okay, so the Taq polymerase enzyme extend the primer by using nucleotide provided in the reactions, and the genomic DNA template is ready to perform further process. Okay, in the process of replication of DNA repeated many times, the segment of DNA can be amplified to accumulate billion times. Okay, 1 billion copy can you made by the help of a single DNA which with this process. Okay, and this is the diagrammatic view of this thing. The first we have denaturing, in which we are separating the DNA at the temperature of 94 to 95 degrees Celsius. Then in annealing we are adding DNA primers at your third end of both the sides. Okay, and at last we have extension by the help of your Taq polymerase. We increasing that primer side. Okay, we're increasing the length of that primer side on your third to fifth end. Okay, next, this is again the diagrammatic view of your PCR. Okay, now application of PCR. Okay, there's different uses of PCR in today in medicine. Okay, first, to detect the gene sequence in infected agents. Okay, it will use to detect your viruses which is present inside your body during the disease time. Okay, it can detect the mutation that causes certain diseases. Okay, the cancers which is caused due to the mutations, okay, as can easily be detected by your PCR. Okay, next, it can detect genetic diseases in fetus. Okay, and it is used in DNA fingerprinting. PCR is also used in DNA fingerprinting to increase the count, the number of DNA sample which we get from the mother side. Okay, next, ligation of the DNA fragment into the vector by the help of your ligase enzyme. Okay, now you have the sufficient amount of DNA which you want to transfer into the host cell. Now you have to ligate that thing inside the vector with the help of ligase enzyme. That thing written here, that passenger and vehicle DNA is joined with the help of ligase enzyme. This gives to rise a recombinant DNA, rDNA, which we are transferred in, which we have to transfer inside the host cell. Okay, next, transferring the recombinant DNA into the host cell or gene transfer. There is two methods. First is indirect method and one is direct method. Indirect method of gene transfer in bacterial cell, in which we give heat shock and cool shock to your bacterium, due to which what happen, the cell wall, the cell membrane of bacterium become perforated, and you can easily insert the DNA inside, the recombinant DNA inside the bacterial cell. Second, in the plant cell, by the help of bacterium, your agrobacterium tumerofacium. Okay, this bacterium is also called as natural biotech engineer. Okay, biotech, the process of biotech has been influenced by this bacterium, your that agrobacterium tuberculosis. Okay, so that bacterium is used to transfer, use to give the DNA inside your host cell. Okay, you put the DNA inside your bacterium, okay, agrobacterium, and it will make the copies of that thing, and then you insert that thing inside your desirable host. Okay, see how this thing will work. This is the plasmid of your agrobacterium. It will contain two sites, which we the plasmid is called TI plasmid, tumor inducing plasmid. Okay, so it contains two sites which we call V site or V region. Okay, which we with the full form of V region is virulence region, and second part of this thing is your tDNA. tDNA tDNA or tumor inducing DNA. Okay, so we put your desirable DNA, the desirable thing, inside this TI plasmid. The desirable, the passenger DNA has been inserted this TI plasmid with the help of different procedure, and now your agrobacterium start to make more copies of that recombinant plasmid. Okay, basically what were you doing? You are decreasing the virulency, you are deleting the virulency, you are destroying the armory of that bacterium. Okay, the tumor producing power of that bacterium is deleted when we insert our desirable gene inside the TI plasmid. Okay, the V reason, the tDNA, does not get that power to perform tumor formation inside the plant body when we induce our desirable foreign passenger or alien DNA inside the TI plasmid. Okay, same thing is written here. Vir region contains genes which are essential for tDNA transfer and integration of host plant cell. This Vir region get incorporated inside the plant genome. Okay, and in plant genome it will start to produce tumor. Okay, next, when we use TI plasmid as a vector, first we remove the tumor causing gene, TI gene. Okay, because the TI gene is responsible for tumor. If we do not remove TI gene from that bacterium, then tumor will easily occur inside the plant body. So first we have to disarm the bacterium plasmid, then we can incorporate our desirable DNA inside that thing. Now your desirable DNA is ready, your desirable plasmid is ready. Now you can insert the desirable DNA, the recombinant DNA, inside the body of the plant, and it will start to give effects. Okay, this is that effect of that bacterium. Okay, tumor inducing bacterium, your agrobacterium causes these type of tumor inside the plant body. Okay, and this is the tDNA, the TI plasmid. Okay, tumor inducing plasmid of your bacterium, agrobacterium की ये plasmid है. This is the plasmid of agrobacterium which is responsible for development of these tumor inside the plant body. Okay, and in this you will find this is the tDNA region or your V region, virulence region. Okay, here is viral region and this is tDNA region which is responsible for developing tumor inside the bacterium. Okay, inside the plant from the bacterium. Okay, next we have direct method. Okay, now we are directly inducing that desirable gene, that desirable DNA inside the organism. Okay, inside the host. The examples, that methods are your electroperforation. Okay, it is created by the temporary pores. Okay, we are giving electrical shock to that particular organism, and due to which the cell membrane, the cell wall become porous, and you directly inject that desirable DNA, recombinant DNA, inside the organism, inside the host. Okay, second one is your chemical mediated gene transfer, in which we use PEG. Okay, polyethyl glycol. This is a form of chemical which is used to make your cell membrane perforated, and you will directly insert the desirable recombinant DNA inside the host cell. Okay, next one is micro injection. Okay, it is a micro type of injection which is very minute. Okay, you cannot see the tip of that injection by your neck dice, you have to see that thing with the help of microscope, and that injection is inserted inside the cell of the host cell without damaging any cell organelle, and you inject that desirable, that recombinant DNA inside the cell easily. Okay, next one is your particle gun, ballistic gun method, or gene gun method. In this technique, what will you do? You will take small gold particles, and you will incorporate that thing with the help of that recombinant DNA has been coated on that gold particles, and you do shooting of that thing in the cell. Okay, host cell. Next is your liposome mediated gene transfer. It is the most recent use of that transferring recombinant DNA inside the host cell, in which you take bags of liposome, and that bag has been inserted and will go inside the cell, because it is made up of lipid, and your cell membrane, it is having lipid part, so it will easily get inside your cell, and it will, when it go inside the cell, it will blast down, and your recombinant DNA will get inside the genome of the organism. Okay, so these all are the direct methods of inducing the recombinant DNA inside the organism body. Okay, so this is your gene gun. Okay, that's how this gene gun work. Okay, in which you have the particle, this in this plate you have gold particles. Okay, and when the pressure applied on this, that recombinant DNAs get coated with that gold particles, and that gold particles go inside your cells, targets the organism site. Okay, next we have micro injection. This is your micro injection. This injection directly injected inside the nucleus, carrying the recombinant DNA. Okay, and this is your PEG, polyethyl glycol. Okay, which is used to perforate the cell membrane, nucleus membrane, and that you can induce your recombinant DNA inside the host cell. Okay, next is your selection of transformant with recombinant DNA. Okay, now you have to identify which DNA is recombinant, which DNA is not recombinant. Okay, so we have two methods. First one is your tetracycline and ampicillin resistance method. And second one is your beta galactosidase method. Beta galactosidase white blue colony method. Okay, these are the two methods by which you can easily identify that DNA is recombinant DNA or not a recombinant DNA. Okay, so selection by two antibiotic resistant genes, your tetracycline gene and ampicillin resistance gene. Okay, if a bacterium which get transmitted, which get rDNA inside the tetracycline gene, it will not grow in tetracycline, because you cut down the connection of tetracycline. Okay, just like for example, you have a wire carrying electricity. In the middle of the wire, you add a resistance or a insulator, then the electricity will not flow from this point to this point. Just like same thing, it is your this is your tetracycline gene. Okay, you cut this part and add desirable gene, foreign gene or anything. Okay, now your bacterium plasmid is not going to be survive in tetracycline, because the continuity of the gene has been destroyed. The tetracycline gene has been destroyed, and it will not grow inside your tetracycline medium. Okay, on the other hand, you have ampicillin gene. Okay, and you incorporate your for desirable DNA in it. Now your ampicillin resistance will be lost, due to which you cannot continue to produce ampicillin resistance inside the bacterium. Okay, that's how you do selection by the help of two antibiotic resistance genes. Okay, next thing, this is the example. Okay, plasmid are cut. Okay, this is can resistance. Okay, canacycline, it is an antibiotic which is present in bacterium also. Tetracycline, canacycline, ampicillin, these all are different resistance genes present inside the bacterium. So in this example, I have taken canacycline. Okay, and this is your tetracycline. Now this is your E. coli plasmid, which containing canacycline and tetracycline both. Okay, now you will cut these both and join these both. Okay, now this bacterium can resist they can easily grow inside both mediums, okay, because it have resistance against the both thing, okay. Now this is the again the same thing, okay, in which we have put the canacycline resistance gene fully and tetracycline gene fully, no E. coli doubtfully resistance, okay, because this content ampicillin, okay, something other will be contained inside this medium and these bacterium does not get grow inside that, but in this it will grow easily because E. coli are grown in the medium containing kanamycin and tetracycline. Each colony is cloned for millions of the cell, all of derived from the single bacterium, okay. It can easily grow inside this colony because it contains the resistance against your kanamycin and tetracycline. Both genes resistance are present inside your bacterium, so it can easily grow inside that thing, okay. Next, selection by the leg Z gene or antibiotic resistance gene, okay. Leg Z gene means your beta galactosidase genes, okay, in which it will give the coloration. This method is more appropriate than your tetracycline resistance gene medium experiment, because in this medium you do not have to take different petri dish. In one petri dish you can easily distinguish between the recombinant one and non-recombinant one, okay. This is that your lag chaperon which you study in molecular basis of inheritance. So they see what happened, okay. First we have ligation, okay. This is your lag Z gene in which you are going to incorporate your desirable DNA, okay. Now this thing is your inserted carrying vector which is your recombinant DNA, modern DNA, and this is the insert less vector, okay, normal vector, and this is your recombinant vector, okay. In this DNA, foreign DNA is added. Now this vector is become recombinant and this is non-recombinant, okay. Now when you grow this thing, okay, the bacterium which contain this desirable gene, the recombinant DNA one, it does not give the color, while the bacterium which does not contain any recombinant DNA, does not contain any foreign DNA, it will give color, okay. So this thing will easily distinguish between recombinant and non-recombinant DNA, acts gel plate with antibiotic. In this we also use antibiotic also, ampicillin gene also be used in this method also, okay. Next is your obtaining the foreign gene product, okay. Again same method, okay. The recombinant DNA is inserted into the bacterial cell, plant cell or animal cell, then the foreign DNA is multiplied. The recombinant DNA should produce the desirable protein after the cloning of the gene. Optimum condition should be maintained for the production of protein, okay. Means these are the processes how can you obtain the product from that recombinant DNA. So you have to provide the profitable condition to that, normal pH, controllable pH, normal temperature, the temperature that DNA want to divide, all these things you have to be required, you have to give, then that thing will grow easily inside that cases, okay. Next thing is your bioreactor or fermenter. Now you have to multiply that thing more, okay. That thing will be done with the help of bioreactor and fermenters. These are the large vessels in which you will multiply, you will increase the quantity of your biologically modified products, okay. These can be that it can be of metal and it can be used to produce antibiotics for fermentation processes, okay. So large vessels in which the raw material and biologically converted into the product by the microbes, plant and animal cell or enzymes is known as bioreactors, okay. Bioreactors are of two types: simple stirred tank bioreactors and second one is spray stirred tank bioreactors, okay. Simple stirred tank bioreactors, these are the simple type consist stirrer. They have a simple stirrer, okay, which is present in the center of that bioreactor which do stirring, okay. But in this Spraggett stirred tank reactor we have some fan type of mechanism which is present on the base of the bioreactor, which continuously give oxygen gas, and that oxygen bubble which is present inside these bioreactor increases the surface area, and due to that increased surface area your bioreaction that processes will easily continue, your process will increase, the level of the process is rapid, okay. The product which is produced in Spraggett stirred tank bioreactor is very fast, while in the simple stirred tank bioreactor it is very slow. This is used for making different alcoholic beverages, and this is used to produce vaccines by antibiotics on large scale. ये भी large scale में use होता है. This is also used on large scale, but it's used to produce alcoholic beverages and different materials, okay. And this is used to produce antibiotics and vaccine because we need that substance very rapidly. And in this thing we do not need that alcoholic beverages at that much fast rate, okay. So that's why we use this tank, this bioreactor to produce alcohols. Okay, and this thing is used to produce your antibiotics and your vaccines. Okay, this is your simple stirred tank bioreactor, and this is your packet stirred tank bioreactor. Okay, in this you will see these are the oxygen bubbles which increases the surface area to do that biocatalytic reactions. While in this there is no bubble, there is large fans are there, means stirrers are there which do revolution, and due to that revolution the substance which is present inside this bioreactor mix up. In this, that thing is done by oxygen bubbles, gas bubbles. Okay, now next is your downstream processing. Okay, after the product has been made in the bioreactor, it need to be processed before it marketed. Okay, before that thing go inside the market, you have to do testings. Okay, you have to perform different procedure on that. You have to increase the potential of that product, and this thing is done by your downstream processing. Okay, the process includes separation and purification of the product before it's marketed, and this together known as downstream processing. Means you are separating that material from bioreactor plus purifying that material, then this process is called downstream processing. Okay, after downstream processing, many clinical trials needed to be done, so that product is checked before making its commercial. Okay, many tests on rats, on monkeys, on dogs, on cats has been done to test that material has been useful on humans or not, and at last checkpoint you will test that thing on human. Okay, so this is the procedure. Okay, intracellular product, then cell distribution, removal of the insolubles, then isolation, then purification, and that finishing, then at last you get final product which is sale in the market. Your vaccines, your antibiotics has been go through these all processes. Okay, now next chapter, the second chapter is completed. Next is your biotechnology and its application. Okay, till now we see that how biotech works. Okay, now we have the application level. Now we have to apply that biotech in the form of examples. Okay, so biotech can be applied in biopharmaceuticals, thermaceuticals, diagnostic, genetically modified crops, processed food, bioremediation, waste treatment, and energy production. These all are different uses of your biotechnology in different fields. Okay, again, biopharmaceuticals means production of drugs. Okay, therapeutics means development of different methods of the disease findings. Okay, diagnostics, disease finding rates. Okay, genetically modified crops means your transgenic animals and transgenic plants. Okay, processed food, all the food which you eat in your cafes, junk food, all these things are all processed food. It is also produced with the help of biotech. Next, bioremediations to check that all the danger signs which is present in your biodiversity, all the things just like oil spills, all these are checked with the help of different microbes, and these microbes are biotechnological modified, just like your Pseudomonas pudica. Okay, that is used for bioremediations. Okay, waste treatment, as you know, in that water treatment plant we use different microbes. Now we are using modified, genetically modified microbes which will eat plastic. Also, the some fungus has been created and some fungus are discovered which can eat your plastic. So in waste treatment you will use biotechnology. Next, energy production, biogas plant. This again use genetically modified methanogen to increase the productivity, to increase the rate of fermentation, to produce more biogas from your waste products. Okay, so introduction of biotechnology. Biotechnology deal with the industrial scale production of biopharmaceuticals, biological, using genetically modified microbes, fungus, plants, and animals. Different genetically modified organism is used for human welfare. That is the good use of biotech. Okay, application of biotechnology include therapeutics, diagnostics, genetically modified crops for agriculture, processed food, bioremediation, waste treatment, and energy production. These all are different uses of biotech in different fields on earth. Okay, सो बायोटेक्नोलॉजी एप्लीकेशन जितना भी बोला सब कुछ वही है वापस. ओके, थ्री क्रिटिकल रिसर्च एरियाज ऑफ बायोटेक्नोलॉजी provide best catalyst in the form of improved organism using a microbe or pure enzymes. Okay, that bt toxin, nematode resistant plants. Okay, insulin, huminilin. Okay, ada deficiency, that all things come under your biotechnology advancements. Okay, create optimal condition through engineering for catalyst to act Okay, now there should be some conditions in which your catalyst should work. So that condition can also created by biotech also. Okay, and last, downstreaming processing technology to purify protein and organic compounds. Okay, means producing that product on large scale. That is your downstream processing. Okay, so application of biotech in agriculture. That's your BT cotton and nematode resistant tobacco plant. Okay, so agrochemical based agriculture, organic agriculture, genetically engineered crop based agriculture, these all are the fields which come under biotech of agriculture. Okay, so genetically modified organism, GMO, okay, are the plants, bacterium, fungi and animals whose genes are altered and manipulation by humans to produce more efficient products. Okay, so these are the top 10 genetically modified food which we use daily. Soybean, okay, tomatoes, papaya, pea, potato, corn, rice, canola oil, cotton, seeds, and dairy products. These all are genetically modified material which we drink, which we eat daily. Okay, so these are the advantage of genetically modified plants. Okay, what are the advantage? What are the good thing which we get when we use genetically modified plant? First, tolerant to abiotic stress means that plant which are genetically modified, they can easily withstand against high temperature, low temperature, high pH, low pH, high salinity of soil, low salinity of the soil. Okay, different abiotic forces, sunlight, temperature, soil, water, air, they can easily withstand against these abiotic forces. Second, pest resistance crop by reducing dependency on harmful chemicals. Okay, by using genetically modified crops, we are avoiding pesticides, chemicals which causes destruction of our ecosystem has been reduced. Okay, second, reduce post harvest loss. Okay, see, when the crops are not genetically modified, they are less genetically modified or they are not genetically modified, there is chance of post harvest loss. Means when you cut that thing, it will cut very fast. Okay, or the time of ripening of fruit is 2 months, it is ripening in 1 month, then it is called post harvest loss. Okay, so these genetically modified crops particularly get harvested on a particular time which should be harvested. Okay, so it avoid reduce post harvest losses. Enhance nutrition value of food, for example golden rice which is vitamin A enriched. Okay, we can increase that content of that genetically modified food. We can increase the fertility of that food by inducing different minerals with different vitamins, different nutrients in that food. That thing is called biofortification. Okay, inducing out from outside, you are introducing different vitamins, different minerals, different nutrient inside that crop. That process is called biofortification. Okay, then increase ability of mineral uses by plant. Okay, see, soil is full of nutrients, but it is on your plants how they can take that thing. Okay, so genetically modified organism, genetically modified plants, they can easily take that material, that nutrient from soil which is present inside the soil. Okay, next we have green revolution. Okay, green revolution is the revolution which is induced inside the India to increase the productivity. Okay, to increase the quantity of food, to increase the reservoir which is present in India. It is done, it is called green revolution, in which we increase the fertility, we increase the productivity of the crops. Okay, different revolutions are there. Green revolution for plants, white revolution for milk, silver revolution for egg, red revolution for red meat. Okay, black revolution for petroleum, coal. Okay, so these are different revolution take place at different times to increase the productivity of these material in the nature. Okay, so we have pest resistant plants. Okay, pest resistant plants act as a biopesticides. It reduces the need of insecticide. Example: BT cotton, BT corn, rice, tomato, potato, soybean. All these are BT crops. Okay, BT cotton, BT tomato, BT corn, BT potato. Okay, these all are called BT crops in which we introduce the gene of Bacillus thuringiensis. Okay, so first we have genetically modified crop, your BT cotton. Okay, that cotton in which we introduce the gene of Bacillus thuringiensis, the BT toxin gene has been indeed introduced inside the crop. Okay, so to what are the need to enhance the genetically modification inside the crop? First, to enhance the nutrition quantity of the food, example golden rice, vitamin A enriched. Okay, next, made crop to mold tolerant to abiotic forces like cold, drought, salt or heat, इन सबसे बचाने के लिए. Okay, next, help to reduce post harvest loss, example flavored savor tomato. This is a transgenic species of tomato which you can taste the tomato even after the cut. Okay, it contains large number of anthocyanin inside this. Okay, this is your flavored savory tomato. This dark red one is your flavored savory tomato, and this red one is your normal tomato. Okay, so flavored savory tomato, transgenic variety of tomato, pectin degrading enzyme, the enzyme which degrade the pectin which is present inside the tomato is reduced, due to which the life longevity of your tomato increases. Means if a tomato getting degraded in 2 days, now it will degrade in 5 to 6 days. Okay, that increase, that thing is your flavor savor tomato, means it is saving the flavor of your tomato. Okay, next, increase efficiency of mineral uses by plant. Okay, again I am saying that soil contain all minerals, but it is on your plant, how can it using that mineral? Okay, so genetically modified plants use the mineral which is present in the soil very adequately. Okay, next, to produce biopharmaceutical products, example production of hirudin. Okay, hirudin, it is produced by your rephus, means your radish. In the radish seeds we have introduced the genes which can produce hirudin. Okay, hirudin, it is present in your an organism from annelida, that is your hiridonuria. Okay, hirudin. So hirudin is a protein that prevent blood clotting. Okay, means it will stop the blood to clot. Okay, so the gene encoding hirudin was chemically synthesized and transferred into brassica nephis. Now the seeds of brassica nephis, mean the seeds of your radish or your mustard plant, is start to produce this gene inside your body. When you take, means hirudin is now producing by plant and you can take that thing inside your body. Okay, next, to produce herbicide resistant plant, example first transgenic plant was tobacco. Okay, tobacco is the first transgenic plant which is produced with the help of biotechnology. Okay, it contains resistance genes against herbicides. Okay, glyphosate is an herbicide which is used to kill unwanted plants. Okay, but sometimes that herbicide can also destroy the desirable plants. So this tobacco plant contain the gene which is resist the activity of herbicide inside them. Okay, and at last we have pest resistance crop that reduce resilience of chemical pesticide, which will make your ecosystem safe from the chemicals. When that chemicals flush out from the soil, it will start and go inside the water bodies, so it increase the toxicity inside the water bodies also. So dependency on genetically modified crop increases the loss of ecosystem by these chemicals like biopesticides, like chemical pesticides and chemical fertilizers. Okay, so first we have your bt cotton. Okay, bacillus thuringiensis cottons, or the cotton which is resistant against some insects called bomb. Okay, called bollworms or other that cotton borers. These are different of insects which will eat your cotton. So by introducing bt gene inside the cotton, it will reduce the damage to the cotton plant. Okay, so these are the insects: lepidopteran, celopteran and diphtherians. Okay, these all insects damage your cotton field. Okay, that will destroy the cotton crop inside the cotton fields. Okay, in the lepidopteran we have tobacco budworms and armyworms. Then in celopterans we have beetles. Okay, and in diphtherians we have flies and mosquitoes. These all things destroy the cotton production. Okay, so for rescuing this thing we introduce bt gene inside the cotton plant. Okay, bacillus thuringiensis gene inside the cotton plant, due to which now the gene is been introduced, it will produce by your cotton plant itself. Now these insects will come eat that cotton and die. Okay, because the toxin which is producing by your cotton, it will kill that insect by introducing, by going inside the gut of that insect. Okay, your insect gut is alkaline in the pH. Okay, and bt toxin get activated when it get alkaline pH. Okay, so when that bt toxin get inside the gut of your that insects and it get that alkaline pH, it will start to do perforation inside the gut of your insect and that insect will die. Okay, that is the introduction of Bt toxin inside your in the cotton plant. Okay, it contained a gene which is called cryogene. That cryogene is responsible for the production of Bt toxin. Okay, and there's different varieties of cryogene, that is cry 1, cry 2, cry AB2, cry AB23. Okay, these are different varieties of cryogene which is produced by your cottons plant. Okay, see, cry 1 AC, cry 2 AB, cry 1 AB, cry 3 AB, and cry 3 BB. Okay, this is for your control cotton bollworm. This cry 1 AB is for your corn borers. This cry 3 AB is responsible for your potato beetles. Okay, and cry 3 BB is responsible for the root worms. Okay, means we develop a technique in which we are specifically killing a specific organisms. Okay, means in the market these all different Bt resistance, so that Bt cotton is available which contains this gene only. So it will kill only bollworms. Okay, it will kill only corn borer. It will only kill your potato beetle. It will only kill your root worms. Okay, so this is a toxin which is synthesized inside the body of the plant to kill that arthropod. Okay, next, this is the procedure how this thing will act. Okay, Bt toxin protein. Okay, it is an inactive protoxin. It will ingested by your insect. Now it is converted into active form because the gut of your insect is an alkaline in pH. Okay, now it binds with the surface of the midgut. Okay, it start to create pores, and after that lysis and swelling of the gut take place and the death of the insect take place. Your crop is safe from the insects. Okay, so one thing is there according to this, the enemy of my enemy is my friend. मेरे दुश्मन का दोस्त मेरा दोस्त होता है. Okay, that thing. Okay, the enemy of my enemy is my friend. मेरे दुश्मन का दुश्मन मेरा दोस्त होता है. Okay, so cotton plant, cotton bollworm and Bacillus thuringiensis. Okay, it is the enemy of your cotton plant, and cotton bollworm enemy is Bacillus thuringiensis, and Bacillus thuringiensis friend is cotton plant. Okay, next is your nematode resistant plant. Okay, the plant with the tobacco plant which is resistance against nematodes. Okay, nematodes belongs to your class of animal kingdom in which you have that platyhelminthes. Okay, flatworms. Okay, then several nematode parasite are widely variety of plant animal include human beings. Okay, human beings की body के inside the human being body they also do infections of different types. Okay, a nematode Melangiana inguinata infects tobacco plant. It will go inside the root of the tobacco plant and induce the growth inside the root, and due to which the root get dissolved up. Okay, so a novel strategy adopted to interface this nematode, that is your RNA splicing theory. Okay, in which we cut down the RNA, the mRNA which is producing inside the nematode, due to which the toxin of nematode is produced. It's cut down, and now your nematode cannot produce the toxin inside the body, and your tobacco plant is safe. When when the nematode eat the tobacco plant, that gene go inside the nematode body, and uh, so nematode specific gene introduced into the host. Okay, plant using that agrobacterium tubuciferum is used to introduce the gene. The production of sense and antisense RNA in host cell, two RNA being complementary to each other and bind to the double-stranded DNA. Okay, means gene is introduced inside the DNA, double-stranded DNA of your nematode. Now it will initiate RNA interference. It will form that RNA. Okay, mRNA has been formed. Now this mRNA will incorporate inside the genome of your nematode and start to doing cutting the mRNA of nematode, due to which your nematode cannot produce specific proteins for its growth, and after some time that nematode will die. Okay, same thing is written here. Initiation of RNA interference, due to which double-stranded RNA cleave into 21 to 23 segments, which we call siRNA, small interfering RNA, using enzyme dicer. इसमें एक enzyme use होता है, that enzyme is called dicer enzyme. Okay, so siRNA plus ribonucleosides forms an RISC. This is RNA induced silencing complex. This complex cut down the mRNA of nematode, and due to which your mRNA does not produce inside the nematode, and nematode cannot perform normal metabolic activities, and after some time your nematode will die. Okay, next is by using this is the processor again. Next, the double stranded RNA. Okay, then dicer binding take place. Okay, siRNA form. When the dicer is binding with your double stranded RNA, your siRNA is formed. Okay, single stranded RNA. Okay, now this siRNA get attached with RISC. Okay, then RIC do unwinding of that double stranded RNA, and due to which degradation of this double stranded RNA take place, and it will cut down into several small particles, and now your nematode cannot produce specific proteins inside the body, due to which your nematode will die after some time. That process is called RNA interfering. Means you are interfering the procedure of RNA formation inside the body of nematode. Okay, now next is your application of biotechnology in medicine. Okay, in medicine, how will you use biotechnology? Okay, the different applications of biotech in your in your medicine stream. Okay, so first we have and genetically engineering insulin, then we have gene therapy, okay, vaccine production, and at last we have molecular diagnosis, that is your ELISA. Okay, so at present about 30 recombinant therapeutics has been approved for human use in the world. Okay, 30 different genetically modified therapeutic procedure has been used for humans in the world. Okay, so in India 12 of these are presently being marketed in India. Out of that 30, 12 has been used. Okay, so first we have insulin, genetically engineering insulin or humulin. Okay, it is the first genetically modified insulin which is incorporated inside the body of human to face the disease diabetes mellitus. Okay, so this insulin, previously what happened, that you insulin is being getted from the slaughtered animals, that pig. Okay, from the pig pancreas or pig body we get the insulin. Okay, and that insulin can causes allergic reaction inside the body of human. So to prevent, to end this procedure, we start to produce our own insulin by the process of genetically modification. That thing is called humulin. Normally inside your insulin 51 that thing is present. 51 peptide bonds are present, 51 amino acids are present. Okay, now inside normal chain, which we call proinsulin or a it is a previous insulin, or it is a non-functional or inactive insulin. It will contain A plus C plus B, three chains are present. Okay, now what we do, we will remove this C and join A and B. This is your proinsulin or inactive insulin. Okay, and this is your active insulin, which containing only A chain and B chain. C chain is deleted from the proinsulin inside your body normally, and you join A and B chain with them, and this will form your active insulin. Okay, this is the normal procedure of conversion of proinsulin into active insulin. Okay, same thing is there. This is your B chain, this is your C chain. Okay, and somewhere side you have A chain also. Okay, now this A and B in the middle of this C chain is present. Okay, now by the help of different cleaving enzyme, this C chain will be degraded, and again your A chain is being and B chain is joined together by the help of disulfide bonds. Okay, and this will form your active insulin. Okay, So this is your three types of insulin which is used in daily basis. Uses humulin 70 by 30. It is humulin R, which is also called recombinant humulin, and this is humulin N. Okay, and nanotechnology based humulin. Okay. So insulin consists of two short polypeptide chain A and chain B, that is linked together by disulfide bridges. Okay, means if this is your disulfide, means this is your A chain and this is your B chain, and it is connected by a bridge. This bridge is called disulfide bridge. Okay, this is the connection. Just like in the DNA, hydrogen bond is present to connect your two strands. Same thing is present in your insulin, that is called disulfide bridge, which connects your A chain with the B chain. Okay. In mammals, including human, insulin is synthesized as a prohormone. Okay, prohormone means inactive hormone. Like a proenzyme, the prohormone also need to process before it become fully mature and functional. Prohormone or proinsulin, it is non-functional. It does not act on your sugar. Okay, inside your blood. But then that seed chain is been deleted, then it will become into convert into a proper working insulin, which we call active insulin. Okay, or mature insulin. This C peptide is not present in the mature insulin and removed during the maturation of insulin. Now your insulin, when the C peptide is removed from the insulin, it will become a mature insulin. Okay, so this thing we will process biotechnology also with the help of biotech also in the bacterium E. coli. Okay, in E. coli bacterium we can produce insulin by the help of biotechnology. Initially we taking insulin from the slaughtered animals like pig. Okay, which causes that different allergic reactions, but now we producing insulin with the help of biotechnology. Okay, so see what will we take. We take insulin gene from human DNA. Okay, and a bacterium plasmid, and we incorporate that insulin gene inside the plasmid of bacterium. Now we will introduce that recombinant DNA inside the bacterium again and make that bacterium divide into the downstream processing, and after that we purify the product and take the product, and that end product is your modified insulin. Okay. Next is your gene therapy. Okay, gene therapy, in which we incorporate different gene inside the body of human to fight against different diseases. Okay, in our course we have ADA deficiency or SCITs. Okay, gene therapy is a set of way through which gene defects in the embryo of child can be corrected after diagnosis. Okay, after diagnosis you can check the deficiency of the gene and introduce the gene, but this thing should be done in the embryonic stages. When the embryo or fetus become adult, you cannot do that thing. You can, if you do that thing, you have to replace that thing every time. Okay, so it is best to done in embryonic stages. Okay, so first clinical gene therapy was given to 1990 to a 4-year-old child who is suffering from adenosine diamycete deficiency, ADA or SCIDS. Okay, SCIDS full form is severe. What is the full form SCIDS? Hmm. Severe combined immunodeficiency syndrome. Okay, it is the full form of SCID, which is happen due to the deficiency of particular enzyme which is called AD enzyme. Okay, adenosine diamynase enzyme, which is responsible for the maturation of your T lymphocytes. Okay, if this enzyme is not synthesized inside your body, your T lymphocyte does not get activated. Your T lymphocyte does not get matured. Okay, so this disorder caused due to the deletion of gene for adenosine deaminase. This disorder in some can be cured by bone marrow transplantation. Okay. In other, it can be treatment with the enzyme replacement theory, in which functional enzyme ADA is being injected into the patient, but it is not successful because after some time that enzyme will be decomposed or get deleted from your body from the use. Okay, so this enzyme is introduced inside your body during the embryonic stage of development, then this therapy is successful. Okay, so this is the procedure. Patient with ADA deficiency, lymphocyte is taken an in vitro culture of lymphocyte is given by the help of your retroviruses. This harm retroviruses vector. Okay, now lymphocyte with functional gene is introduced inside the body of that particular human which have the deficiency of ADA. Now your human is having that less deficiency of that T lymphocyte, but after some time that lymphocyte will be degraded. Okay, so you have to again attain that gene therapy. Okay, so this is the Asta de Silva, the first clinical gene therapy patient, 4-year-old girl, 1990. Okay, next is your molecular diagnosis. Okay, PCR, polymerase chain reaction. Okay, the same thing we studied in the biotechnology process. Okay, PCR is also used to detect different viral tests, viral infections, just like in corona. Okay, rapid PCR. Okay, in which we use that nasal swab technique, which we take the swab from the neck and do the PCR technique. Okay, so PCR is also used to do diagnostic procedures for different diseases. Presence of pathogen in normal is suspected only based on the symptoms. By the time the concentration of pathogen is already very high in the body. Okay, when you get to find that you have the symptoms of particular disease, in your body the pathogen number is in millions. Okay, so previously you will do PCR to detect the quantity of that virus inside your body. So we do PCR to amplify the number of our body, that pathogen which is present in our body, to find the number of that pathogen. The quantity of pathogen is specially done to recognize with the help of PCR. However, very low concentration of bacteria or virus can be detected by amplification of their nucleic acid by PCR. Okay, so your PCR procedure is done to amplify the genetic material which is present inside that particular bacterium and virus. Okay, so this is your gradient thermal cycle PCR touch screen machine. Okay, in which you embed the sample by this tube, and this will insert it inside the machine and start doing the PCR, increase the amplify the number of DNA of that particular sample. Okay, so to detect HIV in suspected AIDS patient, to detect mutation in the gene in the cancer patients, and to identify many other genetic disorders, we use PCR. Okay, a single strand of DNA or RNA tagged with a radioactive molecule probe is being hybridized to complementary DNA in a clone of cell. It is followed by detection using autoradiography. Okay, to find you that DNA is a radioactive, you have to do autoradiography. In autoradiography, what we will do, we will bombard X-rays on a particular X-ray sheet which contain that radioactive probe, and then we got the print of that X-rays on the X-ray sheet which contain the DNA. Okay, same technique is being used in your DNA fingerprinting also. Okay, next is your enzyme linked immunosorbent assay, ELISA. Okay, this ELISA is also used as a diagnostic aid. It is then that satisfactory test or starting test for AIDS. Okay, it will done to detect either the person is HIV positive or not. Okay, so it is based on the principle of antigen antibody interaction or antigen antibody reaction. Okay, it can detect even small amount of protein with the help of enzymes. Next, infection by pathogen can be detected by the presence of antigens. Okay, different antigens are present by produced by your pathogen. These antigen will react with the antibodies, and that antigen antibody reaction tells you what type of pathogen is present inside your body and the concentration of that pathogen inside your body. Okay, like proteins, glycoproteins, by detecting the antibody synthesis against the pathogens. Okay, the antibodies which is synthesized against the pathogen means they read the antigen, and that antigen produce antibody, that antigen is read by your antibody and it will give you immune response. On that we will distinguish, we will count the number of different pathogen inside that. That is the testing of ELISA. Okay, ELISA के तीन टाइप होते हैं. Okay, ELISA is of three types. First one is your normal ELISA, second one is your indirect ELISA, and third one is your sandwich ELISA. Okay, next is your transgenic animals. Okay, animals whose DNA has been modified to express the foreign DNA is non-transgenic animals. The transgenic animals are the animals which is produced by humans to do and test different procedure and to test vaccines, antibiotics, and different materials. Okay, so step of producing a transgenic animal: first identify and isolate the desirable gene what you want to isolate, what gene you want to insert inside that animal. Okay, first select that gene. Then selection of the vector in which you put that desirable gene. Okay, now insert that desirable gene into the vector with the help of enzyme ligase. Okay, means you are preparing recombinant DNA. Next, introduction of vector into the cell, tissue or embryo by direct or indirect gene transfer technique. After that, express the foreign gene into the transgenic animal and your transgenic animal is ready to use. Okay, so this is the uses of transgenic animals: normal physiological growth and development, study of diseases, biological products, vaccine safeties, chemical safety testing, and replacement of defective organs. These all are different uses of transgenic animals and transgenic plants. Okay, these are your transgenic animals: mice, rabbit, sheep, pig, cow, fish. These all are transgenic animals. This is the first transgenic cow, Rosie. Okay, which milk content alpha trypsin enzyme. Okay, and it will contain natural colostrum also. Okay, so this is the first transgenic cow. Okay, Rosie. Now ethical issues. Okay, problems of unpredicted results. Okay, now sometimes biotechnology can produce irrelevant results, the result which you do not want. Okay, some just like some pandemics, some unwanted animals. Okay, some aggressive varieties, some aggressive species of particular animals can also produced by biotechnology. So that thing is the problem of unpredicted result, because we cannot predict the result of biotech. What we are getting from that particular experiment we cannot know. Okay, so that can produce different hazards, different effects on it. Okay, so genetically modification can cause unpredictable result when such organisms are introduced into the ecosystem. They can degrade the ecosystem also. Basically, biotech is used to increase the efficiency of the particular organism, the product which is produced by that organism. But sometimes that thing reverse. Okay, due to that reverse action, it will damage the ecosystem when it is releases inside the ecosystem. Okay, so Indian government has set up an organization like GEAC, Genetically Engineering Approval Committee. That committee is only the way you can introduce the genetically modified organism inside the ecosystem. Without taking permission from GEAC, you cannot produce any genetically modified organism. You cannot put that genetically modified organism inside a ecosystem. You have to first take the permission from GEAC, then you will use that organism inside the field. Okay, which makes decision about the validity of GM research and the safety of GM organism for public service. Okay, now next is your biopiracy. Okay, now some countries, some particular companies thieve the ideas which is produced by different countries. Okay, that thing is called biopiracy. It is the use of bioresources by multinational companies and other organizations without proper authorization, without proper permission from a particular country. Okay, just like neem, tulsi, haldi, all these things produced in India. Okay, it will establish in India, but some other countries taken that from India and start to sell in their countries. Okay, so that thing is called biopiracy, when you are having the substance, that product from some other country, and it been used without any permission, then it is called biopiracy. Okay, Basmati rice has unique aroma and flavor. It is an Indian species. Okay, India has 27 varieties of basmati. Okay, in 1997, an American company got patent right on basmati rice through the US Patent and Trademark Office. This allowed the company to sell a new variety of basmati. This way actually derived from Indian farmer variety. Indian basmati was crossed with semi-draft variety and claimed as a novelty. Other people selling basmati rice could be restricted by the patent. The person who patented that particular biotech organism or a genetically modified organism cannot be used or grows by other country or other company. That thing is called biopatent. Okay, means you do patency against that thing. If that person, that other that community or other organization use that patent, it is only on the permission of that particular organization or company, then you will use that thing. Okay, next, most of the industries, nations, and poor in the biodiversity and traditional knowledge. The developing and the undeveloped world have rich biodiversity and traditional knowledge related to the bioresources. See, the countries which are less biologically, they're less developed, okay, which are non-developed. Okay, these countries have high bioresources. They contain large number of bioresources because they do not know how to utilize that thing. So some developed countries goes there and take their resources and use them. That thing again come under biopiracy. Okay. Next thing is it has to develop law to prevent unauthorized exploitation of bioresources and traditional knowledges. Okay. Means if they are using these things without any limit, then it will exhaust someday. Okay. That knowledge will lost someday. Okay. So different countries, different organizations establish some laws, some norms against them. So in Indian Parliament has cleared a second amendment of the Indian Patent Bill that takes such action in consideration, including the patent term emergency provinces and research and development initiative. Means a substance is produced in India, permission of Indian government. Without Indian permission, that that genetically modified organism does not go outside from India for sale or other country their own. They have to take permission from Indian government, then they will produce. Okay, that thing patent bill. Okay, so this chapter is over. Your unit biotechnology. Yeah. हाँ हाँ चलो बरस okay, so next is your human health and disease. Okay, so health is the mind of an organism in which it is by mentally, by socially, and by physically it is fit. Okay, means our organism should be mentally, by brain, okay, by physically, by body, and by socially. Meaning that outer by nearby your society, you are fit, then that organism is called healthy organism. Okay, so that is the definition of health. That is health is the mind influence immune system through neutral and endocrine system that nearby the health. Okay, when the functioning of an organ and system of the body is adversely affected, it is called disease. Means when a particular organ, particular part of your body, particular system of your body has been get defected, has been get some that comes some pathogen entry, then it's called disease condition. Okay, so disease can be broadly grouped into infectious and non-infectious diseases. Okay, infectious diseases which get transferred from one person to another person, like your common cold, okay, corona, all these things. Okay, then next is non-infectious, the diseases which is not transmitted from one person to another person directly. Okay, then disease causes organisms are called pathogen. Parasites are also pathogen, but they harm the host more. Okay, parasite, pathogen, both are the same thing. Pathogen causes disease, parasite will stay inside the body of the host and they will use the nutrition of the host. Okay, so next, that theory of good humor hypothesis is proposed by Hippocrates and your Indian Ayurveda system, in which it will says that there is four type of humor. First one is your blood, then yellow bile, black bile, and phalgum. If there is disturbance happen between these all the humors, then the person get disease. But this thing has been disqualified, discriminated by William Harvey, why he discovered that blood circulation it demonstrated normal body temperature in person with black bile using thermometer. मतलब meaning using thermometer, he tell that these four things does not control the immunity of an organism. Okay, some other forces present inside the body of organism that controls the immunity. Okay, next thing is your immunity. Okay, immunity, it is an property is in the power of your body which make you fight against different pathogens, different parasites. Okay, immunity is of two types: innate immunity and acquired immunity. Innate immunity means the immunity which is present in your body by birth, and acquired immunity which is you develop from memory, from the interference, from the interaction, from the pathogen, then it is recorded inside your body. That thing is called acquired immunity. Okay, so first we study about innate immunity. Innate immunity is basically of four types. Okay, first one is your physical, second one is your physiological, then your cellular, and at last you have cytokine. Physical means your skin, okay, your mucosa, your submucosa, okay, the outer covering of your body, it comes under physical. Okay, then physiological barrier, physiological level of immunity of your body, it comes that enzymes, the acids, the tears, the sweat, okay, the chemicals, the temperature, the acidic pH of your body, that defines your physiological barriers, or we called is also chemical barriers. Then we have cytokine. Okay, in that cellular immunity, in that cellular immunity, there are different cells come into play and that act as a actioning heroes. Okay, like your macrophages, okay, dendritocytes, T helper, T killer. Okay, these all different cellular bodies which come into the play and kill the pathogen that come under the cellular immunity. And at last we have cytokine immunity, in which there are interferons, alpha interferons, gamma interferons, different cytokines come in the play, and that will act as an immune booster of your body. Okay. So polymorph, nuclear lymphocyte, PMNL neutrophils, monocyte, natural killer cells type of lymphocyte in the blood, as well as macrophages in the tissue which phagocyte and destroy pathogens. Okay, phagocytic cells, example monocyte, pineal, neutrophils, macrophages, these all are phagocytic cells, means they will directly eat your pathogens. Then we have non-phagocytic cells, means they directly do not eat, they release some type of substances, some type of chemicals which kills microbes. Okay, next we have cytokine barriers. Okay, it is released by virus affected cells. See, this is the alarming system of your body. Okay, means a cell when it is get infected with virus or bacteria, it start to secrete cytokines, and these cytokines goes to the near cell, and then the near cell will release different chemical mediators like interferon, and that interferon go and kill the virus or bacteria to the neighboring cells. That thing come under your cytokine barrier system of your body. Okay. See, this is a virus infected cell, okay, and it is producing interferon. Okay, now that interferon chemical messenger will go into the nearby cells and start to signaling them that your neighbor has been attacked by some pathogens, and then these cell will send some messages, some networks to different cells to activate immune cells, to activate immunity of the body. Okay, these are different your cellular barrier, cellular system, cellular pathogen eating cells which is present inside your body. Okay, macrophages, dendritic cell, neutrophils, eosinophils, mast cells, basophil, natural killer cells. Okay, these all are innate immune cells which kills the pathogen and parasite which come inside your bodies. Okay, next we have acquired immunity. Okay, this immunity is not present inside your body by birth, this will be acquired during your phase of developments. Okay, when you develop from child to adult to old, this time this acquired immunity come inside your body. Okay, in acquired immunity your body is take the shooting of different pathogen entry inside your body, and they recognize when they come in the second time inside your body. Okay, the acquired immunity is totally based on memory. Okay, memory of your immune system, what type of memory your immune system have, that is the basis of acquired immunity. Okay. The future of acquired immunity is first specificity. Okay, it is specific, it only kills specific organisms or specific pathogens. Okay, for a specific pathogen your body produces specific antibodies and antigens. Okay, then diversity is very diverse. Okay, you have many types of antibodies, different types of antibodies inside your body. Okay, then discrimination between self and non-self, that is the most important feature of your acquired immunity, that it can differentiate between your own cells and between the foreign cell which is coming inside your body as a pathogen and parasite. And last and the most important feature of your acquired immunity is memory, because it is based, it runs on the memory. If there is no memory based immunity, then there is no acquired immunity. Okay, it is totally based on memory, what type of memory you acquired, what type of memory your antibodies, your immune system occupied, that on it will works. Okay. Next, cells of acquired immunity. The cells which come inside the community, acquired immunity, these are your B lymphocytes and T lymphocytes. Okay, in this B is your bone marrow and T is thymus. Okay, the cells which is coming from your bone marrow side, they are come under B lymphocyte, and the cells which is coming from your thymus gland or other T sites, it is your T lymphocytes. Okay, so both of the lymphocyte need stimulation by specific antigen to trigger them, and response of that is triggered by antigen presenting cell, dendritic cell or APC cells. These are the initial messengers. The message which is taken from them is sent it to the immune system of your body, and then their T lymphocytes and B lymphocytes get activated and fight against the foreign bodies. Okay, so these are the cell of innate immunity: macrophage, neutrophils, dendritic cell, eosinophils, basophils, and these are the cells of your acquired immunity or adaptive immunity, that is antigen presenting cells, T cells or B cells. Okay, so T cells are of four types: T killer, T helper, T suppressor, and T memory. Okay, and B is of two type: memory B cells, and second one is your antibody producing B cells. Okay, one will take the memory and that plasma cell which produce your antibodies. Okay, So first we have B lymphocytes. Okay, these differentiate into gut associated with lymphoid tissue in vertebrates. In mammals, they are differentiated in the tonsils, Peyer's patches, and appendix. Okay, these all are your secondary lymphoid organs where these things get more matured, get more weaponized. Okay, so the B lymphocytes come in contact with an antigen and produce plasma cells. These plasma cells produce antibodies. Simple mechanism is there. B cell memory, it's recording all the things. Okay. Now your antigen presenting cell take that antigen from the pathogen and give that thing to your T helper cells. Okay. Now these T helper cell take this to T killer and your plasma cells. Okay, when it reaches to T killer cell, T killer reach that area and start killing of that foreign bodies. Okay, and when this T helper cell send that message to your plasma cell, it start to form antibody, and that antibody is the most advanced type of immune system of your body. Means the most deadliest weapon of your immune system is your antibodies, which is produced by plasma cells, B cells. Okay, so B lymphocytes come in the contact with the antigen and produce plasma cell. These plasma cell produces your antibodies. Okay, these antibodies are called immunoglobulin, or it is of various type: IgA, IgD, IgG, IgM, and IgG. Okay, and these different antibodies have different functions inside your body. Next is your T lymphocytes. Okay, T lymphocyte differentiate into different cell and coming in contact with the antigen. Okay, first we have T helper. Okay, T helper take action, take your antigen from your antigen presenting cell. Now this T helper cell send that thing to T killer. See, this phenomena will happen. Antigen presenting cell take your antigen to T helper. Now T helper put that antigen, give that help, that idea to your T killer cell. Okay, now T killer cell go to the site. Okay, and that site it will start killing that your pathogens. Okay, now at that time, after the killing of that all the thing, T suppressor cell will come in the play, and they suppress the activity of T killer cells. Okay, when this all the processes is going on, at that time T memory and B memory cells recording all the things. Okay, they are seeing all the things for the next time. When next time that pathogen go inside your body, they are steady ready for the killing of that pathogen. Okay, that's how your T based immune system work inside your body. Okay. So these all are the cells which is present in your T lymphocyte system, that is helper T cell, killer T cell, suppressor cells, and memory T cells. Helper T cell, the activated helper stimulate the killer cell. Means your helper T cells stimulate the functions of your killer T cell. Now that killer T cell can do over functioning. This over function is being reduced down with the help of suppressor T cells, and after all the thing is going on, your memory T cells is recording all the things. Okay, and this is the mechanism of normal immune system inside your body. This is your antigen. Okay, which is occupied with your antigen presenting cell. Now this antigen presenting cell gives this information, this antigen, to your T helper cell. Now from T helper cell, this information give to two places. One, it will send to your B cell, and B cell start to produce antibodies. Next, this T helper cell give this information to your macrophages and T killer cell. Okay, now your T killer cell will get to that location of infection and start to the killing of pathogen and parasite. but sometimes the T killer cell do its work overly and it will kill down the immune system also. Okay, so for avoiding that thing, T suppressor cell will come in the play and suppress the activity of T killer cell. And after all these things going on, your B memory and T memory cell is recording all these things. Okay, that's how your immune system works. Next is your antibodies, okay, which we call antibody mediated immune system or humoral immune system, AMIS. Okay, in this we have five types of immune bodies. Okay, IgE, IgM, IgD, IgM, IgA. Okay, these are the five antibodies which is present inside your body. Again, IgG, IgE, IgD, IgM, and IG A. Okay, IgG, IgE, and IgD. These are monomeric antibodies, means single. Okay, and your IgM is a pentameric. Okay, means five antibodies combining and form a single structure. And this is your IgA dimeric. Okay, two antibodies are coming together and forming that antibody. Okay, this is the structure of your antibody in which you have two chains. One is your heavy chain and second one is your light chain. This one is your light chain and this one is your heavy chain, and this heavy chain and light chain is connected by each other with the help of disulfide bridge. Okay, and this part of your heavy and light chain is called variable portion. This changes the position. Okay, the sequencing, that area, the region of this area has been changed easily. When your different antigen come inside your body, it will change its action. Okay, means if a round shape antigen will come, it will change its shape to round, and it will capture that thing and start doing killing of that thing. Okay, next, this is your antibodies. Okay, this is your IgG, अकेली ही काफी है. Okay, this is your IgA, दो. Okay, this is your IgM, 5. Okay, this is your IgE, अकेली ही काफी है. Okay, and this is your IgD. This is your IgD. Okay, this antibody is present very less inside your body. Okay, that's why it is like this. बाकी सब the great खल्ली और ये पुनीत superstar. Okay, next, this is the action of your antibody, the specificity power of your antibody. Okay, means this square binding antibody site always bind to the square antigen. This round will always bind to the round one, and this triangle will always bind to the triangle one. This will not going to bind this thing is called specificity of the antibodies. Okay, means antibody is specific to a particular antigen, means it is not going to bind to different antigen. Okay, and this is your antigen binding site where your antigen get binded and your antibodies start to go at the desired position and start to kill that foreign particle or the pathogen. Okay, next, functions of antibody. Okay, there is three functions of antibody basically. First is agglutination. Antibody attached with the antigen which is present on the surface of pathogen and destroy the pathogen cell by the lysis. Means it will get attached to your, this is your antigen. Okay, and this is your antibody. It will get attached to your antigen. Okay, and start to destroy your pathogen. Okay, this process is called agglutination, means attachment of antibody with antigen and destroying that pathogen. That process, that power, that process of antibody is called agglutination. Okay, next is opsonation. Okay, means your antibody is surrounding that particular pathogen and stop releasing of the toxin which is producing by that pathogen especially. Okay, so coating of bacterium with the opsonin antibody IgG and IgG facilitate the phagocytic cell of these antibodies, or opsonin promote phagocytosis by combination with the antigen. Means simply, there is a single people in the in road accident or in a normal road fight. Okay, there is a one theft. Okay, उसके चारों तरफ लोग खड़े हो जाते हैं, उसको पीटते हैं. Okay, there is a normal, listen. Okay, there is an one people. Okay, around that one person, 5-6 people come and start to beat them. Same thing is opsonation. That single person is your bacteria. Okay, and the 5-6 people with coming to beat that single person is your antibodies. Okay, now that good bhaji will make to kill that pathogen, and that process of your antibody is called opsonization. Okay, next is your neutralization. Neutralization means neutralizing the toxin which is producing by pathogen by eating that toxin. Okay, that thing is called neutralization. Okay, antibodies neutralize the toxin of bacteria by attaching with them. They will attach to the body of the pathogen or that antigen and they will start to neutralize that thing. Okay. Next, your different antibodies present inside your body. IgM 5% monomer pentamer in the design, okay, structure. Then IgD 0.3 monomer, okay. IgA 7 to 15% monomer again. IgG 85% monomer shape and IgE 0.02% monomer, okay. See, that is 0.3%, okay, and it has very main role inside the body of your immune system, but still that function is not known. Okay, जो IgD एंटीबॉडी उसका फंक्शन अभी तक स्टिल अननोन है. Okay, but IgM, IgA, IgG and IgE, these antibodies actions are known inside your body. Okay, the basically the most important work is done by your IgG. Okay, this is the main antibody of your body. Okay, this function is mainly that main function of antibody, it is done by your IgG. Okay, most abundant class with four isotope: Ig1, Ig2, Ig3, Ig4. Okay, these are the four subtype of IgG antibody. It can cross the placenta and it do opsonization process. Okay, then your IgE, it is come in the action when the body faces allergic reactions. Okay, when the allergic reaction take place inside your body, IgE releases. Okay, IgA, it is present inside the secretions of your body like saliva, colostrum. Okay, next IgM, membrane bound immunoglobulin on the surface of immature and mature B cell. Okay, this is the most active immune antibody of your body, IgM. Okay, it is the gangster of your body. Okay, ये ऐसा गैंगस्टर है जो सारे पैथोजेंस को मारता है अकेला. Okay, that is your IgM. Okay, next is your acquired immunity. Immunity you develop during your life. Okay, so it is of two types: active immunity and passive immunity. Active immunity again of two types: natural and artificial. Passive again natural and artificial. See, active immunity is that immunity which develop after being exposed to the infection or getting a vaccine. Okay, means you get a disease, you get common cold. Okay, and second time common cold do not affect you more because your active immunity in the interference. Okay, it will affect the power of common cold for next time. Okay, so it is of two types: natural antibodies made after the exposure to an infection. Okay, and artificial antibodies made by getting a vaccination. Okay, mean you are getting a vaccination in the form of dead of that dead matter of that virus or bacteria, or you are taking antigen of that bacteria by outside, then this thing is your artificial. Artificially you are taking that bacteria inside your body. Either it is in dead form or you are taking the antigen of that bacterium or virus inside your body. Okay, natural antibody made after the exposure to an infection. Okay, you are get infected that organism first, then the memory is developed inside your body. That thing is your natural active immunity. Okay, passive immunity, immunity you are acquired from someone or something else, from somewhere other, some place other you are taking that immunity inside your body. That type of immunity is called passive immunity. Example: natural and artificial. Natural antibodies transmitted from mother to baby. Okay, from mother to baby, the immunity which is coming inside your body means the IgG which is can close the placenta, it will coming inside the body of fetus. That type of immunity is called natural passive immunity. That immunity which is not present in your body by birth is called passive immunity, and when it is coming from mother to baby, then it is called natural passive immunity. Okay, artificial antibodies acquired from immune serum medicines. Okay, artificial vaccines. Okay, these both are same, but there is some difference. Okay, the difference is that this is incorporated inside your body and your body making antigen antibodies against that organism second time also. In this, artificially you are taking antibodies from outside. Your body is not producing antibody against that bacterium or virus. You are taking that antibody, that bacteria, you are taking that vaccine from outside. It contains already that bacterium and viruses. That thing is artificial passive immunity. Okay, so next is your vaccination and immunization, means injection of vaccine inside your body to treat to get rid of about different viral diseases or bacterial diseases. Okay, so vaccine is a suspension of attenuated inactivated pathogen or antigen protein of pathogen which is taken orally or injected to provide immunity for that pathogen. Okay, means it is in dead form or in the form of antigen of that particular pathogen which going to infect you is been injected inside your body. Then this process is called vaccination or immunization. Okay, and then again it is of two types: active immunization and passive immunization. Active, in this, a preparation of vaccine, antigenic protein or pathogen or activated pathogen is introduced into the body. It results in development of antibodies. Means it start to develop antibodies inside the body. Means you have to take that thing from the outside, and when it is get inside your body, it will make the copies. Okay, means first you have to face that thing. After that, if we introduce, it will make antibodies inside your body. That thing is your active immunity. Passive immunity means it is direct injection of preform antibodies. Means directly you are injecting antibody inside your body. Okay, in this active immunization, your body first take dead organism or antigen inside your body and produce antibodies. Okay, but in passive immunization, your body directly taking antibodies from outside. Okay, again, active immunization means your body first take dead organism, dead virus, dead bacteria or antigen and make antibody inside your body. Okay, but in passive immunization, your body is not making any antibody. You are taking prepared antibody from outside and that prepared antibody is injected inside your body. Okay, example of passive immunization is tetanus vaccine and snake antivenom. Okay, so these are the different types of vaccine which is present in the market. Live attenuated inactivated subunit toxoid or RNA base. Tuberculosis, oral polio vaccine or measles, rotavirus and yellow fever. These all are live attenuated vaccines. Means in this that organism is living. Okay, next is inactivated kill antigen. Means you are introducing the antigen inside your body, but that antigen is inactive. Okay, next is purified antigens. Okay, cellular pertussis, hemiphalus influenza type B or pneumococcal. Okay, PCV10, PCV7, PCV13. These all vaccine are come under subunit of purified antigens. Okay, means you are injecting the purified antigen of that bacteria. Means the bacteria with the antigen which is present on the cell wall of that bacteria is injected inside your body. Then this thing is your subunit. Okay, toxoid inactivated toxins means you are injected toxin inside your body, but the toxin is inactivated that do not cause any disease inside your body. Okay, next is your RNA base. This is the most recent advancement in vaccine. This is your next generation vaccines also. That is your hepatitis B vaccine, which is also called rDNA vaccines. Okay, next is your HLA system. Okay, this system is the system which is introduced inside your body to detect that your body is not malfunctioning or not. Means it is the system which will know that it's the outer cell is of your own body cell or it is outer cell pathogen. Means it is a type of outer organism finding detecting system inside your body. The system which detect the outer pathogen inside the body, that is your HLA system. Okay, HLA stands for human leukocyte antigen, also known as major histocompatibility complex, MHC. Okay, this MHC complex is responsible for the tissue organ transplantation. If MHC is crossed, and it will same means your organ transplantation is successful. And if MHC is not successful, means that histocompatibility complex test is not successful, means that organ transplantation can be failed. Your body will produce antibodies against that new transplanted organ inside your body. Okay, next, human leukocyte antigen are genes in major HMD complex that help to code for proteins that differentiate between self and non-self. This is the most important part of your immunity. If this system fails in your body, then autoimmune disorder develop inside your body. Your own immunity start to eat your body. Okay, they play a significant role in disease and immune defense. These array of LA alleles on homologous of our chromosome 6 is known as halotype. Okay, this thus the genes of HLA system is present on chromosome number 6 and it is called halotype. Okay, an individual inherits one HLA halotype from each parent. Okay, three will come from mother, three will come from father. Okay, only identical twins can have identical halotypes. Okay, because they are developed from a single zygote, that's why they have same type of halotypes. Okay, next, tissue grafting or organ transplantation means ejecting of different organs inside your body. Okay, means if your liver get damaged, you can transplant your liver. Okay, if the heart get failure, then heart is not working, so you can transplant heart inside your body of somebody else. Okay, same for kidney, same for that eye. Okay, someone get burnt their skin, they can be transplanted by different part of the body of the skin can be transplanted here. Okay, that thing is called grafting or tissue grafting. Okay, so it is of four types. Okay, autograft, transplantation of tissue from one part of the body to another part. Okay, it is the most successful type of tissue grafting. Example, if my skin burnt from here, I can fix the skin from this part to here. Here the skin will grow normally, but in this part, the burnt part of the my skin, it not grow that much rapidly. Okay, and it cannot heal. That's why if I put the skin part from here to here, it will automatically heals fastly. Okay, then this type of graft is called autograft. Okay, next is isograft, transplantation of tissue in between the individual of same genetic constitution. Okay, example, identical twins or monozygotic twins, bhai. Okay, next, homo or allograft, tissue grafting in between the genetically dissimilar individual of same species. Example, family, means I can install the skin or hairs of gorilla inside my body, or inside the body of gorilla my hairs can be transplanted. That type of graft is called allograft between the same species member. Okay, and then is your heterograft or xenograft between two different species. Okay, autograft in your body. Okay, isograft between your brother sister. Okay, homo or allograft means between the same species member. Okay, and heterograft between different species member. Okay, just like in some cases of bun, you can place skin of fish or reptile here. Okay, mammal is different thing, reptile is different, totally different species, so that type of graft come under xenograft. Okay, then organ transplantation requires three things. Okay, blood group matching. Okay, same blood group should be there. Then MHC matching tissue typing. Okay, in which both the tissues have same type of MHC. Okay, and at last, use of immunosuppressive drugs like cyclosporin A to reduce down the immune system of your body. Okay, failure reason of organ transplant. What is the failure reason between that organ transplant processor? Okay, cell mediated immune system mainly, means your immune system is start degrading, eating that outer organs. Okay, then your that transplantation is it become failure. Okay, so success graph order, self, खुद का टिश्यू खुद की बॉडी में लगा लो, then it is successful. Okay, next, identical twins, next siblings, then parents, and then unrelated donors. Okay, unrelated donors graft is rejected usually. Okay, but the graft of your own body is never rejected, means you are taking some part from here and attaching here, it never get rejected. Okay, because it's your own body. Okay, this is your allograft. Okay, this is your bone graft substitute, or it is called bone graft. Okay, it is a particles which will place when your bone get fractured. Okay, and this is your xenograft. Okay, these are different skin grafting methods. See, in this image you are seeing that fish skin is being grafted. Okay, here again fish skin is grafted on the human skin, and at last you get this type of lizard. Okay, this is the A, then B, then C, then D, E, F, G. Okay, in the A you get injury. Okay, in B the injury get clean. Okay, in C you get a graft, skin graft. In D image you are seeing that graft is placed on the affected tissue. After E, it is the removing of that unused part. Okay, and EF, in F part you are seeing that the tissue is get matched with the human skin, and at last G you will see that your human skin get fully developed. Okay, it will grown over that animal skin. Okay, next is your immune system. Okay, the immune system which is present inside your body, there's immune system having two types. Okay, it will come under two types. Okay, first is main primary immune system, and second is your secondary immune system. or primary lymphoid tissues or secondary lymphoid tissue. Okay, so primary lymphoid organ and secondary lymphoid organs. Primary lymphoid organs is the organs where the formation of B lymphocyte and T lymphocyte take place. Means your bone marrow and thymus. Bone marrow for B lymphocyte, the thymus for T lymphocyte. It is the place. Primary lymphoid organs are those organs where your formation of B lymphocyte and T lymphocyte take place and their maturation take place. That organs are called primary lymphoid organs. Then we have secondary lymphoid organs, that is your Peyer's patches, appendages, lymphoid, that lymph nodes, okay, lymphatics, or your Peyer's patches. All these things are come under secondary lymphoid organs. In these areas, your T lymphocyte and B lymphocyte get more weapons. Okay, they are developed more, they will have more power, they will get more power. Okay, so these are your secondary lymphoid organs: spleen, lymph node, marred mucosal associated lymphoid tissue, means your lymphatics, appendix, and your that Peyer's patches. Okay, these come under your mucosal associated lymphoid tissues. Then your lymph nodes, okay, these are present in your axillary parts, below the arms, in the legs, okay. And spleen, it is a phagocytic and lymphocytic part in which we trap blood borne microbes and act as a filter. Okay, it is reservoir of erythrocyte because it is also called blood bank of your body. Okay. Next, this is the different lymphoid organs present inside your whole body. Okay, this is your tonsil which is present below your oral cavity. Then it is thymus. Okay, then your spleen, then your lymphoid nodes. These all are lymphoid nodes, green. Then your lymphatic vessels, then bone marrow. These all come under your lymphoid organs, primary and secondary. Okay, next is your allergy. Okay, it is then reaction of immune system to to some antigen surrounding is known as allergy. Okay, it is an allergic reaction in which your IgE antibodies work and kill the allergens. Okay, allergens, common symptoms of allergy is sneezing, watery eyes, rashes, running nose, and asthma. Okay, and the most advanced type of allergic reaction is your asthmatic shock. Okay, or that shock which is due to the asthma which happen inside your body. Okay, release of histamine and serotonin is the main cause of having allergy. Okay, serotonin and histamine, they will increase or decrease the dilations of your arteries and veins. Okay, that histamine, it will increase the diameter, means do vasodilation, and serotonin, it will constrict your arteries and veins, do vasodilations. Okay, drugs, steroid, antihistamine, and adrenaline septin reduces the symptoms. Okay, means they act as a inhibitors of histamine and serotonin. These all drugs, steroids, and antihistamines act as a anti-allergic drugs. Okay, they are used to give in allergic persons. Okay, now next is your autoimmune disorders. Okay, your when your immunity get chapter के बाद. Okay. Autoimmunity. Okay, so this is the condition of your body in which your body own immunity start killing your own body cells. That condition is called autoimmune disorders. Okay, so when the immune system does not discriminate between self and non-self, then that condition is called autoimmune disorder. Okay, so these are the some autoimmune disorder: myasthenia gravis, against the acetylcholine receptor which is present between the nerve endings. Then pernicious anemia, cell again, and that antibodies created against the castor instinctive factor and vitamin B12 absorbing cell which is present inside your gut. Next, Hashimoto disease, in which your antibodies are prepared against the thyroid gland. Okay, next is your rheumatoid arthritis, in which that antibodies are formed against the synovial joint. Okay, next is your insulin dependent diabetes mellitus type 1 diabetes, in which antibodies are doing work against the pancreatic cells. Okay, and your multiple sclerosis, in which your antibodies are made against your myelin sheath, in which the myelin sheath get degraded. Okay, so in our course we have rheumatoid arthritis. Okay, it is the joint pain in which that antibodies are formed against the synovial fluid which is present between the joint, due to which a solid structure which is called pannus is will develop, and when you move your fingers or joints then you feel intense pain. Okay, treatment: pain and inflammation by heat treatment or physiotherapy can be done, and in severe cases of rheumatoid arthritis, the ultimate treatment is joint replacement surgery. Okay, means you have to replace the joint. Okay, next is your immune deficiency disorder, means now your immune system is down. Okay, shut down, or your immune system is lost. Now it is your immune deficiency disorder. It is two diseases come under this: skids or AIDS. Skids is your severe combined immunodeficiency in which enzyme like ADA deficiency occur inside your body. Your body is not synthesizing ADA due to mutation, and your T lymphocytes and B lymphocyte does not get mature, and they do not having the power to kill pathogens. Then it's called SCIDs. Okay, treatment: gene therapy. Next is your AIDS, acquired immunodeficiency syndrome, in which a virus called HIV virus come and get incorporated into your T helper cells and your macrophages and start to make them factory of HIV, and due to which your body immune system collapsed. Okay, these are the patients of AIDS. Okay, their body get totally damaged, their immune system totally shut down. Okay, a normal common cold can kill them. Okay, now next is some different diseases which is present inside your course. So first we have typhoid fever. Okay, this is a life threatening condition caused by bacterium Salmonella typhi. Okay, its causes is: typhoid fever is caused by bacterium Salmonella typhi, which is related to the bacterium that cause Salmonella food poisoning. It usually spread through contaminated food or water. If you eat or you drink contaminated food, so that will cause your typhoid fever. Okay, treatment: typhoid fever can be treated with antibiotics. Okay, this is the typhoid fever symptoms: white coating on the tongue, rashes on the body, enlarged liver and spleen, ulcer in the intestine. The person who is infected by typhoid, its body look like this. Okay, it will fatigue. Okay, full body is fatigue, full body is pain during that time. High fever occur inside your body. Okay, next one is your pneumonia. Okay, it's caused by streptococcus pneumonia or Haemophilus influenza. It is a respiratory disorder in which your lung is filled with fluid. Fluid is increased inside your lung, due to which the air absorbing capacity of alveoli decreases and you feel suffocation. The chest will feel heavy. Okay, next, this is the X-ray of a person who having pneumonia. Okay, pneumonia is the most common symptom during COVID. Next one is your dysentery. Okay, this is a GIT disorder in which your GIT does not start or doing proper absorption and proper elimination of food. Okay, it will start to produce watery stool or bloody stools. Okay, in which it is high of blood. Okay, so there is two type of dysentery: bacillary dysentery, which is caused by bacteria, or the second one is your pylori dysentery, which is caused by amoeba. Okay, and amoeba historica. This is the case of your dysentery in which the pathogen is present in all over your small intestine and large intestine, mainly in large intestine. Okay. Next is your plague. Okay, plague or Black Death. Okay, it is the pandemic during the World War One and Two in Europe. Okay, in this disease, what happened? Your body develop different skin rashes of black color. That's why it is called Black Death. The body is become black color due to the development of these black rashes all over the body. Okay, the causative agent of plague is that your bacterium that is present on that rat, means when your rat, that infected rat, cuts you, bites you, then the bacterium of that disease, Yestera pestis, get inside your body and cause this disease. Okay, this is the disease effect inside your body in which the whole organs have been colored black. Okay, that's why it is called Black Death. See, and this is the X-ray of that, and this is the pathogen Yestera pestis, which causes plague. Okay, next one is your diphtheria. Okay, it is again respiratory disorder in which your air pathway get blocked due to the development of pseudomembranous structure inside your throat. Okay, and this thing is caused by corynebacterium. Okay, corynebacterium diphtheria, in which that bacterium releases a toxin which act inside your body, plus it produces a pseudomembranous membrane inside your throat like this. Okay, this is a diphtherian patch, a pseudomembranous covering which is developed inside your throat, due to which you cannot swallow food. You will feel suffocated during inspiration of air. Okay, next is your tetanus. Okay, tetanus disease, which is done by Clostridium tetani bacterium. Okay, in this disease, Clostridium tetani produces tetanus toxin, which will affect your muscular system and neurological system. Neuromuscular system of your body get damaged during this disease. This disease is spread when your body is cut down with any rusted iron or a rusted metal. Then that bacterium go inside your body and start doing neurological damage to your neurons. Okay, the person who getting tetanus having this type of shape, when he will having high power of high, the toxin get more injected in the body. Then okay, it is also called jaw lock disease, because in this your mouth cannot move, all the muscles of your body get locked. Okay, you cannot move your body, your body cannot move in this disease. Okay, their body become a bow arrow like structure. इसको हिंदी में धनुष बाण डिजीज भी बोलते हैं, धनुष बाण बीमारी, जिसमें बॉडी का डिजाइन धनुष की तरह हो जाता है बिल्कुल. Okay, next thing is your tuberculosis. Okay, TB caused by mycobacterium tuberculosis. Again, it is a respiratory disorder of lungs in which your lungs get pneumonated, means the level of your blood increases inside your lungs and you cannot breathe properly. The surface area of alveoli decreases and person cannot breathe properly. Okay, treatment, usually treatment with a 6-month course of antibiotics such as rifampicin, isoniazid. Okay, prevention, BCG vaccine is there to inject it to the person and it will get treated with this disease. Okay, this is infectious disease from one person to other person, can it be transmitted easily, and this is the lungs of a person who having TB. Okay, next one is your leprosy. Okay, leprosy, it is caused by mycobacterium leprae. It is an skin and neurological disorder in which your skin start to degrade it, eaten by bacterium mycobacterium leprae. Okay, it will affect your lymph node system, your neural system and your skin, the dermal system. Okay, treatment, leprosy is curable with multi drug therapy, MDT. Okay, once a person with leprosy begin appropriate treatment, the cure become non-infectious. Treatment usually lasts between 1 to 2 years. This treatment goes long. If you do not take proper treatment of this disease, it can cause death also. Okay, this is the action of this disease. It will eat the organs. Okay, it will eat the skin part. See in this image, you will see that nose is eaten. Okay, that bacteria eat out all the skin, even in the bone also. That is a very deadly disease. Okay, next is viral diseases. Okay, first is common cold. Okay, this common cold is caused because of rhinovirus. Okay, in which your upper respiratory tract, means your nose, nostrils, pharynx, till that part get only affected. Okay, one of the most infectious human disease caused daily and it is common in winter. Okay, next is your chikungunya. Okay, this is a viral disease spread by infected mosquito that cause fever, joint pain and rashes. Okay, treatment, no medicine, but most people recover. There is no more medicine for chikungunya, but you can recover with proper care. Okay, this is the Aedes albicophytus, which is a mosquito which causes chikungunya, which is the vector of chikungunya. Okay, next one is your dengue fever caused by dengue virus. Okay, or arbovirus. Okay, in this the disease within the bacterium, the virus, that arbovirus will eat platelet. The platelet count of your blood decreases in dengue. Okay, then this is the virus which this is the vector of your dengue disease. Okay, a normal platelet count for a healthy person is 1,50,000, but when a person get dengue, it goes down to the 5,000 or 10,000. Okay, next thing is your hepatitis B caused by hepatitis B virus. Okay, it is a sexually transmitted disease. Okay, in which your liver get affected mostly, and simultaneously your digestive system get also cracked. Okay, treatment, there is no cure for hepatitis B. Okay, initial symptoms of hepatitis B is jaundice. Okay, the person who get hepatitis B, its tummy become like a pot. Okay, next is your amyloidosis. Okay, amoebial disease caused by your protozoan. Okay, in which your entamoeba historica come in the play and start to start to degrade the walls of your small and large intestine. The food does not get easily absorbed inside your body, and it will directly pass out from your litter, okay, from that outside in the morning, okay. This is intrathecal inoculation. This is intraportal vein inoculation. Okay, it is present all over the body when the infection get high. Okay, it will make your portal vein perforation. It will make your interthecal vein perforation. Means your cecum will get perforated when this disease happen. Okay, next is your ascariasis. Okay, ascariasis, in which this Ascalementhes will make home inside your gut. Okay, it will absorb all the nutrients inside your body, and you will get bloody stools. Okay, or your stools is not that much proper. Okay, and this disease is being activated inside your body when you eat uncooked meat or a fruit which having the eggs of this organisms. Okay, next is your elephantiasis, also called lymphatic filariasis. Okay, it was caused by again from an organism which is called Wucheria brandcofti, and the vector of this disease is Culex mosquito. Okay, in this disease your lymphatics get induce your lymphatic get involved, and which make your lymphatic system swell up. Okay, it is also called elephant leg disease or elephantiasis. Okay, in which your leg look like elephant, your all lymphatic system gets swelled up. Okay, and that make swelling inside your leg. Okay, in male it can cause infertility also, it will destroy testes also. Okay, this is before surgery and this is after surgery, and you can see here the graft which is placed in the feet of that person is the skin of fish. Okay, so it is a type of xenografting then. Okay, next is your ringworm. Okay, fungal disease, normal during the rainy season, caused by candida fungus. Okay, or normal different funguses are there which cause ringworms. Okay, it is a fungal disorder caused when you use towels which is not washed properly, or you will use towels or combs of infected persons. Okay, this is the ringworm. Okay, this is the skin dermal ringworm, and this is scalp ringworm. Okay, this will happen when you use the towel of infected person, or you will not use proper towel of the function. Okay, and this is your ringworm, in which your scalp get incorporated with the fungal growth. Okay, next is the most important disease according to NCERT, is malaria. Okay, caused by female anopheles mosquito, and causative organism is your plasmodium falciparum. Factor is your anopheles, female anopheles mosquito, and the causative organism is your plasmodium falciparum. There is three species of plasmodium: the plasmodium vivax, plasmodium falciparum, and falciparum plasmodium malariae. Okay, the most dangerous species is plasmodium falciparum. Okay, so they see what will happen in this disease. Okay, first that sporozoite will get inside your body. Okay, when your infected female anopheles bite you, that sporozoite will get inside your body. Okay, now that sporozoite will regulate with room inside your body, and finally it reaches to your liver. In hepatocyte it will convert, it will do mitosis, and it will convert into merozoites. Okay, now that merozoite rupture out from that liver cell and come inside your blood circulation and develop into schizozoites. Okay, now these schizozoites will convert into tropozoites, and now this tropozoite have two fates. One, it will convert into tropozoite, remain the tropozoite form, or it will convert, it will do meiosis and convert into male and female gametophytes. So half of them remain in tropozoite phase, and half of them convert into male and female gametophyte. Now these things will reach on the skin, dermal area, dermal blood area. Okay, and then the normal mosquito come and bite you. Then these gametes of your malarial parasite get inside the mosquito, and after some time it will do sexual reproduction inside the mosquito gut. Okay, after doing that sexual reproduction, it will produce again your sporozoite and will reaches to the salivary glands of the mosquito and wait for the next victim of that malaria. Okay, they will not die because there's a salivary gland of mosquito contains some anticoagulants, okay, which make the blood not to coagulate, okay. Next, we have sexually transmitted disease STD. Okay, name of STDs: gonorrhea, genital herpes, genital wart, syphilis, trichomonasis, chlamydiasis. Okay, and pathogen: Neisseria gonorrhea, genital herpes, herpes simplex virus, genital wart, human papillomavirus, syphilis, Pteranoma pallidonium. Next, trichomonasis, Trichomonas vagalis, and chlamydia, Trichomonas is in bacterium. Okay, so these all are STDs in which we have to study about HIV/AIDS. Okay, so first we have AIDS. AIDS, acquired immunodeficiency syndrome. Okay, इस चैप्टर के बाद. Okay. Acquired immunity, that is your acquired immunodeficiency syndrome, AIDS caused by HIV virus. Okay, so it is the deficiency of immune system. Syndrome means a group of symptoms. Means in AIDS there is only not a single disease. Many syndromes comes together and induce inside your body. Means a simple common cold can cause death of the organism, which is caused, which contain the AIDS disease inside them. Okay, so AIDS is a viral disease caused by human immunodeficiency virus HIV and member of a group of virus called retroviridae family. Means its genome contains reverse transcriptase, that's why it is called retroviridae family. Okay, has an envelope enclosing that RNA genome. Okay, characterized by decrease in the number of helper T cell, also called slim disease, because in this disease the metabolism of that person gets zero and the person becomes slim trim. Okay, first detected in homosexual males in USA in 1981 at Disease Control Center, Atlanta. In India, first case of AIDS was reported in 1986 from Chennai. Okay, so these are the how the transmission of AIDS takes place. First, sexual contact with infected person. Second, transmission of contaminated blood products. Third, sharing of infected needles from the infected person, and fourth, from infected mother to the fetus. Also, these viruses can easily cross your placenta also. Okay, next, high risk people who getting HIV, means the person who having high risk to get HIV. First, individual with multiple sex partners. Second, that is individual who require blood transplant, repeatedly do blood transfusions. Third, drug addicts who take drug intravenously regularly. Then children born on HIV infected mothers. Okay, the mother who having HIV positive, that have high chance of developing AIDS inside their body. Okay, then there is some things, that is some taboo related to AIDS, that by kissing it is not transmitted, by hugging it is not transmitted, by sharing food it is not transmitted, insect bites it is not get transmitted, toilet using toilet seat, means previously if the toilet seat is used by HIV patient, then a normal person go and use that toilet seat, it is not get transmitted. Bathing with HIV infected person, it is not getting transmitted. Sneezing and coughing, it is not get transmitted from one person to another person, or sweat touch, it is not get to transmitted from one person to another person. Okay, next, इसका स्ट्रक्चर की बात करते हैं, द स्ट्रक्चर ऑफ योर रेट्रोवायरस HIV वायरस. ओके, सो रेट्रोवायरस लेंटिवायरस जीनस और रेट्रोविरिडरी फैमिली belongs from your lentivirus genus and retroviridry family. Okay, core has two identical molecules of single stranded RNA. Okay, so these are the two identical molecules of single stranded RNA. An enzyme reverse transcription, for the reverse transcription process, protease enzyme for making proteins, infected proteins, and integrase enzyme to integrate all the proteins simultaneously. Okay, core also have capsid protein P24 and matrix protein P17. Okay, next, envelope is made up of lipoprotein and contains G120 and G41 proteins. Okay, and these two proteins G120 and G41 is responsible for not developing antigen antibody reaction against the HIV, because this GP120 is the same thing which is present on the your H helper T cell receptors. Means the G120 is the same receptor which is present on your T helper cell, so it is easily get enter inside your helper T cell. Same thing is written, 120 has complementary sequence to CD4 receptor present on the macrophages, HIV factory and helper T cells. Okay, next, replication or retrovirus, how this process runs. Okay, this is your helper T cell, this yellow. Okay, So seen is that retrovirus get entered inside your helper T cells because it has having this receptors. Okay, GP120 receptors is present on your viral capsid. Now this thing will get enter inside that by accepting that thing. Okay, now your viral DNA get inside your helper T cells. Okay, it will do reverse transcription. It will start doing reverse transcription by the help of reverse transcriptase. Now your identical DNA which is present inside your genome of helper T cell is produced. Now this viral DNA is go inside the genome of the helper T cell, okay, and start to forming new viral DNA by the help of integrase and protease enzyme. Now this new viral RNA is formed and start to producing new viral, and your normal T helper cell converted into HIV factory, or your macrophages is converted into HIV factory. That's how the mechanism of HIV works inside your body. Again, this is your HIV virus. It will contain GP120 receptors. That receptor code with the receptor which is present on your cell membrane of your T helper cell. When these two get matches, your HIV genome get entered inside your T helper cell. Now reverse transcriptase come in the action. It will convert viral RNA into viral DNA. Now that viral DNA get into the genome of helper T cell and start doing production of viral DNA. Okay, now that viral DNA come out from that nucleus and start production of new viruses, and that new viruses now start to degenerate and get infect neighboring T helper cells. Okay, this is the whole process. HIV enter into the body. Two macrophages act as HIV factory. Then RNA replicate in the presence of reverse transcriptor to form viral DNA. Then viral DNA incorporate into the host DNA. Infected cell produce viral particles. Then HIV enters into T helper cells, replicate and produce progeny virus. Attack on other helper T cells. T cell decreases, weaken immunity. Okay, means the first site of entry of your HIV is macrophages. After destroying all macrophages of your body, it will act on T helper cells. Okay. Next is your symptoms. What are the symptoms of HIV/AIDS? Okay. So we have three. First is asymptomatic phase. Okay. In this phase, your body you does not know that you have AIDS. Okay, but you have AIDS. Okay, means basically your body does not showing any symptoms of AIDS. Okay, that thing is called asymptomatic phase. Okay, there is no symptoms in your body of AIDS. Next is AIDS related complex arc period. Okay, in which mild form of AIDS, means mild symptoms of AIDS is being discovered inside your body. You will show mild symptoms of AIDS like body weakening, small infections will happen inside your body. Okay, then the last stage of AIDS, which is called full blown AIDS. Now your body immune system is ready to collapse. Okay, means the immune system is towards the collapsing side. Okay, whole immune system is collapsed. Now a small infection like common cold, tuberculosis can kill you. Okay, and the main reason of death of AIDS patients is not HIV virus. It's that opportunistic infections like tuberculosis, leprosy, tetanus, common cold, pneumonia. Okay, these all infection causes death of the person who is having AIDS. AIDS is the not the cause of the death of the organism. That cause of death of that organism who having AIDS is opportunistic infections like candidiasis, tuberculosis, pneumonia, common cold, okay, tetanus. AIDS only open the gates for these infections. Okay, जो एड्स होता है वो बस दरवाजे खोलता है दूसरे इन्फेक्शन के लिए. ओके, इम्यून सिस्टम को गिरा के उस पर्टिकुलर ह्यूमन का. Okay, the main functions of HIV virus is to close the immune system of a particular organism and open the gates of other organisms to get enter inside the body of the human and start the killing of the human cells. Okay, next is your investigations. Screening test is your ELISA, and confirmatory test is the Western blotting, which detect the antibodies in patient serums. Okay. Next is your treatment. Reverse transcripted inhibitor drugs. Okay. Protease inhibitor drugs and integrase inhibitor drugs, या फिर HART, highly active antiretroviral therapy. It is a mocktail cocktail in which all the drugs, that reverse transcriptase inhibitor, protease inhibitor, and integrase inhibitors are mixed and given to the patient. That thing is called HART, highly active antiretroviral therapy. Okay, AIDS disease cannot escaped from your body. Okay, it will make you kill, but taking proper treatment, it will increase your living years. Okay, death is confirmed in AIDS. Okay, but by taking proper treatment of AIDS, it will increase the lifespan. Okay, it will not hinder your death time, but it will increase the टाइम ऑफ मतलब जो मौत होने का टाइम होता है वो इंक्रीज हो जाता है. ओके, next is your preventions of AIDS. Okay, first educate people about AIDS. Second, use disposable needle and syringe. Free distribution of condoms. Make blood banks safe from HIV. Means HIV testing should be there. Next, regular checkups for AIDS, HIV/AIDS. Advocating safe sex. Okay, using condom. All these contraceptive methods should be there to avoid the AIDS. Okay, controlling drug abuse. Okay, stop. Say no to drugs. Next, cancer. Okay, next, cancer. Okay, so cancer, it is an abnormal and uncontrolled growth of cell inside your body. Okay, see, in normal process cell division takes place. That cell division is controlled in the way by the help of contact inhibition. Okay, contact inhibition is the property of your cell division. Contact inhibition is the property of your body in which when the cell is in stop the process of cell division, but when cancer take place, your contact inhibition property of cell lost. Okay, it will not stop doing cell division. Okay, so it is the property, it is a disease in which your body forget to stop division. Okay, uncontrolled cell division is called cancer. Okay, which develop into meristematic tissues. Okay, in which your cell in which the cell will does not stop dividing. Okay, so tumor, types of tumor, two types of tumor, benign and malignant. Benign tumor are those tumor which can be hindered, which can be stopped. The cancer growth can be stopped in benign tumor. But malignant tumor are those tumor in which cancerous growth cannot be stopped. Means these things can undergo metastasis, but these things cannot get under metastasis. Benign tumor can convert into malignant tumor, but malignant tumor cannot convert into malignant tumors. Okay, if benign tumor come under the contact of mutagens, then it will convert into malignant tumor, and if malignant tumor is been treated with anti-cancer drug, it cannot convert into benign tumors. Okay, so benign tumor and malignant tumor. Benign tumor confined to the place of its origin, they do not spread to other parts, cause little damage. Okay, means if a tumor develop in my skin in that area, if it is benign, it will remain here, it will not distribute in my body. Okay, but malignant tumor, it will develop in my body in this area, and it will show start doing metastasis, and it will spread down to the different part of the body. That thing is your malignant tumor, and it shows a deadly property which we call metastasis. Okay, cells lot out from tumor reaches other site of via blood, forming them a new tumor. This is called metastasis. Okay, metastasis is very deadliest property of malignant tumor in which it will circulate all over the body in different parts, and where it will stop, it will again start going tumor. Okay, next, cancer ke types. Okay, first we have carcinoma. This is the cancer of epithelial tissue. Then blood cancer, it is of two types, lymphoma and leukemia. Okay, and sarcoma, it is the cancer of your that connective tissue. Okay, so epithelial tissue types, different skin cancer, cancer of dermal tissue, lung cancer, cancer of neural that pleural tissue. Okay, breast cancer, cancer of breast. Then prostate cancer, it is happen in males. Okay, cervical cancer, it is a cancer of cervix. Okay, happens in female. Oral cancer, mouth cancer. Okay, adenocarcinoma, gland cancer. Okay, happens in the gland. Next we have cancer of connective tissue, that is bone cancer, muscle cancer and lymph node cancer. These are different types of cancer. This is in ectodermal and endodermal in origin, and your sarcoma is mesodermal in origin. Okay, now next, these are some example of cancer. This is your ameloblastoma, cancer of mouth. Okay, in which the whole bone is eaten by the cancerous growth. Okay, this is the CBC, this is the OPG of your cards, that ameloblastoma, in which whole the mouth is eaten by cancerous growth. Okay, this is the bone. Okay, in which cancer growth is present, and it is been done with the help of this sprinting method. Okay, next thing is your bone cancer. Okay, in which bone is grow like this. Okay, it is called odonto cancer. Okay, or odontonema. Okay, this is again an odontoblastoma, which happens here. Okay, next thing is your cervical cancer. It is a cervical cancer. Okay, next this is your skin cancer. Okay, this is basal cell carcinoma, this is squamous cell carcinoma, this is melanoma, and this is macral cell carcinoma. Okay, next is your cause of cancer. Okay, what are the causes of cancer? The causes of cancers are called carcinogens. Okay, so there is three type of carcinogen. First is physical agents. Okay, which is your ionizing radiation like UV rays and X-rays. Next is chemical agents. Tobacco smoke, major cause of lung cancer. Vinyl chloride, which will come and when your that pipes, okay, PVC pipe burns, the flumes which is coming out from the PVC pipe that produce vinyl chloride. Okay, caffeine, nicotine, mustard gas, all these are the chemical agents which causes cancer. Next is your biological agents, that is your oncogenic viruses, cellular oncogenes or proto-oncogenes, etc. These all are biological agents which causes cancer. Okay, next is cancer detection diagnosis. Okay, how can you detect the cancer? So first is biopsy. Okay, you take the tissue and treat the tissue and see the growth of the cancer of that tissue. That thing is biopsy. See, there is two type of study. When you study a living tissue, then it is called biopsy, and when you are reading dead tissue, then it is called autopsy. Okay, biopsy means studying, diagnosing, okay, of a living tissue, then it is called biopsy. When you study dead tissue, it is called autopsy. Okay, imaging techniques, CBCT, X-rays, OPGs. Okay, use of antibodies means monoclonal antibodies. Okay, molecular biological techniques. Okay, different interferon, alpha interferon, gamma interferons are also used to diagnose that you are having cancer or not. Okay, next, biopsy detail में. Okay, a thin piece of suspected tissue stand and examine under microscope. See, this is the injection for biopsy. You take the tissue from the that affected part and you will see that thing under the microscope. Okay. Next is your X-ray, CT scan or MRI. That are visual diagnostic features of cancer. Okay, you can see that cancer under the X-ray, CT scan and MRI. Okay, like this. Okay, in this you are seeing that this part is called lung cancer. Okay, in the previous slides you will see that this this is the imaging. Okay, this is your OPG. Okay, and this is again OPG. Okay, X-ray of face is called OPG, oropentogram. Okay, see, these all are the radiotherapies or different things to diagnose, so treatment of your that cancer. Okay, so first we have radiotherapy, in which you will use gamma radiations. Okay, chemotherapy, in which you will use different chemicals to treat cancer, and immunotherapy, in which you use immune response, means immunity, that immune bodies. Okay, anti immunities, and last one is your surgery. Okay, so surgery, radiotherapy, chemotherapy. In chemotherapy, we have three these chemicals: vincristine, vinablastin, and Taxol. Okay, these two are extracted from Vinkia rosa, and this one is extracted from Texas baccata gymnosperm. Okay, then immunotherapy, monoclonal antibody, and alpha interferon, or for Kaposi sarcoma, which happens in AIDS. Okay, next, drugs and alcohol abuse. Okay, so we have two types of drugs. Okay, one is stimulant, another one is depressant. Stimulant means it will stimulate your CNS, and depressant it will depress your CNS. Okay, so stimulant have three types: caffeine, cocaine, and amphetamine. Okay, cocaine is extracted from plant Erectoxylum coca. Okay, and it is native of North South America. Okay, next, depressant, tranquilizer, sedatives, hypnotics, and opioids. Okay, tranquilizer, sedative, hypnotics examples is phenethylamine, benzodiazepine, and barbiturates. Okay, and opioids are morphine, codeine, heroin, smack. Okay, heroin is modified morphine. Okay, when you do acetylation of morphine, heroin is produced. Okay, psychedelic drugs, which is vision producing drugs, means दिन में तारे दिखाने वाली drugs. Okay, in morning you can see stars when you take these drugs. Okay, LSD, it is extracted from fungus. Okay, dhatura, okay, the seeds of dhatura plant, it is halogenic in nature. Then atropa belladonna, it is used to produce medicines. Okay, and cannabis, charas, hashish, marijuana, bhang, complete shoot, and ganja. inflorescence. Okay, so next we have different these are opioids, cannabinoids, and coca-alcoids. Okay, so first we have opioids. Okay, opioids are the drugs which is bind with a specific opioid receptor present in our central nervous system and gastrointestinal tract. Example: morphine and heroin. Okay, heroin commonly called as smack, chemically diacetyl morphine, means you do acetylation of morphine and you produce heroin. Okay, which is a white odorless bitter crystalline compound. This is obtained by acetylation of morphine, which is extracted from the latex of poppy plant. This is your poppy plant. Okay, next, morphine is a very effective sedative and painkiller. It is given after major surgeries to person who get under the surgery. Okay, just like that organ transplant surgery, heart surgery, cardiac bypass surgery, neurological surgeries, or that nephronic surgeries. The person who undergoes these type of surgery, they will give morphine to them. Okay, because it is a potent painkiller. Okay, in market, in medical shop, you can get artificial morphine also in the name tradamol. Okay, it is an artificial morphine. Okay, it is the same just like morphine, but is a morphine which is produced inside that chemical procedure. So this is your morphine and this is your heroin. Okay, acetic anhydride, by heating and adding additional acetyl group inside the morphine molecule, you will get heroin. Okay, next is your cannabinoids. Okay, these are the group of chemical which interact with the cannabinoid receptor present principle in the brain. Okay, this acts on your brains. Okay, then the flower, top, leaves, and the resin and cannabis plant is used in various combination to produce marijuana, hashish, charat, and ganja. Okay, so these all are different products which will produce from cannabinoids. Okay, these days cannabinoids are also being abused by some sports person because it increases the intensity of to play the game. Okay, these are the different chemicals which is extracted out from the cannabinoid plant. Okay, next is coca alkaloid or cocaine. Okay, these are obtained from coca plant, erectoxylon cocaine, native from South America. Okay, South America, it is from your South America, native plant of South America. It is not present all over the world. It is grown in South America basically. It interferes with the transport of neurotransmitter dopamine. It increase the dopamine concentration inside your body. Okay, cocaine, commonly called coke or crack, is usually snorted. It will be taken by nose mainly. Okay, it has potent stimulating action on central nervous system, producing a sense of euphoria and increased energy. Okay, as it is a stimulant, it increases the power, that increase the alertness of CNS when taken. Okay, excessive doses of cocaine cause hallucination. दिन में तारे दिखना. Okay, now next is smoking. Tobacco has been used by human beginning for 400 years. Okay. Tobacco has been used by human being for over 400 years. Mughals started the use of tobacco in India firstly. Okay, previously in India there is no use of tobacco. When Mughals come in India, they start the production of tobacco. They start the cultivation of tobacco in India. Okay, it is smoked, chewed, and used as a snuff. Okay, it contains many chemical substances like nicotine and alkaloid, which stimulate adrenal gland to release adrenaline and non-adrenaline, causing high BP and heart rate. When nicotine goes inside your body, heartbeat and heart rate increases. Okay, smoking causes cancer of lung, urinary bladder and throat, emphysema, bronchitis, coronary heart disease, gastric ulcer, etc. All the respiratory disorders and disorder related to the heart is caused by smoking. Tobacco chewing causes oral cancer. Okay, smoking increases CO content in the blood and reduces oxohemoglobin. Okay, this causes O2 deficiency in body, due to which your body does not get sufficient amount of oxygen and due to which your body gets suffocated. Okay, next is your adolescence and drug alcohol abuse. Why adolescents do drug abuse? Okay, the reason behind the drug abuse. So adolescence, this is a period in which your child become mature in term to their own self. Okay, adolescence is the time in which a child become adult and it will goes towards the adultness. Okay, at this time that easily can be distracted from its path in the form of drug abuse. Okay, or alcohol abuse. Okay, so what is the cause of your drug, that cause of drug or alcohol use in adolescence? First, curiosity and experimentation. Means I have to do some tufani thing. Okay, चलो कुछ तूफानी करते हैं. ओके, नीड फॉर एडवेंचर एंड एक्साइटमेंट, सेम थिंग, चलो कुछ तूफानी करते हैं. टू escape facing problem. Okay, to escape the facing which you are, the problem which is you facing daily, to escape this thing. But it is not true. Drugs and alcohol does not escape the problems. Okay, stress from pressure of excel in academics and examinations, television, movie, newspapers and internet, etc. Bolo zuban kesari type 8. Okay, unstable or unsupported family structure and peer pressure. Okay, means the that parents are not supporting that particular child, it is not listening the problems of that child, due to which these things will happen. Okay, next thing is effects of drug, alcohol and abuse: reckless behavior, vandalism and violence. The person who is a normal deer, it will convert into tiger when it will do drug alcohol abuse. Okay, so it is not a good thing. Then coma, death due to the respiratory failure, heart failure, cerebral hemorrhage. Okay, drugs mediated with alcohol may cause death also. Means if you take drug and alcohol simultaneously, it can cause death also. Okay, then damage to the nervous system and cirrhosis of liver. Okay, and mental and social distress to family and friends. Okay, so these all are the effects of drugs on adolescents. Okay, next thing, effect of drug abuse on other thing also. Use of drug and alcohol on pregnant women affect the fetus development. Okay, if a pregnant mother taking alcohol or drug, it will affect that developing fetus. Okay, and that fetus which will convert into baby, it having mental retardness. Okay, next, loss of sexual drive and necrospermia in male. Okay, next, misuse of drugs by athletes, this is common nowadays. Okay, now warning signs of drug alcohol abuse in adolescence period. Okay, first, drop in academic performance and absence from the school. They do not go to school, they will go in different drug places to take the drug. Okay, or they can sit in the bar to drink alcohol. Okay, so this is the first drop in the academic performance. Second, lack of interest in personal hygiene. They do not take bath, they do not clean their teeth. Okay, they can do not do brushing. Okay, personally their hygiene has been decreased. Withdrawal and isolation. Okay, they will look like that, they will tell like that, सब मोह माया है, सब कुछ मोह माया है, we will live alone. Okay, they will live alone, they will withdraw and isolate from everything. Okay, then they feel depression, fatigue, aggressiveness, and rebellious behavior. They become rebellion. Okay, they will start doing bad things. They will do vandalism. They will destroy public property or their own property also. Okay, like this they are doing fighting. In this you are they seeing they are sitting in the road and doing smoking. See, they are living alone, they are not to go want to go in the group, and they do not feeling well doing study. So these all are the warning signs of alcohol, okay, and drug abuse. Next, change in sleeping and eating times. They will eat in night, sleep in morning. Okay, fluctuation in weight and appetite. Okay, their weight gain take place or their weight lose simultaneously take place when they will do drug abuse and that alcohol abuse. Okay, loss of interest in hobbies, they do not do any particular work. Okay, they do not perform any hobbies, they are lost their watch in the hobbies, they will not successfully doing in hobbies. Okay, again, deterioration relationship between family and friend, they will not sit there in the family functions, they avoid family gathering, they want to live alone. Okay, so these all are the warning signs of drug and alcohol abuse in adolescence age. Okay, next, what will the effect of drug abuse on male? Okay, first, premature baldness. Okay, जल्दी से गंजा हो जाएगा. Next, acne. Okay, acne on face, enlarged breast, etc. Okay, gynecomastia. Next, enlargement of prostate gland and can cause prostate cancer. Okay, increased aggressiveness, depression, potent for kidney and liver dysfunction, then reduction in the size of testes and decreased sperm production. These are all the effects of drug abuse and alcohol abuse on male. Okay, on female, increased aggressiveness, then masculinization means beard development on the face. Next, deepening of voice. The voice of female is of high pitch, it will become deep pitch like a male. Okay, next, abnormal menstrual cycle, then enlargement of clitoris, mood swings, and excessive hair growth on face. Okay, so these all are the effects of alcohol abuse and drug abuse on female body. Okay, that's all for your this chapter, fourth chapter completed. Experiment for DNA isolations. Mostly. Okay. Okay.
This much also. You want? One and do you want? Really you want?
Really.
Huh?
Okay, so now we Okay, now we are doing the experiment for DNA isolation on strawberry. Okay, Rahul sir is here for smashing these strawberries. Official killing is taking place. Official killing.
You need me to smash it completely.
हाथ से हाथ से
Wait. Hold this. Oh, wait, we'll remove it. How to remove the air? Remove the air.
हो गया.
No?
Now our paste is ready. Now we'll add water in this. Now little vim base. Now चुटकी भर नमक. चुटकी भर नमक. Now again we will smash it, mix it. Now you will see that our strawberry milkshake is ready. Now we will filter it by strawberry milkshake is getting ready. This is full of that cells of strawberry in this sludge in this slurry. We have the full cells of strawberry which we produce by smashing that strawberries. Now the main ingredient of this process, your ethanol, absolute chilled ethanol. Okay. Now we'll put this thing in this medium. Now you will see that some layering is coming. Okay, this layering, okay, is the DNA. Okay, DNA started to condense on the top of this beaker. You can see this white cloudiness, that cloudy structure which is coming on this beaker. This is your DNA. Okay, this is the DNA which is coming on the surface of this medium by adding your chilled ethanol. Okay, and this is the process which we studied in biotech. This is your DNA of strawberry. Okay, DNA of strawberry. See, Okay, so this is a glass full of DNA of strawberry. Break. Okay. Now it's time for 10 minutes break. After 10 minutes, we'll come back. Okay, so now it's time for evolution. Okay, so evolution in this chapter, in the starting of this chapter, your faith from God is being pulled down. Okay, you will think that initially your parents, you will know that everyone say that this world is created by God, but in the starting part of this chapter, you will think that world, earth, solar system, our universe is created because of some scientific reasons, some scientific reactions, some chemical reactions. Okay, so it's time for evolution. Okay, so evolution is the order of change from one form to the another form. Means a backward form will convert into its advanced form. Then it is evolution. Okay, and evolutionary biology is the study of history life form on earth. Means the different forms of life occur on earth, that study is come under evolution. Okay, just like this in image, we humans is evolved from monkey, apes. This thing is evolutionary history. Okay, so stellar distance, now this is the line from your NCRT. Okay, stellar distance are measured in light years, where look at the star on a clear night sky, we are in the way of looking back in the time. Okay, see the distance from one planet to another planet, or one star to another star, it is read it is given in light years. Okay, means when you are seeing a star in the sky, maybe that star is being disappeared or it will destroyed at the same time. Okay, the light which is coming from that star to your eyes, it is taking millions of the time. Okay, and that thing which we studied in light years. Okay, so in a way, looking back in the time, what we see today is an object whose emitted light started is generally millions of years ago. Okay, the light which is coming in your eyes now, at that time, at this time, the light which is coming from the star in your eyes, it started from that star to come on your eyes retina. It takes millions of years. Okay, and that thing is studied into light years. Okay, so generally millions of years back from the trillions kilometer away and reaching to your eyes now. However, when we see objects in our immediate surrounding, we can see them instantly. Okay, now just like this receiver is falling on my lower hand in the strength of time. I can see this in a seconds. But the light which is coming from the star, it is reaching to my eye in millions of years, taking millions of years. At that time, that star can be diminished, can be rebirth again. Okay, so that thing is called light year. Therefore, when we see stars, we apparently are peeping into the past. Means we are seeing into the past of that star. Okay, means the time that light reaching to your eye retina, at that time it can be happen that that star is destroyed, or that star is not even there in the atmosphere. Okay, so that thing we see, we say that when we see the star, we apparently are peeping into the past of that star. Okay, so the Big Bang theory state that universe originate about 20 billion year ago by a single huge explosion, and this theory is called Big Bang theory, proposed by George Lemire. Okay, it is again an important point that Big Bang theory is proposed by George Lemire. Okay, now next, how our solar system formed, how that solar star, sun, moon, Saturn, Uranus, Neptune, Mars, Venus, Mercury, or other planets is formed in our solar system. See, there was a spinning solar nebula. Okay, means a mass of planets, or a mass of base is revolving inside our solar system. That thing accumulate in the middle of our solar system. Okay, so solar nebula condense and the rotational momentum throughout a ring of cloud around the sun. Solar nebula means this yellow part is your solar nebula, and around that thing some particles, some matter is start to surrounding. Then this matter more condense, it will start to condense more, and the solar nebula condense further and form the sun. Okay, now this red center of your solar nebula convert into the sun. Okay, the sun condense further and become hotter and brighter. See this yellow circle, when in the starting it is red and it become hotter and more condensed and convert into the sun, and after millions of years the revolving area around that sun convert into our that 9 planets. Okay, the ring of cloud broke up into the celestial bodies, planet, asteroids, comet, thus forming our solar system. That's how our solar system is formed. First, it is a cellular nebula which revolving in outer space. Now it has become condensed. In the middle of that thing, a round big area has been condensed with having high temperature, and after millions trillions of years, that center part convert into the sun, and after million trillion years of that time, that near the sun, different planets, comets, asteroids are developed. That's how our solar system is formed. Again, in this thing, you will not gain that this thing is made by our God. Okay, this is a scientific phenomenon happening. Okay, now there is some theories to the related to the origin of life on Earth. First is theory of abiogenesis, theory of panspermia, theory of creationism, and theory of chemical evolution. Okay, so first we have theory of spatial creation. Okay, this theory states that the greatest supporter of this theory was Father Source. Okay, this is a mythological based theory. This theory has three content. All living organisms that we see today were created as such. Means when the earth is created at that time, the human which we are now, we are same at that time. Okay, second thing, the diversity was always the same since creation will be the same. Means the number of organism which is present on earth now, it is the same at the time when earth is formed in our solar system. Okay, then earth is about 400-year old. Okay, 4000-year old. Okay, it is not true. Again, our earth is not 4000-year old, it is billions-year old. Okay, so this theory is rejected again. Okay, next is cosmic panspermia theory. This theory states that aliens come on earth and they made humans. Okay, some scientists believe that life came from the outer space. Okay, early Greek thinkers thought that unit of light called spore were transferred to different planets, including Earth. Panspermia is still a favorite idea for some astronomers. Means they are saying that some spores, some particles come from different areas of space, and some particles accumulated on Earth, and it gives birth to the right life on Earth. Okay. Next is theory of spontaneous generation. This is a very useless theory. According to this theory, life on Earth come from mud. Okay, from raw material, from that dead material. Okay, the thing abiogenesis, okay, means life coming from abiotic things, that this theory supports. Okay, this hypothesis was supported by ancient Greek philosophers. According to this theory, life came out from decaying and rotten matter like straw, mud, etc. spontaneously. That's why it is called theory of spontaneous generation. Okay, they believe that the mud of Nile River could give rise to fish, frog, crocodile, etc. when warm under the light rays. Okay, useless. Okay, means according to these Greek philosophers, light falls on the mud of Nile River and it will give birth to crocodile, different animals in Egypt. Okay, so this theory is very waste theory. Okay, next theory is theory of biogenesis proposed by Harvey and Huxley. Okay, they state that new life can be originated on Earth only by pre-existing life, and this theory is the most appropriate theory. Means that living thing give birth to living thing. Okay, so experiment of Louis Pasteur, this experiment also state and also explain this theory easily. Okay, so in his experiment is also known as swan neck flask experiment, in which he take a swan neck like flask. Okay, so he prepared a sterilized syrup of sugar and killed yeast by boiling them in the in the flask. See, they boil a solution of syrup in which yeast is present, and then they boil that this yeast will die. Okay, after some time when they remove that heat, they will say they will see there is no growth in the flask. Okay, but when they break the neck of that flask and that air, that oxygen get inside that flask, they will see that microbial growth take place. Means life give birth to life. Okay, so he showed that the pre-sterilized swan neck flask life did not came from killed yeast because germ-laden dust particle, means the micro particles in the air were trapped by the curved neck. This curved neck may trap the microbe, but when you break this neck, that microbes can easily go inside that syrup and microbial growth take place. Okay, so this is theory of biogenesis. Next, Operan Halden theory or modern theory. This is the most accepted theory of life on Earth. Okay, so according to Operan, a Russian scientist, and Halden, an England scientist, proposed that the first form of life could have come from pre-existing non-living organic molecules like RNA, protein, etc. Means according to Halden and your opinion that life on Earth come from RNA, protein, carbohydrates, lipids, and DNA. Okay, operant theory was published in his book Origin of Life. First, life originated in sea water, so water is essential form of origin of life. Okay, as you studied in further in this chapter, that first life form on Earth it comes from water. Okay, not on directly on land. This is your Halden and this is your Operon. Open. Okay, so Alden soup. This is the soup means the liquid material. Okay, in which simple molecules come. Okay, these simple molecules become complex molecules, convert into complex molecules, which are the building block of life. Okay, and then they aggregate, they form a aggregate microsphere, which are now further develop and increase their mass and their number. Then they are called protocells. Okay, or we can call them protocells, a primitive form of life on Earth. Okay, now protocell plus nucleic acid and a cell replicating system created and which will convert into a cell. Okay, so this theory states that some non-living molecules like proteins, RNA, or your carbohydrates, lipids, they come together, and when DNA and self-replicating unit forms, the cell formation take place. Okay, next is chemical evolution theory or chemogeny. Okay, according to this theory, what will happen? First, there is the atomic phase. Okay, in which hydrogen, carbon, oxygen, nitrogen, sulfur, and phosphorus is there. Okay, now formation of simple inorganic molecule take place. Means these atomic phases react with each other and form some initial molecules on Earth like water, methane, CO2, and ammonia. Now these water, ammonia, these molecules react with each other and first primitive life molecule on Earth is formed that we call simple organic molecules or monomers. Okay. Now these simple organic molecules become more modified, become more complex, and convert into a large simple organic molecules like your purines, pyrimidines, amino acids, which are a simple molecules. Okay. Now these inorganic molecules, simple inorganic molecules convert into complex organic molecules which we call macromolecule, and this is the starter initiator of life on Earth. Okay, so next, some scientists also postulated that biological origin of life started how the first cell was formed. Now, in this theory, biological evolution, we are going to see how the first cell produced on the Earth. Okay, so these are the steps of production of life on Earth. First, aggregation of organic molecule. All the organic molecule, amino acids, your nucleic acids, carbohydrates, lipids, proteins, they come together and they forms and cellular mass. Okay, then that cellular mass convert into prokaryotic cell. Okay, the first life on Earth. Then evolution of that prokaryotic mode of nutrition take place due to which eukaryotic cell develop. Okay, now we are going to see this in series. Okay, so origin of protobionts. Protobionts mean the material, the organic materials like your amino acid, nucleic acid come in the action. These probionts do mutation, evolution, and convert into protocells, or we call ebionts. Okay, protobionts are the initial micromolecules which is present in the atmosphere of your primitive Earth. Then that convert into protocells. Okay, protocells which we are called ebionts. Okay, so nucleic acid develop the ability of cell duplication due to the sudden changes called mutation occur. In ebionts, mutation occurs. Due to that mutation, the life starts. So the point of life procession, the procession of life on Earth is mutation. Evolution come further in the play. First, mutation take place due to which evolution occur in the wild. Okay, so nucleic acid and proteins combine to form nucleoproteins. Nucleoproteins were the first sign of life on Earth. Okay, presence of nucleoprotein on Earth is the first sign of life on Earth. Okay, so cluster of nucleoproteins surrounded by lipid coat called protocell, the first form of life. Okay, means this is your lipid layer. Inside that lipid layer, you have nucleoproteins, and this structure is similar to like one organism which is present in our atmosphere now, virus. Okay, it looked like a virus. Okay, so that thing is the first form of life on Earth, means a protocell, okay, which is surrounded by lipid layer. It is the first sign of life on Earth. Okay, so these first non-cellular form of life could have originated 3 billion years ago. Okay, 3 billion years ago, first non-living cells come into the play, which is the initiator, activator of life on Earth. Okay, now next, these would have been giant molecules, RNA, protein, polysaccharide, etc. These capsules reproduce their molecule and perhaps means they start to multiply, they start to increase their number. Okay, next, origin of first cellular form, prokaryotic cell. Now these forms, these nucleic acid, these proteins, these polysaccharides start to doing reproduction, start to doing multiplication, start to increase its number in the atmosphere. Okay, so as a result of mutation, protocell become more complex and efficient to use material available in the surrounding medium and evolve into prokaryotic cell. Means your protocell ebions convert into prokaryotic cell by using the atmospheric substances which is present inside the atmosphere near them. Okay. Now next, the first cellular form of life did not possibly originate till about 2000 million year ago or 2 billion year ago. Okay, 2 billion year से पहले अपनी लाइफ कोई नहीं थी अर्थ पे. That life comes possibly originate till about 2000 million year ago on earth. Okay, the first living being were single celled bacteria like prokaryotic with necked DNA. Okay, their DNA is necked, they do not having any histone. Okay, they are probably chemo heterotroph and anaerobic because at that time there is no oxygen on earth atmosphere. Your earth atmosphere is reducing atmosphere, it does not contain any oxygen in the atmosphere, so it will perform anaerobic respiration. Okay, and second thing, it is a chemo heterotroph, means it prepare their food, it cannot prepare their food and do chemical reactions to perform its metabolic activities. Okay, now next, change in the mode of nutrition in early organism. So now they cannot produce their food, so they will evolve, they do mutation inside their body, and then now they will convert into from chemoheterotrophs to autotrophs. Okay, chemoheterotrophs absorb nutrition from their surroundings, they will convert into chemoautotrophs, synthesis organic food from inorganic material. Okay, now these chemoautotrophs again do mutation and convert into non-oxygenic photosynthetic organism using hydrogen sulfide to produce their food. Now again mutation will occur and oxygenic photosynthesis occur, they use water as a source of hydrogen, and this is the first point of view where the oxygen using oxygen producing organism come on the earth. Okay, cyanobacteria, BGA, the first organism which release oxygen in the environment of earth, that is your cyanobacteria. Due to this evolution of nutrition in prokaryotic cell, it induced the production of oxygen, and that organism which come on earth first, who produce oxygen, make your atmosphere with make full of oxygen inside your atmosphere, this is your cyanobacteria. Okay, next, oxygen revolution. Okay, now your BGS start to produce oxygen in high level, atmosphere is full of oxygen, now the reducing atmosphere of your earth convert into oxidizing environment. Okay, so liberation of free oxygen by cyanobacteria by revolutionary changes in the history of earth, it includes some major changes like primitive reducing atmosphere turn into oxidizing, which triggered oxidation of methane, ammonia, and produced life forms, oxygenatic life forms on earth. Okay, ozone appear which absorb ultraviolet radiation. Now from that point of view, mutation stops, the rate of mutation decrease because the reason of mutation, the mutagen which causes mutations in the pro cells, in the probionts, in the ebionts, in prokaryotes is your UV radiation. It is stopped to coming in earth atmosphere because of development of ozone layer. Now begin of aerobic life due to the production of oxygen on high level and production of ozone layer in the outer atmosphere of earth, due to which the earth atmosphere becoming aerobic from anaerobic. Okay, so origin of, now due to all these changes, the earth become home of eukaryotic cell. Okay, in earth you get different eukaryotic forms. So nucleus, mitochondria, other cell organelles develop in the cell and metabolically become more active. These free living unicellular eukaryotic organisms originate about 1.5 billion years ago in the primitive oceans. Okay, still there is no life on land. All thing is going in water. Okay, so this is the map of Earth development. Origin of universe 20 billion years ago. Then we have origin of Earth 4.5 billion year ago. Then origin of prokaryote 3.5 billion year ago, and origin of eukaryotic cell 1.5 billion year ago. Okay, and that thing is the starter of aerobic life on Earth. Okay, next, evidence in favor of chemical evolution. Now there is one experiment, Louis Pasteur, that Harley-Urian Standard Miller experiment, which will tell you that how life originated on Earth, how that initial biomolecules will come in the Earth. Okay, so they take and they will create an primitive Earth atmosphere in a laboratory by taking high discharge electrodes and that type of temperature which is present at that time. Okay, so in 1953, S.L. Miller, an American scientist, created similar condition at laboratory scale which were present in the primitive Earth. They took methane, ammonia, hydrogen, and water vapors at 800 degrees Celsius under the large flask. Same condition is present on primitive Earth. Methane, ammonia, hydrogen, water vapor is present at that time. When these inorganic things comes together and they mix in the high that temperature 800 degrees Celsius, and they combine and they form amino acids. Okay, amino acids which will the precursor of life on Earth. So this experiment shows that life originates on Earth due to some chemical reactions. Okay, he created electric discharge by using two tungsten electrode as source of energy. He observed the formation of simple amino acids like glycine, alanine, and aspartic acid. These amino acids are the precursors of life on Earth. Formation of these amino acids show that life on Earth come because of some chemical reactions. Okay, the similar experiment other scientists observe formation of sugar, nitrogen, bases, pigments, and fat. Again, this is the reason that life originates on Earth because of some chemical reactions which is present in the starting time of on Earth. Okay, so this is the again the experiment in the form of flow chart that performed by Stanley L. Miller and Harold U. Urey. Okay, apparatus used glass flask, a conventional liquid flask, interconnected white tubes. Okay, the setup apparatus following primitive Earth condition were maintained reducing atmosphere and ocean lightning by electrical spark of 75,000 volts. Next, pass four gases ammonia, methane, hydrogen, and water vapor in airtight apparatus and pass electric discharge of 800 degrees Celsius. And after that, all things mix up and we get some basic amino acid like glycine, aspartic acid, and alanine. Okay, these all shows that life on Earth started from a basic level, form of chemical reactions. Okay. Now next, Earth primitive atmosphere. As you know, Earth primitive atmosphere, it is reducing atmosphere. But when your prokaryotic cell get mutated and converted into BGA, it start to evolve in oxygen, and your Earth atmosphere convert into oxidizing nature. And that is the again is the this experiment, this thing is again the explanation, again the result that the Earth is created, Earth organism on Earth is created by different chemical reactions. Okay. Now next, evidence for evolution. What are the evidence of evolution? Okay, so first evidence is paleontological evidence, means your fossils, the fossils which you find in different areas of world. Okay, fossils are nothing, they are the remains, okay, or impigments of dead organism which is impressed in the inside the soil, inside the rocks. Okay, fossils are the imprints of bones and hard tissues of the organism. Okay, so study of fossils is called paleontology. According to Charles Lyell, fossils are impressions of remaining of hard part of life form found in the rock. Hard part means your bones and cartilages or hard tissues which is present inside your body. Many fossils are formed in sedimentary rocks. Sedimentary rocks are the soft form of rocks in which you will find the skeletons and the fossils of different paleontological animals, last animals. Okay. Next, different age rock sediments contain fossils of different life forms. Okay, the soil, the rocks belongs to different eras. Show you different types of animals which live, which rule that particular era. Okay, new forms of life have arisen at different time in the history of Earth. Evolution has taken place. The geological history of Earth closely correlate with the biological history of the Earth. The geographical history and biological history of Earth, they are mixing up together when you study the fossils. They are merging up with each other because they run parallelly to each other. Okay. Next, generally fossil found in older rocks are simple type and found in newer rocks are complex time, because they older rock means they are of old time. So old time having simple form of life, while the complex, that new stones, new rocks comprises of complex life form, because when you go from the that phylum Porifera to echinoderm, from echinoderm to mammal, the life form complexity increases. As same thing will happen in paleontological scale. Initially, the life is simple on Earth. Then it will get evolved, that can modify. So old rock contain simple fossil, while your new rocks contain complex fossils. Okay, evolution of horse was described by C. Mars. Okay, so this is the evolution of horse, in which you will see that initially horse having finger like us. Okay, an evolution take place that fingers get attached to each other. After some years, that will attach more, and at some time, that Pilochippus in Pilocene age, you will find that horse all fingers are fused and form a small hoop-like structure. And the modern horse equals now it having a big hoop-like structure to run. Again, this is the fossil of Archaeopteryx, which shows, which preserved inside the stone. Okay, in which you will see the bone structure of Archaeopteryx. Okay, next, geological time scale. Okay, so there is different periods, different eras come on Earth. Okay, which we study in a chronological order. Okay, see this. This is Parazoic era. Okay, which is the era which is going on now. The first era on Earth is Hadean era, then Archean era, then Proterozoic era, and now it is Paleozoic era. Okay, Paleozoic era which is running now on Earth. Okay, in Paleozoic era we have different small eras, like Paleozoic era, Mesozoic era, and Cenozoic era. That era which is running, which is a part of Paleozoic epoch. Okay, next we have periods: quaternary periods, Neogene periods, Paleogene periods. Okay, and the time which is running now, it is your Holocene era. Okay, sorry, Holocene epoch, which is ruled by your humans. Okay, in this you will see that some eras also, just like Permian, Triassic, Jurassic, Cretaceous, this era is ruled by dinosaurs on Earth. Okay, this Mississippian, Peninsulian, this is ruled by pteridophytes and some amphibians. Okay, and Devonian, Silurian, Odorian, and Cambrian, it is ruled by amphibians and Pisces on Earth. Okay, so these are different eras, geographical time scale which running on Earth from the starting time of Earth. So this Hadean era, this is the starting era of Earth. Okay, which is ruled by prokaryotic life, bacterias. Okay, so a brief account of evolution. So what is the brief account of evolution? What you will see during the phase of evolution. In brief account of evolution, you will see how Earth is evolved, how different life forms come on the Earth, and how that different life forms evolve from each other. Okay, so about 200 million years ago, the first cellular form on life is appear. Okay, about time of 500 million years ago, invertebrate were formed and become active. Invertebrate organisms come on Earth and they will become active. They will ruling the Earth. Okay, that is the time of Proterozoic era. Okay, in which your invertebrates ruling on the Earth. Next, jawless fish comes on the earth and evolved around 350 Maya. Okay, seaweeds and few plants existed probably around 320 million years ago. Next, the first organism that invaded land were plants. They were spread on land when animals invaded land. First organism which come on land is plant. After plant, animal come on land. The reason is that because in water they will does not getting sufficient food. Okay, in ecology we studied that the bioproduction, the production of food is more is of land than water. On earth, water is more, land is less, but still the productivity of land is more and water is less. That's why the organism come from water to land. Okay, the fish which snort out and strong fins could move and long land and go back to the water. This was about 350 million years ago. In 1933, a fish caught in South Africa happened to be coelacanth, which was proved to be extinct. Okay, coelacanth, it is the connecting link between Pisces and amphibian. It is searched in Africa, South Africa, and it think it is extincted, but it is a living fossil now, and it is present in the ocean in the surrounding the South Africa region, African region. Okay, these coelacanth lobe fins evolved into first amphibians that live on both land and water. Okay, as I say, Coelacanth is the connecting link between Pisces and amphibians. Okay, so this coelacanth ancestor comes on land and it can also go back in water and convert into amphibians. Okay, the amphibians evolve into reptiles that develop scales. Their egg become become full of calcium. Okay, it contains calcium on a high concentration. That thick shelled egg which do not dry up in the sun, like those conditions. Again, we only see their modern day descent, the turtle, tortoise, and crocodile. These all are the ancestors of reptiles. Okay, which are present. मतलब जो dinosaur which are whose ancestors, that crocodile, turtle, and their ancestors is dinosaur, which is evolved from amphibians. Okay, next, synapsids were the mammals like early reptiles which give rise to mammals. Okay, in this scale, you will see that there we have a common ancestor. This is a amphibian less reptile. Okay, from this we have two evolutionary scales. One is sauropsid and one is synapsid. So this sauropsid is responsible to give birth to your reptiles. Okay, turtle, lizard, snake, tortoise, and crocodiles. Okay, and this synapsid will go and give birth to your mammals. Okay, same thing is here. Sauropsid were Sauropsid were lizard-like early reptiles which give rise to different dinosaur, modern reptiles, and birds. And that synapsid is the ancestor of today's mammals. Okay, mammals evolved from synapsids, and your reptiles, modern reptiles, dinosaurs, and birds evolved from sauropsids. Okay, next, in the next 200 million year or so, reptiles and differentiate shape and size and dominant earth. That is your Jurassic era, Triassic era. Okay, in that era, reptiles rules on the earth in the form of dinosaurs. Okay, giant forms were present, but they all fell to form coal deposit slowly. That thing is present now in Dubai region. Okay, that all the Gulf countries, that region is occupied by pteridophytes, and now they are converted into fossil fuels, coal, petroleum. Okay, next, some of them land reptile were back into the water to evolve into fish like reptiles probably 200 million year ago. Example, ichthyosaurus. Okay, you watch the movie Jurassic World. Okay, in that Jurassic World you see a dinosaur coming out from that water ring. That thing is ichthyosaurus. Okay, some reptiles, they went back inside the water and convert into fishes again, and this thing example is your ichthyosaurus. Okay, it is the top predator of that time in the oceans. Okay, even T-Rex have the fear against this ichthyosaurus. Okay, the land reptiles were of course the dinosaur. The biggest of them was Tyrannosaurus rex. Okay, about 20 feet in the height and had huge fearsome dagger-like teeth. Okay, about 65 years ago, Maya, the dinosaur suddenly disappeared from the earth. We do not know the true reason. This may be happened due to climatic change killed them, or most of them evolved into the birds. Third reason, meteor collision killed them. The truth is still known. See, these are the three reasons. First is climatic changes. Second one, that turn into the bird. As you know, the body design of hen is similar to T-Rex. Okay, but still we do not have the confirmed result that how dinosaurs disappear from the earth. But still there is one, we cannot say that it's a fully confirmed result, fully confirmed reason of extinction of dinosaur from earth, that is meteor strike. Because in that North America we have a crater which is large enough. If this crater is made due to the meteorite, and when we do the carbon dating of that area stones, we will find that it is near that of Jurassic era. So it tells that there is a potent reason behind that extinction of dinosaur is meteorite collide. Okay, next thing is your small size reptile of that era still exists today. Crocodile, turtle, tortoise, lizards, okay, snakes. These all are the small size reptiles of that era. Okay, the first mammal were like shrew, their fossils are small sized. Mammals were viviparous and protected their unborn young inside the mother body. Mammals were more intelligent in sensing, avoiding danger, at least so. Now this era, Holocene era, is ruled by mammals. Okay, next we have, this is the evolutionary line of different animals which is present on earth, and the past of the animals which is present on earth. Okay, now next is evidence from comparative morphology and anatomy, that is the most important part of this chapter, homology and analogy. Okay, these are the similarities of two types, homology and analogy. So homology, when organ which have common origin, embryonic development and some fundamental structure but perform similar or different function, they are called homologous organ. Okay, means their function is different but their anatomy is similar. Okay, then this type of thing is called homology. Okay, example of homologous organs or homology is first four limbs of mammals. Okay, see this. These all are anatomically similar, but their function is different. Okay, legs in male is for walking, legs in cheetah is for running, flippers, which is the modified form of four limbs in whale, is for swimming, while in bat it is for flying. All our structure are similar, bones, name, bone design are similar, but function is different. Then it is called homologous organs. Okay, then second example, thorn of bougainvillea and tendril of cucurbitate. Okay, these all are similar in that structure, but their function is different. One is for protection and one for that crawling on long trees. Okay, all vertebrate hearts, their structure, their design is that same, but the function is different. Okay, all vertebrate brain. Okay, again that is the example of homologous. Okay, mouth parts of insects, different class of insects have same type of mouth but their function is different, some for sucking, some for cutting, some for that piercing. Okay, next is potato and ginger, both are modified shoot. Okay, but functions is different. Okay, next radish and carrot, both are modified root but both have different functions. Okay, molecular homology found on the molecular level. For example, the plasma protein found in the blood of man and apes are similar but their functions are different. Okay, so these all are the examples of homologous organ. Next we have analogy. Okay, structure is different now but function is same. Homology, structure is same, function is different. Analogy, structure is same but function is, sorry, structure is different but function is same, that is analogy. Okay, example, wings of butterfly and bird. Okay, both have same function, flying, but their structure is totally different. One is made up of chitin, other is made up of bones. Okay, next is eye of octopus and mammal. Okay, both function is same, vision, but their design, their structure is totally different from each other. Next, flipper of penguin and dolphin. Okay, both having same function, that is swimming, but their design is different from each other. Okay, next, sweet potato and potato. Okay, both are having same function, that is storage of food, but structure is totally different from each other. Okay, so analogy does not indicate common ancestry and it is based on convergent evolution. See, there is two type of evolution, divergent and convergent. Divergent means it is diverging from the point, it will going away, and converging means it is coming from from two different ways and mixing in a same point, that is your convergent. Okay, and divergent means it is coming out from a same point but is diverging different ways. Okay, so these are the examples of your analogy. This is your eye of octopus and that eye of human. Both work is same, that is vision, but the shape, size, anatomy of the eyes is totally different. Okay, that's other example, wings of birds and butterfly, potato and sweet potato, and flipper of penguin and dolphin. Okay, now next is your adaptive radiation, which is an example of divergent evolution. Okay, its example is your Darwin finches. Okay, they have come from in land from same point. Their origin is same point, but they will go in different atmosphere, different areas, different geological areas, and they adapt according to the nature of that area. That thing is called adaptive radiation, and adaptive radiation always follows divergent evolution. Okay, so the process of evolution of different species in given geographical area starting from a point and literally radiating to other area of geography is called adaptive radiation. See, these finches are evolved from a common ancestor. Okay, when they will radiate to different geographical areas, from that point they will find different type of food. According to that, their beaks develop into different types. That is your adaptive radiation. Okay, example, Darwin finches. Okay, next example is your Australian marsupials. A number of marsupial, each different from the other, evolved from ancestral stock but all within the Australia island continent. See, this is Australian continent, marsupial radiation, Australia. They will go in different area of Australia and adapt to different condition. This is again. Okay, placental mammals. Placental mammals, again, we can say that this is an example of adaptive radiation in which one common ancestor of placental mammals go in different areas and evolve to different area and perform different functions. Okay, so these are your placental mammals and these are your marsupial mammals. Okay, next we have convergent evolution or adaptive convergence. Okay, which follows convergent evolution. Normally, that is your meaning that coming from all the point to a single point. Okay, so when more than one adaptive radiation appear to occur in a isolated geographical area, one can call this convergent evolutions. It is opposite to divergent evolution. In divergent, we are going away from a single point, but in convergent, we are coming to a single point. Okay, so convergent evolution always go with parallel evolution. Okay, when adaptive convergence is found in closely related species, it is called parallel evolution. Okay, means two different species evolving parallelly to each other. Okay, just like marsupial mammal, placental mammals, they are evolving in same area of Australia. Okay, parallel evolution occur when two independent but similar species evolve in same direction, thus independent acquired similar characters. For example, this placental mammal mole, Australian mammal marsupial mole, both are mole, but both have different structure, but they evolve at the same time and in the same place in Australia. All these are the example of your parallel evolution or convergent evolution. Okay, so this is your placental mammals mole, marsupial mole, placental mammal anteater, Australian marsupial numbat or anteater, mouse, marsupial mouse, lemur, spotted cuscus, flying squirrel, flying phalanger, bobcat, Tasmanian tiger cat, wolf, Tasmanian wolf. Okay, so these both animals present in Australia, but they evolved in the same pace. Okay, so this is your parallel evolution. Okay, next is your evidence from vestigial organs. Okay, so some organs in your body is now not of use, but they are still present, and that shows that it is present in your ancestor. In ancestor they are working, but in your body it is not working. So these type of organs are called vestigial organs. Okay, okay, for in humans this is cecum and vacuum appendix. Okay, nictitating membrane, caudal vertebrae, wisdom teeth, third molar. Okay, third molar of your buccal cavity, it is vestigial organ. Okay, second thing is your caudal vertebrae. Okay, caudal vertebrae means the vertebrae which forms tail in monkey. It is not making tail in your body. Okay, but it is present in your body. Okay, nictitating membrane, its small part is present in your eye, but it is not working, but it is present in amphibians, some reptiles. Okay, in birds also. Okay, so it will, when the bird fly in the sky, their eyes can easily hit by particles present here. So nictitating membrane is there, it covers the eye, but it is a transparent membrane. Same thing in crocodile, in some reptiles, in all amphibians, nictitating membrane is present and they cover the eyes. Okay, they are the remnant organ which were complete and functional in their ancestor. In ancestor they are functional, but in now they are not having some work. Okay, so these are vestigial organ, and again show the evidence that we evolved from someone that contain that organ and that organ functional in their body, but in our body it is now not functional. Okay, next is your evidence from atavism, reversion. Okay, means developing characters of individual of ancestor inside your body, means reappearance of primitive characters, primitive structure inside your body again. So these are called atavism or reversion. Example is tail in newborn baby. Okay, see this tail. It is present in our ancestor, but now it is not present in our body. Okay, extra long and potent canines. Okay, this shows that our ancestor purely eat meat. Okay, long and thick body hair. Again, it shows that our ancestor contain that thick hair in their body, and it will develop in some persons. Okay, extra nipple in females, this is again show that our body contains extra nipple which is functional in females. Okay, next cervical fistula. which is present in neck. This shows that we evolved from some ancestors which contained gills in their neck area. Okay, next we have evidence from connecting links. Okay, the two different organisms which share the same properties of different phylums, then it will call connecting link. Okay, just like your virus, connecting link between living and non-living. Bellengloss is connecting link between chordate and non-chordate. Chimera, connecting link between cartilaginous fish and bony fish. Platypus and Echidna, connecting link between reptile and mammal. Then Archaeopteryx, one of the most famous connecting link between reptile and bird. So connecting links tells that that organism is sometimes some other phylum property showing, and sometimes it will showing other property of that phylum. Okay, so one of that Archaeopteryx we have, it is a missing link between reptiles and birds. The connecting link which are not found in the present times are called missing links. So there is two type of thing. One is connecting link and one is missing connecting links. Missing connecting links are those connecting link which is extincted from the earth now, and connecting link which is present. Platypus is connecting link which is present on earth now. It is a connecting link between mammal and reptile, or mammal or bird. Okay, Archaeopteryx as missing connecting link because it is extincted from the earth now. Okay, so the characters of reptilian present in Archaeopteryx is long lizard like tail and free caudal vertebrae. Okay, this is a reptilian character. Okay, that but the caudal vertebrae which is present on the tail region, it is free, which is the characteristic of reptile. But birds, in birds this character is not present. Next is teeth present in jaw. You can see the teeth. Any bird today do not have any teeth in their jaw. Okay, next is weak sternum. Okay, weak sternum is the characteristic of reptile, but your birds and mammals, they are characterized for strong sternum. Again, the property of reptile. Next, non-pneumatic bone. Their bone is very heavy. As you know, birds fly, so they have to reduce their body weight. So their bone become hollow, which we call pneumatic bone. This is absent in Archaeopteryx. Then next, bird character. Forelimb modified into wings. That is the most important character of bird in Archaeopteryx, that they can fly. They can fly for small distance, but they can fly. Okay, next, feather on the body. Second characteristic of bird, that their body is covered with feathers. Next, jaw modified into beak. You can see here they do not have any jaw. Okay, mandible, maxilla they do not have, which is present in your crocodile, turtle, reptiles. Okay, but in birds we do not have any mandible and maxilla. On the place of mandible and maxilla we have beaks. Okay, so this is the characteristic of bird. Next, hind limbs built in avian plane. You can see that hind limbs of Archaeopteryx is just like birds. Okay, that avian plane means it is just like a aeroplane. Okay, once it is fly, it will sub down, means it will condense down, means if it is straight, when it will fly, it will like make like this. Okay, when they fly, it will rule inside. Okay, so this is your connecting on the evidence on the basis of connecting link. Next, evidence from biochemistry. Okay, on microbiological level, on biochemical level, we can also see evidence. Okay, different organisms have same biochemistry, that shows that our ancestor contain that type of biochemistry, that type of protein, that type of carbohydrate present in the same ancestors. Okay, next is evidence from embryology. Okay, the father of embryology, von Baer, said that organism in their embryonic stage, either it is reptile, either it is ape, either it is mammal, pisces, amphibian, they shall look similar. Okay, you can see in this image also. See, there's misprinting. This is fish and this is salamander. Okay, so this is the embryo of fish, this is the embryo of salamander, this is the embryo of chicken, this is the embryo of rabbit, and this is the embryo of human. You cannot differentiate in all these. Okay, they look similar to each other. So this tells that there sometime that embryological history says that the organism evolved from a same ancestor which look like same to each other. Okay, but when we go down, okay, we will see that we develop into different. Okay, see, this is your axolotl. Okay, this is your this is your axolotl, this is a fish, this is chick, this is the fetus of your rabbit, and this is the fetus of human. Okay, but in the initial stage of embryonic development all look same. Okay, so this shows that we evolved from a same ancestor. Okay, next is your evidence from biological distribution. Okay, distribution of organism in a biological, in a geographical area also says that we evolved from a same point of view. Okay, the same point of view, the same region, the same areas we evolved from that. Okay, that come under evidence of biogeographical areas, biogeographical regions. Okay, so these are different biogeographical areas according to the Wallace. That is Nearctic, which is now known as North America, Ethiopian, which is now known as Africa, then Oriental, which is now known as Asia, Neotropical, which is now known as South America, then Palearctic, which is now known as Europe, and Australian, which is now known as Australia. Okay, and he divided that Earth map in different areas in which different biological hierarchies, biological development take place, and then again shows that we develop from a common ancestral areas. Okay, now theories of organic evolution, Lamarckism, Darwinism, all these things we are going to discuss in this. Okay, so first theory is Lamarckism. Okay, theory of inheritance of acquired character. See, according to Lamarck, what he says, that thing which we acquired now, it will go inside our progeny. Means if I am a swimmer, then my baby will be swimmer. Okay, if I am a cricketer, then my baby will be cricketer according to Lamarck. Okay, so the first logical theory of evolution was proposed by French naturalist Jean Baptiste de Lamarck in 1742 to 1829. Okay, basic concept of Lamarckism, that internal vital forces run the evolution, effect of environmental makes new things, new needs. Okay, use and disuse of organs will produce evolution, and inheritance of acquired character. Okay, these 3 points can show evolution, but this thing cannot show the evolution. That is the not the result of evolution. These can produce evolution, but these points cannot produce the evolution. And we give the example of long neck of giraffe. Okay, if in the African savanna, that giraffe height is very short. Okay, but the tree's fruit is very on height, very long on the height. So every year, every time of year, that giraffe want to eat that thing, so it increase the length of its height, neck. Okay, so he disused that thing and increase the height of that neck, and now you get the long headed giraffe. This is again a wasteful theory. Okay, second example of this, aquatic birds stretch their toes and develop into web. As you know, duck, they have some web like structure between their fore in the hind limbs. Okay, so Lamarck says that they want to swim, they don't want to paddle in the water, so they will stretch their skin between their fingers, and they develop pads. Again, this is not a logical theory. Okay, and last one, snake lost their leg. Okay, their long body cannot run with legs, so they lost their leg, and they will move like a, so to move fast they will start doing crawling. That is again a wasteful thing. Okay, so Lamarck had said that evolution of life from had occurred but driven by use and disuse of organ. Nobody believes this conjugate anymore. Okay, according to Lamarck, what use and disuse of organ makes evolution. If you want that organ, if you do not want that organ, you will evolve. Okay, that is the basis of Lamarck theory, but it is not true. Okay, and that thing is discussed by Weismann theory of continuity of jump laws. According to your Lamarck, the thing is that from father to son, from the progeny to other progeny, the acquired character will go, but it is not true. Okay, and that Weismann do an experiment on rat. Okay, in which he made a tailed mouse with non-tailed mouse, and he said that all progeny which is coming having the tail. Okay, so again it will discontinue the law of Lamarckism. Okay, next, according to this theory, two types of protozoa are present in organism, which is germplasm and somatic plasm. Germplasm is responsible for the character, inheritance of character. Somatic plasm is not responsible for the inheritance of character. Okay, so second example is boring of ear, pinna, and nose in Indian women is never inherited to the next generation. Okay, if I have my ear having this thing, okay, so my son do not have that hole inside that ear. Okay, so this is the again second example that inheritance, acquired character does not go inside the progeny. Okay, next, Chinese women used to wear iron shoe in order to have small feet, but they still have normal feet in their child. Okay, in Chinese tradition, they say that small legs make women beautiful in China. So they will wear small shoes and their leg look like that. All the fingers get fused, but their children does not have this type of thing. Okay, they have normal feet. Okay, so all these acquired character does not go inside the progeny, because all these things happening in somaplasm, somatic cells, all these things happening in somatic cells. Somatic cell does not inherit their property in next generation. Only changes in germ cell will go inside the next generation. Either it can be evolution or it can be mutation. It can be due to evolution, it can be due to evolution, mutation. Okay, so next is your most accepted theory, Darwin theory. Theory of natural selection means selection of best. Okay, selection of the fittest. Okay, Charles Robert Darwin was born in 12th February 1809 in England. Okay, Darwin traveled by HMS Beagle ship to South America, South Africa, Australia, and Galapagos Island. Okay, this is the ship of your Darwin. Okay, Charles Darwin, the child of Charles Darwin, its name is Francis Darwin. ओके, जो चार्ल्स डार्विन का बेटा था उसका नाम था फ्रांसिस डार्विन. ओके, डार्विन वाज इन्फ्लुएंस्ड बाय टू बुक, प्रिंसिपल ऑफ पॉपुलेशन ऑफ मैथड्स एंड प्रिंसिपल ऑफ जियोलॉजी बाय चार्ल्स लेल. ओके, Alfred Wallace, a naturalist who worked in Malice, Archipelago, had also come to similar conclusion around the same time and sent his conclusion to Darwin in the front form of chart. Okay, this Alfred Wallace who gives that map of Earth in different regions, the evidence of biogeographical era. Okay, it works with Darwin and explain its work to Darwin and tell that there is something you are telling is true. Okay, then this theory was later explained by Darwin in his book on the origin of species by means of natural selection in 1859. Okay, so basic concept of Darwinism first is branching descent and natural selection. Okay, are the two key concepts of Darwinism. Theory of evolution, natural selection is based on the certain observation which is factual. Okay, branching descent is the evolutionary process where a single species split into two or more new species which then evolve separately, like this. See, if there is species A, okay, it will get in other atmosphere, it will reach in different isolated geographical area, then it will can convert into A1 and A2. Now this A1 and A2 is totally different from each other and also different from A. So we have a new two species developed from this A. This A developed two new species A1 and 2, and this is called branching of descent. Okay. Next thing is your natural selection. Natural selection is a simple theory in which it say natural select the fit one. Okay, who is physically, reproductively fit, he will select it, he will transfer this next progeny, it will produce next progeny. Okay. Next thing is your Darwinism theory of natural selection. It will have the form for some points. First is heritable minor variation. Okay, hereditary property, it shows minor variations. Okay, overproduction, population size grow exponentially due to the maximum reproduction. Example, bacterial population. Then limited natural resources are not increased in accordance with the population size. Means if population increase, it is not that resource size also increase. Okay, the one person who will survive on limited resource, it will be select, it will be remain in that particular ecosystem. Okay, next, struggle of existence. Okay, it is the competition among organisms for resource so that population size is limited. Okay, struggle for existence. Okay, you have to struggle in every point of your life. If you do not struggle in life, then you do not facing your life properly. So struggling in life is the struggling of your existence. Okay, and the most important point that Darwin says is the survival of the fittest. Okay, the struggle for existing organism with beneficial variation can utilize resource better, hence they survive and reproduce. This is called survival of the fittest. The fittest one, the person, the organism, the animal, the plant who can survive in the best way, who will utilize its resources in the best way, it will survive and that become the king of that particular ecosystem. Okay, so it leads to the change in population characteristic and new form of life appears. Okay, Darwin ignore about origin of variation and mechanism of evolution or speciation. That is the mistake done by evolution, that done by Darwin in the theories of that Darwinism. Okay, and this thing is corrected with the help of theory of mutation by Hugo de Vries, and further we studied about new Darwinism. which is again corrected theory of Darwinism. Okay, Darwinism का correction था neo Darwinism. Okay, so next theory is theory of mutation theory given by Hugo de Veris. Okay, this theory was proposed by Hugo de Veris based on work on evening prime roles. Okay, large difference arising suddenly in a population called mutation. According to Hugo de Veris, mutation runs produce evolution. Okay, the cause of evolution according to the Hugo de Veris is mutation. Okay, actually mutations are sudden changes of genetic material and hence all are inevitable. It is correct. Okay, in addition to recombination, mutation is another phenomena that lead to the variation. Recombination is the normal phenomena which make and which induce, which may evolve the organism, which create new characters in that organism. But mutation is a rapid one and it will make more rapid and more sudden changes in the physiology and genotypic or phenotypic appearance of that organism. Okay, mutation is discontinuous source of variation and provide raw material for evolution. Okay, again this line is very important. Mutation is discontinuous source of variation and provide raw material for evolution. Mutation is discontinuous. It can be happen in reverse, it can be happen in forward way. Okay, and provide raw material for evolution. Okay, if you want to make something, you have first required a raw material. From that raw material you will prepare some food. Just like when you are preparing some vegetable in your house, in the starting you will require raw vegetables. Okay, then raw vegetable you cook and then you will prepare a dish. Okay, same thing, the raw material for evolution is mutation. Mutation causes evolution. Okay, according to Hugo de Veris, it is mutation which cause evolution. Okay, that I said now, and not the minor variation. Minor variation से कभी evolution नहीं होता. Okay, means just like you are not same to your dad and mother. Okay, same thing this thing here. Okay, that small variation never do evolution according to the Hugo de Veris. It's mutation which causes evolution. Okay, then mutations are random and directional lengths while Darwin variation are small and directional. According to Darwin, that variation which is coming inside the organism is small and it is direction. But according to the Hugo de Veris, the mutation it is directional less and random. Anytime it can be happen. Okay, evolution for Darwin was gradual, while Hugo de Veris believed mutation caused speciation and hence called as saltation. Single step large mutation. Okay, due to which new species formation take place. Okay, criticism, what is the criticism upon this theory is that natural mutations are not very common as Hugo de Veris thought. Okay, mutation is not happening every day, it is not a everyday process, it takes millions of years. Okay, or thousands of years or hundred of years, but it is not happening regularly in a day, in a hour, in a minute. Okay, second, mutations are normally recessive and harmful while the characters taking part in evolution are usually dominant. Okay, in mutation the character which is expressing that are recessive. Okay, but in evolution we studied that characters, that phenotype and genotypes are of dominant type. So this is the criticism of Hugo de Veris theory. Okay, next we have neo-Darwinism or modern synthetic theory of organic evolution. Means it is nothing, it is repair, it is refreshment of Darwinism. Okay, advancement of Darwinism, modification of Darwinism is called neo-Darwinism. Okay, so neo-Darwinism is a modified form of Darwinism along with recent research of Weissman, De Veris, Stebbins, Dubinsky, Swellwright and Mayer. That much number of scientists do that thing to make that Darwin's theory modified and which will we know now as a neo-Darwinism. Okay, so according to this theory, following factor responsible formation of new species. First is rapid multiplication, which is we studied in Darwin. Okay, limited food and space, it is again studied in the Darwin. Struggle for existence, this thing also studied in Darwinism. Okay, genetic variation, this thing is not told by your Darwin, which is added by these guys in this neo-Darwinism theory. Okay, and natural selection, this is told by Darwin, and isolation, this is again told by Darwin. Darwin does not tell us about genetic variation. That is the modification in the theory of Darwin. That is addition of genetic variation. Darwin do not know what is genetic variation, how genetic variation is occurring. But these guys know about that and they add genetic variation in Darwinism theory and we get neo-Darwinism or modern synthetic theory of evolution, organic evolution. Okay, so genetic drift, which is given by Swirl-Wright, which is also called Swirl-Wright effect and given by Swirl-Wright. Okay, so random change of gene or allergic frequency in a population merely by chance is called genetic drift. Okay, genetic drift, just like a car making a drift. Okay, normally a car is going in straight when it do a drift, so it will turn around. Okay, same thing happen in a population also. A normal population is flowering, occurring in, and suddenly a mutation take place, and due to that all the phenotypic effect, all the phenotypic character of that species as goes down, new phenotypic effects is developed inside that species. That thing is genetic drift. Okay, it operates rapidly in small population. One thing to be remembered, genetic drift always occur in small population, not in large population. Okay, it is due to the habitat fragmentation, isolation, natural calamities or any epidemics. Okay, these are the reason of genetic drift. These how this genetic drift occurs, that is habitat fragmentation, isolation, natural calamities and any epidemic like corona. Okay, founder effect and bottleneck effect are the two forms of genetic drift. First one is your founder effect. It is a genetic phenomena that occur when a small group of individuals from a large population become separated and form a new subpopulation. See, like this. This is the area of population A, B, C, D, E. Okay. Now from this area, your E population go out and live in a new area, and it will develop new character which is not found in A, B, C, D now. Your E is cut it down from this geographical area and go in new geographical area and start to living there. And this new geographical area have new conditions which is not present in this area. So they develop new adaptations in their body. So this adaptation which is developed in species E, it is not present in A, B, C, D. So this effect is called founder effect. Okay. Next effect is your bottleneck effect. Okay, this occur normally. Okay, ये वाला इफेक्ट जब होता है जब किसी भी कंडीशन में नेचुरल कैलेमिटी आ जाती है, just like some type of natural disaster, earthquake, it can be earthquake, it can be volcanic eruption, it can be cyclone, it can be tsunami. Okay, so the bottleneck effect is a genetic phenomena that occur when a population size is significantly reduced due to environmental or human caused events. It can be human caused event like forest fire. Okay, all other that pandemics. Okay, like corona. Okay, all these are human cause effect, and natural environmental like tsunami, earthquake. Okay, so see, by this example you can know that this is a parent population in which you will see there is blue and yellow color. Okay, now something will happen in this particular event, particular population. Okay, so drastic reduction in population take place. See, this is the population, these all things which is present in this bottle, it will die, only this population remain. Now this surviving individual will reproduce and you will see in next generation the number of blue increase and yellow decrease, because in this natural calamity time, during that human cause event, only blue survive more and yellow survive less, due to which when you see in next generation, blue number is increased and yellow number is decreased. That thing is your bottleneck effect. Okay, I'm again explaining that founder effect. Again there is this, there is one geographical area A, B, C, D, E, in which your E species escapes from that area and live in new area. This new area have new geographical adaptation, new geographical condition which is adapted by E. Now that character which is present in E is not seen in your A, B, C, D. Okay, and this E species now is totally different from A, B, C, D. Okay, this is your founder effect. And bottleneck effect, when the sudden natural calamity or a human caused event take place, the one who survive more in that calamity, this generation will go in next generation, and the one who cannot survive in that condition, it remains less. Okay, that is your bottleneck effect. Okay, next, natural selection. Okay, so the example of natural selection is your industrial melanism. Okay, the phenomena was studied by Barak Kettlewell in England. Okay, so in England we have two species of moth. One is white wing, another one is black wing. Okay, so white wing and black wing, which is our scientific name is Betullaria, and the black one is your Carbonaria. Okay, so these two lives in England peacefully. Before industrial revolution means formations of industries, factories. This look like this. Before industrial revolution, okay, before industrial revolution there is no pollution in England. Okay, so lichens are growing easily. Okay, and the color of lichen is white. Okay, so this white moth species can easily camouflage with this lichen. Okay, and black species, it cannot camouflage with light, that white lichen. Okay, so it easily eaten by birds. Okay, now after industrial revolution, now pollution increase in the atmosphere, sulfur dioxide concentration increase in the inside the atmosphere, lichen start to die, and the color of lichen when they die it become black. Okay, now the lichen of black color easily camouflage by your black moth, and when the after industrial evolution when pollution is high, your black moth survive more and white moth survive less, while before industrial evolution when there is no pollution in there, lichen color is white, that white moth camouflage with lichen and it is not eaten by your birds, black is eaten, the number of black moth is less, and after the industrial evolution in England the number of white moth is less and black moth is increased. That is the natural selection. Who camouflage best with the nature it will survive, and the one who cannot camouflage with the nature it will easily eaten by their predators. Okay, so this is an example of natural selection. This is before industrialization you will see more number of white moth, and after industrialization you will see black moth number is more in the atmosphere, in the environment of England. Okay, see this is a life example. Okay, this is your white lichen in which black moth is doing camouflage, and this white moth is white moth doing camouflage, and this black moth is easily visible and it is eaten by your bird. But after that industrial revolution you will see that all the lichen will die, and this is your black moth which is easily doing camouflage with that black background, and this white moth cannot do camouflage now, and this is easily eaten by that birds. Okay, next is your drug resistance. Drug resistance again in bacterium is against the example of evolution, that natural selection. The bacterium who can easily develop resistance against the antibiotic, they can withstand, and the bacterium who cannot develop resistance against the antibiotic, they can easily destroyed by that antibiotic. Okay, the natural selection is the best process of evolution. Okay, next sickle cell anemia and malaria. Okay, it is again the example of natural selection. Okay, the persons who having normal blood, they can easily get malaria. Okay, while the persons who have sickle cell anemia, their red blood cells are just like moon. Okay, this is your normal RBC. Okay, and this is your sickle RBC. Okay, so Malarian parasite can easily live in this, do its sexual cycle or asexual cycle in this, but that Malarian parasite Plasmodium cannot live in this. Okay, so these people who are having normal RBC, they cannot escape from malaria, but they can easily escape the malaria. Okay, so it is again the natural selection, and it is seen in Africa mostly. Okay, next is your Ladder-Birth replicator plate experiment. It is again the example of natural selection. Okay, so in this he take a pot-like structure in which he put a velvet sheet. Okay, sterile velvet is pressed on the growth colonies on the master plate. Okay, this is the master plate. Okay, in which he pressed this plate. Okay, now this plate contained bacterium. Okay, now he take two different petri dish, in which one petri dish it will contain histidine and antibiotic, and this plate does not contain histidine. And when he will see that thing under microscope, that colony which are present here, it is missing here. Okay, the colonies which is grow in this antibiotic, they develop resistance, they will naturally select it, but these species are not present here. Okay, because they cannot grow in this medium. Okay, they can grow in this medium, but they are not present, but they are grow some species which are mutant, which is naturally selected, they are easily grow inside the histidine medium. Histidine is an antibiotic, and antibiotic kill microorganism, bacteria. But after that after that experiment, he see that some species can easily grow inside the histidine solution, means they develop resistance. Okay, this thing, in the previous slide we studied that drug resistance. Okay, so that thing is developed in that bacterial species, and this show that the natural selection really works. Okay, so it was concluded that bacteria will survive their penicillin resistant because they have penicillin resistance mutant gene which enable them to survive in change environment. It is a change environment. Okay, it means mutation are predictive and natural selection fix them in population over generation to generation time. Okay, mutation is the cause of evolution. Again, this example says that. Okay, next is your artificial selection. Okay, this is the selection done by human. Okay, artificial selection. This is human done, not natural. Natural selection done by nature, artificial selection done by us. Okay, so artificial selection is similar to natural selection except that the role of nature is taken over by man. Okay, example, breeds of dog. Okay, initially grey wolf, common ancestor of dogs. Okay, it is hunt, we do hunt with humans. Okay, but the character we want, okay, the thing we want in them, we do that thing with the help of artificial selection. We select the dogs with the wolf which does not bite us, the wolf which does not attack us, and the wolf which is going to attack us, we kill them. In the previous time when the man are not, means that that australopithecus time, at that homo habilis times. Okay, at that time the species of dog, that is species of wolf which attacks us, we kill them, and the one who can be domesticated, we keep them. And by the doing natural selection will produce different species of dog. Now the different species which is present now in the dog, the different breeds of dog which is present now in the world, is because of artificial selection done by us. Second example of artificial selection is mustard plant. Okay, we artificially selected different characters, different parts of the mustard plant, and develop it into different foods. Just like cauliflower, it is an fertile flower. Okay, or inflorescence, for the sterile flower or inflorescence of mustard plant. Okay, second broccoli, it is again the flower, inflorescence of plant. Then cabbage, okay, it is a flower, it is the that leaf part of the mustard plant taken. Okay, kohlrabi, selection for enhancement of lateral meristem, the lateral meristem has been taken and it will develop into a new plant. Then kale, selection of enlargement of leaf, we enlarge the leaf of mustard plant and we develop kale. So all these, cauliflower, broccoli, cabbage, kohlrabi, kale, these all once time a mustard plant, and we evolve that thing naturally. We select the some characters and develop into a new organism. Okay, second example, the rock. Third example, the rock dove. This is the common ancestor of all these different species, different breeds of pigeons, and it is developed by our natural selection, not developed by our artificial selection, not by natural selection. Okay, next is reproductive isolation. Okay, reproductive isolation means we are isolating two same species with different areas. Okay, isolating for reproduction in different areas. So it is the prevention of interbreeding between the population of two different and closely related species. Okay, it maintains the character of the species but can lead to the origin of new species. For example, when tiger mates with lion, we get liger. Okay, and it is the result of reproductive isolation. Okay, this mechanism of reproductive isolation was explained by Stebbins in his book Process of Organic Evolution. Okay, so the two main types of reproductive isolators in this, we have pre-zygotic isolation, prevention of mating and formation of hybrid zygotes. This is your pre-zygotic isolation. Okay, it's have two types: ecological isolation, isolation due to the difference in the habitat of two species. For example, one may be living in fresh water and another living in the sea, due to which it cannot mate. Okay. Second, temporal isolation due to the difference in the breeding season or flowering time of two species. It cannot again do reproduction. One will reproduce in different season, one will reproduce in different seasons. So again, we are producing a reproductive isolation. Next one is your that behavior isolation due to the differentiation in sexual or coitus behavior of two different species. Okay. Then mechanical isolation due to the incompatible external genital. The external genital are not same. One have large external genita, other have smaller genital extreme, that external genitalia. Okay. So they cannot reproduce again. Okay. And other one is gametic isolation. The sperm and ova of different species cannot fuse due to a difference in the surface chemicals. The antigen which is present on the surface that recognize each other, that are different. Okay. So these all are pre-zygotic isolation, means before formation of zygote you isolate the organism. Okay. Next we have post-zygotic. Okay. Zygote is formed, then we isolate them. Okay. So a hybrid zygote is formed, but it may not develop into viable fertile adult. Means zygote is formed, but the zygote when develop into adult, convert into adult, then it will cannot do reproduction. It cannot produce its progeny. Then it is called post-zygotic isolation. In the previous pre-zygotic, we are not making a zygote, but in post-zygotic isolation, we make a zygote. That zygote develop into adult, but that adult cannot reproduce. Okay, then that type of isolation is called post-zygotic isolation. Okay, so it has three types: hybrid inviability. Hybrid zygote fail to develop in plant embryo arising from interspecific cross or not viables. Okay, next, hybrid sterility. Hybrid adults are sterile and do not produce gamete. Example: mules, hilly, liger, tigon, all these things. Okay, and hybrid breakdown, sometimes interspecific mating produce a hybrid which give rise to next hybrid, but back cross they have reduced vigor and fertility or both. Okay, two hybrid do reproduction, they produce a child, but that child lost its fertility. Okay, that thing is called hybrid breakdown. Okay, the hybrid is breakdown. The fertility of that hybrid is present, but after some generation, that thing is breakdown. That is called hybrid breakdown. Okay. Next thing is your speciation, means formation of new species. Okay, development of new species is called speciation. Okay, so formation of one or more new species from existing species is called speciation. Speciation are mainly of two types: divergent speciation and transformation speciation. Divergent speciation, when one or more new species are formed from ancestral species, means if there is species A, from that A species we forming A1, A2, then it is called divergent in speciation. From one point you are going to diverge. Okay, then it is called divergent speciation. Okay, divergent speciation is of two types: allopatric speciation and sympatric speciation. When a species is split into two or more geographical isolated populations, and these populations finally form a new species, it is called allopatric speciation, just like Darwin's finch. Again, the example is same. This is a geographical area. In the center of that, you have a species. Now, some species of this part migrate to this area, and it will become A1. Some will migrate to this area of this geographical area. This will convert into A2. Some migrate to this area, it will become A3. This become A4. Okay, due to these different areas have different adaptation, their geographical things, they will adapt to that geographical material, and they will develop into new species. Okay, then this is called your allopatric speciation, or in other ways we can also call this adaptive radiation also, because the example is Darwin finch. In adaptive radiation we studied that example is Darwin finch. So allopatric speciation equals to that your adaptive radiation. Okay, next is sympatric speciation. In this type of speciation, a subpopulation become reproductive isolated from its parental population. It is formation of species without geographical isolation, means species is there. Okay, sympatric देखो, species A is there, they are living in same area, but they are reproductive isolated to each other, and that reproduction isolation make them to new species. Okay, this is the parental species sometime some year ago. Okay, and now there is isolation. Some flood take place, earthquake take place. That's why this species cannot go here to reproduce. So they will develop new character in them and they will convert into new subspecies. Okay, that type of speciation is called sympatric. In this, they are migrating to different geographical areas, but in sympatric, geographical area is same, but due to reproductive isolation, they cannot reproduce. Okay, next is transformation speciation. When an ancestor species changes into the new species, means एक ही species अलग ही species में बदल गई due to sudden mutation. Here mutation not take place, here mutation take place. Divergent speciation it causes because of evolution. Transformation speciation causes mutation. Okay. Phyletic speciation, ancestor species changes into new species by gradual change in thousands of years. Example: Epiospas, Mesocyppus, Merychippus, Pileoippus, Equus. That is the evolutionary lineup of horse. Okay. And quantum speciation, in this process sudden major change appear in the ancestor species and ancestor species immediately change into the new species. No connective link are present in this type of speciation. It is caused by mutation. Okay, as I said, transformation speciation occurred due to the mutation and divergent speciation occurred due to the evolution. Okay. Next, mimicry. Okay, the term mimicry was given by Bates. It is a kind of adaptation. Okay, mimicry is the resemblance of one organism to another organism or natural object of the purpose of complement, protection, and for other advantage like attack. Okay, mimicry is a thing in which you will mimic like other. Okay, so the organism which do mimicry it is called mimic, and the organism doing its mimicry it is called model. Okay, so the organism which exhibit mimicry it is called mimic, and the organism or natural object which is mimicked is called model. Okay, so Batesian mimicry, there is two type of mimicry: Batesian mimicry and Mullerian mimicry. Okay, the organism resemble and distasteful or poisonous organism. In this Batesian mimicry, the organism resembles a distasteful or poisonous organism. Means the organism is looking like an organism which is distasteful or poisonous. For example, this is coral snake, very venomous. Okay, and this is milk snake, non-venomous. These both look like same. You cannot distinguish between which one is coral snake and which one is milk snake. So this will do Batesian mimicry. Okay. Next is Mullerian mimicry. When two or more inedible or unpalatable species resemble each other, then this type of mimicry is called Mullerian mimicry. Okay. In this, it is poisonous or distasteful. In this, it is inedible or unpalatable. Means you cannot eat that species. Then this type of mimicry is called Mullerian mimicry. Okay. Mullerian mimicry is done by two species for increasing warming effect to predators. Okay, it is for making that predators escape away from, making you hide, making them predators, making them to tell you that telling predators that I am very dangerous, away from, stay away from me. Okay, then that type of mimicry is called Mullerian mimicry. Okay, example is your queen butterfly and monarch butterfly. Okay, this is your queen butterfly and this is your monarch butterfly. Queen butterfly is not that much dangerous, but queen butterfly is dangerous and monarch butterfly is not that much dangerous. Okay, and queen butterfly is the model while your monarch butterfly is the mimic who do mimicry of queen butterfly to escape from birds that they not eat them. Okay, next one is their centipede moth resemble a wasp. It is a normal insect. Okay, it is not dangerous, but it look like it do mimicry of wasp. Okay, in which wasp is model and tenacid moth is an mimic. Okay, it look like dangerous, but it is not dangerous. Okay, and these are some caterpillars who look like snake, but they are not really a snake. They are caterpillars. Make them enemies, stay away from them. That I am a snake, stay away from. This is an example of Mullerian mimicry. This is an example of Mullerian mimicry. This is an example of Batesian mimicry. Okay. Next is your Hardy-Wimbuck principle, third part of this chapter. Okay, so Hardy-Wimbuck, these are two scientists. Okay, Hardy and Wimbuck. It says that allele frequency in a population are stable and constant from generation to generation in the absence of disturbing factor. According to them, every time the frequency of gene is equals to 1. Okay, if that one disturb means evolution or mutation take place. Okay, so gene pool, total gene and their allelic in a population remain constant. This is called genetic equilibrium, Hardy-Weinberg equilibrium. Okay, sum total of all allelic frequencies equals to 1. Okay, a population of 20 beetle, each beetle is in a gene pool. There is totally in the gene pool is that of the population. Okay, means all these are red and these four are recessive. These are dominant. Okay, so allelic frequency refers to how frequently a particular allele appear in the population. See in this 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, uh, 13, 14, 15, 16. 16 time you have red and 4 time have you yellow. So frequency of red one is frequently more. So that thing is governed by your Hardy and Weinberg. Okay, that again I have an example. Okay, in this consider in a diploid organism P and Q are the frequencies of allele A and small a respectively. Means QP is your dominant one and Q is the recessive one A. Okay, so frequency of AA equals to P square, frequency of small a equals to Q square, and frequency of heterozygous is equal to 2PQ. So hence when we put this thing and form a formula, we get P square plus 2PQ plus Q square equals to 1. And if this result not get 1, means there is some mutation or evolution take place in that particular population. Okay, so change of frequency of allele in a population disturb Hardy-Weinberg equilibrium, and the causes are these are genetic migration, gene flow, okay, genetic drift, evolution, mutation, genetic recombination, natural selection. It is mutation. Okay, some printing mistake is there. It is mutation. It is mutation. Okay. So genetic migration, genetic drift. Okay, genetic recombination, natural selection, these all are the factor who affects our Hardy-Weinberg equilibrium. Okay, so first we have gene migration. Okay, means migrating of one gene from another gene pool, and from another gene pool new gene will come into the new, that in the old population, then it is called gene migration. See this blue word, go inside that red colony and do reproduction there, and now that offspring can be content color of both combination, and this red will come inside the blue and it will reproduce here and will produce new genes. Okay, new color of genes. Okay, population A going in population B producing new frequencies, population B coming in population A producing the new frequencies, then again your Hardy-Weinberg principle has been get altered. Okay, second one is your genetic drift that we studied already, in which we have founder effect and bottleneck effect. Okay, next one is your mutation, which is the most important cause of Hardy-Weinberg equilibrium affection. Okay, it result in formation of new phenotypes or new, over few generations this lead to speciation. Okay, new species will form. Okay, next is genetic recombination. This is the small level of variation, new variations, new characters will develop. Okay, it is the reshuffling of gene combination during the crossing over resulting in genetic variations. Okay, the variations occur inside the genome. This, see, this is A B C. Now the crossing over will take place. Now will you see the red part of this chromosome will come in the blue part, and the blue part of this chromosome goes into the that red part, and we got recombinant chromosome. Okay, so natural selection, that is again a factor which affect your Hardy-Weinberg rule. Okay, so natural selection is a scientific theory that explains how organisms adapt to their environment involve over time. How it works? Organisms that are best suited to their environment are more likely to survive and reproduce. Means the organism which adapted to the particular environment, it will survive. If it does not adapt to that particular environment, it will not survive, it will get extincted. Okay, so they have three types of natural selection. First is stabilizing, second one is directional, and third one is disruptive selection. Okay, so it favors the average and normal phenotype and eliminate the extreme variants. Okay, means if there is three variants A, B and C. ओके, दिस वन इज नॉर्मल। ओके, वेट से समझते हैं अपन। ओके, थ्री टाइप्स ऑफ बेबीज आर देयर, 7.5 kg, okay, and this is 10 kg, and this is 8 kg, okay, 8.5 ले लो। okay, so the line is saying it favors the average or normal phenotype। okay, this is your normal, sorry, this is the average and this is the normal। okay, and this one is the extreme, so it will select these both and extend this one। okay, this is your stabilizing selection। okay, next one is your directional or progressive selection। okay, it favors one extreme value and eliminates other extreme value of an average value। okay, again same example, a, b, c, 7.5 kg, 8 kg, 10 kg। okay, okay, so it favors one extreme value and eliminate another extreme value and average value। so this is your average value, it get extinct, this is your extreme value, this is get extinct, or this will survive a species। okay, next one is your disruptive selection। okay, in this selection member of both extreme are selected simultaneously and average values get rejected, means the middle value a, B, and C. These both get selected because these both are extreme values, and this B value it will get extincted. That thing is your disruptive selection। okay, so this is the graph, okay, in which population after selection, and this is the population originally, see both are same thing। okay, graph is same, this is the that lighted line is of original population, and this the dotted line is of original population, and the continuous line is population after the selection, it is same। okay, in this selection against the extreme phenotype and both ends of the traits। okay, population after selection, okay, and original population में से जो extreme phenotype थे that thing will be not selected, and the normal one and some part of extreme one is being selected। okay, and last one is your selection from one extreme and against the mean। okay, so population after selection, these both are your selected one, and the middle one, this dotted one, is not selected because it is average, and both the extremes are selected, that type of evolution is called disruptive selection। okay, now next, the last part of this chapter is human evolution। okay, how can we evolve, from what ancestor we evolved, it tells you। okay, so normally humans is the member of order primata, okay, of class mammalia। okay, first real primate ancestor were tree shrews, this is the ancestor of all mammals। okay, ये छुचुंदर सबका ancestor था, इससे सारे मैमल्स बने अर्थ पे, जितने ये मैमल्स अभी अर्थ पे हैं उसका कॉमन एंसेस्टर ये छुचुंदर था। okay, it is the mother of all the mammals which is present now on earth। okay, the first real primate ancestor was tree shrew, originated in Palestine epoch। okay, from that thing all mammal develop on earth। okay, then these are the lines of the mammals। okay, these are prosimians, and this is your anthropoids, and in anthropoids these are hominoids। okay, hominoids means who walk on two legs। okay, gibbons, orangutan, gorilla, chimpanzee, humanoids। okay, these are called hominoids। okay, now these all are anthropoids, and in which gibbons, orangutan, gorilla, chimpanzee, and hominids means humans, these come under hominoid category। okay, next, evidence for common ancestry become between human and apes। okay, so you can see the skull। okay, this is human skull, and this is the skull of baby chimpanzee, both look like same। okay, and this is the skull of your adult chimpanzee, it is very different, large canines, elongated jaw। okay, so development of same skull in the human, adult human and baby chimpanzee says that we are having sometime having same type of body। okay, same type of skeleton design। okay, so first similarity is chromosomal similarity, bending pattern of chromosome number 3 and 6 of human and chimpanzee is 100% same। okay, the band which is present like this। This is a chromosome number 3 in If this is a chromosome number 3 in your chimpanzee and this is chromosome number 3 in human, okay, the band which is present here in chimpanzee, okay, the band which is present in this chromosome in human, it is same present in chimpanzee, okay. So that banding pattern in chromosome number 3 and 6 is same in both chimpanzee and your human, okay. Second, skull of baby chimpanzee is more like adult human skull than adult chimpanzee skull, okay. ये खोपड़ी इससे ज्यादा इससे मिलती है, okay. The खोपड़ी of your baby chimpanzee is similar to human skull than your adult chimpanzee, okay. Then again this is a result that shows that we evolved from a common ancestor, chimpanzee and us, okay. Then composition of hemoglobin is same in both, only one amino acid is different in human and gorilla, okay. Only one amino acid difference is there between human and gorilla hemoglobin, okay. Next, blood group of AB series is present in both plasma protein of all are same, okay. In gorilla, in chimpanzee, in human, that blood grouping is same, AB blood grouping, okay. Then menstrual cycle is present in both females, okay. Tail is absent in both, having grasping hands, both chimpanzee and male, human, both having grasping hands, okay. They can grasp things easily, both chimpanzee and human, okay. So this is the similarities between human and apes, okay. Next, origin and evolution of meal. Okay. So apes fossil about 15 million years ago, primates called Dryopithecus and Ramapithecus were exist. They were hairy and walked like gorilla and chimpanzee. Dryopithecus is considered as common ancestor of human and ape, and Ramapithecus fossil discovered from Shivalik Hills in India. Ramapithecus were more mane-like while Dryopithecus were more ape-like. This is your Dryopithecus and this is your Ramapithecus. See, this is the timeline of evolution. Dryopithecus, Ramapithecus. Okay. इससे आगे गए अपन, then we have Australopithecus. Okay. From then ahead we go, we have Homo habilis. Okay. After that we have Homo erectus. Okay. After that we have Neanderthal, and after that we have Homo sapien, and after that we have Homo sapien sapiens B. Okay, Homo sapien sapiens. Okay, Dryopithecus they will convert into your apes. दोनों का स्टार्टिंग पॉइंट एक था, but they will differentiate into different organism. They will convert into apes, and Ramapithecus convert into Australopithecus. Australopithecus evolved in Homo habilis, Homo habilis in Homo erectus, Homo erectus into the Neanderthals, and Neanderthals into Homo sapiens, Homo sapiens into Homo sapiens sapiens. Okay, this is the evolutionary history of human. Now these we are going to study in detail. Okay, so first is your Australopithecus. Okay, few fossils of mane-like bones have been discovered in Ethiopia and Tanzania. These revealed hominid future, leading to the belief that about 3 to 4 million years ago, man-like primate walks in Eastern Africa. Okay, they were probably not taller than 4 feet and but walked upright. Okay, ऐसे होके चलते थे, पर वो 4 मीटर लंबे नहीं थे, उससे ज़्यादा लंबे नहीं थे हाइट में. Okay. Next, Professor Ramdad discovered a fossil of skull of 5 to 6-year-old baby from the Pliocene rock of Tuang region, South Africa, and named that Tuang baby. Later, he renamed it as Australopithecus africanus. Okay, African ape-one. This A is equals to Australopithecus. Okay. This A is Australopithecus africanus. Okay. 2 million years ago, Australopithecus probably lived in Eastern African grassland who do hunting of deers for their meat eating. Okay, but still they not eat that thing. They hunt that thing to show other carnivores which is living nearby them that we are the boss of that area. Okay. Evidence show they hunted with stone weapons but essentially eat fruit. Okay, that I said now. They hunt, but they do not eat what they hunt. Okay, they eat fruit. They do hunt because to show that we are the boss of that particular area. Okay. They had complete erect posture and bipedal locomotion. मतलब तुम्हारे रामापिथेकस ऐसे चलते थे बंदरों की तरह. Now they start to doing move like this. Okay. It is also considered as connecting link between apes and men. Okay, connecting link between apes and man. It is your Australopithecus. Okay. Next is your prehistoric man, Homo habilis. Okay, this creature was called the first human-like being, the hominid. Okay, first organism, first ancestor of human which look like human. Okay, which is Homo habilis. Okay, the brain capacity were between 650 to 800 cc. Okay, they probably did not eat meat. Till now we do not eat meat, we are on fruit. Okay, first man who made tool of stone for hunting animal, hence called it first tool maker or handyman. Still they hunt animals but they do not eat them. They show that we are the boss, do not come and attack on us. Okay, this is your Homo habilis. Now next one is your Homo erectus. We have three subspecies of Homo erectus. One is Java man, Peking man and Hindbug man. Okay, fossil discovered in Java in 1895. They exist about 1 million year ago. They had large brain with cranial capacity around 900 cc. As we go ahead, cranial capacity is increasing. Okay, Peking man fossil discovered by W.C. Pei from China. Okay, and Hindbug man fossil discovered from Hindbug, Germany. They are extincted. Okay, next is Neanderthal man, Homo sapien neanderthalensis. These were the first one who eat meat. On the one first time who start to talk, because their broker area which is present in their skull, they start develop. Okay, so they live near the east and central Asian between the one lag of 40 year ago back. They had brain size of 1400 cc, same as modern men. They use hidden skin of animals to protect their body. Now they start to wearing clothes also. Okay, they buried their deads and probably believe in immortality of soul, means they start to doing worship also. They start to say that soul go to somewhere else. Okay, development of speech center and language center started, means they start to talking with each other. Okay, then we have Cro-Magnon man, Homo sapien fossilis. Okay, that is the connecting link between Neanderthal man and Homo sapien. This is very dangerous. Okay, very dangerous. If these things are extended, if this thing is present now na, we are not present. Okay, these are man eater cannibals. Okay, these Cro-Magnon men eat Neanderthal men. Okay, and their brain capacity is the largest one in all humans, 1650. R is 1600. Their cc of brain is 1650 maximum. Okay, the painted beautiful paintings on the cave wall, prehistoric cave are developed about 18,000 years ago. One of such cave painting by prehistoric humans can be seen in Bimthel rock shelter in Raisin district of Madhya Pradesh. Okay, India में घूमते थे ये लोग. They are man eaters. They eat Neanderthal men. Their height is very long. Okay, ये थे ये वाले. Okay, next is your modern man, Homo sapiens, Homo sapiens us. Okay, during ice age between 75,000 and 10 years ago, modern man, Homo sapiens arose. Okay, they arose in Africa and move across the continent, develop into distinct races: cascoid, negroids, mongoloids and asteroids. Okay, these all are the races of human which is present now on earth. This is the main of today, having brain capacity of 300 to 1300 to 1600 cc, average 1450 cc. Okay, agriculture was also studied by this man. Agriculture came around 10,000 years back and human settlement started. That is the evolutionary history of human. Okay, so this is your evolutionary history of human, in which we have different ancestors. Okay, this is us, Homo sapiens sapiens. This is Homo sapien. This is Neanderthal man. This is Homo erectus. This is Homo habilis. This is Australopithecus, and this is your Ramapithecus. Okay, Ramapithecus, Australopithecus, Homo habilis, Homo erectus, Neanderthal man, Homo sapien, Homo sapiens sapien, modern man. Okay, so this is the sequence of human evolution. Okay, this is the hominoid family. Okay, different skulls of different members of hominoid family. Okay, so this is the end of the chapter evolution. Okay, we completed fifth chapter. Break or Now, it's time for molecular basis of inheritance, the most interesting chapter of biology, in which we are talking about the molecular level of genetics. Okay, so we are going to discuss about the nitrogenous basis, the structure of your nucleic acid, different structures present inside your nucleic acids. Okay, so nucleic acids are the polymer of nucleotides. Okay. Example of DNA. Examples are DNA and RNA. Nucleotide nitrogenous bases plus pentose sugar plus phosphate equals to nucleotide. Nitrogenous bases plus pentose sugars equals to nucleoside. When you delete phosphate from nucleotide, it will become nucleoside, and when you add phosphate in nucleoside, it will become nucleotide. Okay, so your DNA, RNA is made up of three things: nitrogenous base, phosphate, and pentose sugar. So first we have nitrogenous base. Skeleton of nitrogenous base contain heterocyclic rings. On the basis of structure of nitrogenous base, we have two types: pyrimidine and purine. Pyrimidine consists of one pyrimidine ring, which is two nitrogens and four carbons. Skeleton of ring composed of two nitrogen and four carbon atoms. Example: cytosine, thymine, and uracil. Thymine is also called as 5-methylated uracil. Means on the fifth position of the ring we have methyl group. Then it will become thymine. Uracil, on the fifth position of that uracil we have methyl group, then it will become thymine. Okay. Purines, it is two, that is your guanine and adenine. Okay. Tricks to remember this: Ag purine. Okay. Ag purine. Purine, the word is small, it have two. Okay. Pyrimidine, long word, it have three. Okay. Cytosine, thymine, and uracil. Okay. Again I am saying this: pyrimidine, long word. Okay, 1 2 3 4 5 6 7 8 9 10 11 11 word, 11 alphabet word it is. It have three: cytosine, thymine, and uracil. Purine, it is a small word of your seven alphabets: 1 2 3 4 5 6 7. Okay, and it have two: Ag, adenine, guanine. Okay, now the structures. And the second structures: purine, this is adenine. Okay, heterocyclic with two cycles. Okay, and then you have guanine, again heterocycle with two different cycles. Then it is guanine and adenine, two different cycles plus heterocyclic in nature. Okay, next we have cytosine, single cycle. Okay, heterocyclic means the cycle, it is made up of two different compounds, two different we can say two different elements. Okay, in this you will have carbon also and nitrogen also, then it is become heterocyclic. If it is this chain, this cycle, if of carbon, then it is called not heterocyclic, homocyclic. But it having carbon and nitrogen both, then it become heterocyclic. But it has single cycle, then it is monocyclic, heterocyclic. Your purines are heterocyclic plus dicyclic. Two cycles, one, cycle two. This is your pyrimidine, okay, single cycle. Then thymine, single cycle. Uracil, single cycle. Now difference between thymine and uracil. In thymine you see there is methyl group. In uracil you do not see any methyl group. On the fifth position of that ring you do not see any methyl group. Then it is uracil. And when you see on the fifth carbon of that cycle you will see a methyl group, then it is thymine. Okay, so this is nitrogenous basis present in your nucleic acids. Okay, next thing is your ribose sugar. Okay, ribose sugar and deoxyribose sugar, these both are pentose sugar. 5 C5, carbon is of 5. Okay, 1 2 3 4 5, 5 carbons are there, then it is called pentose sugar, C5. Hexose sugar, your glucose, C6H12O6, 6 carbons. Okay, this is pentose sugar, 5 carbons. Okay, third part of your nucleic acid is phosphate group, which is acidic and it provides negative charge to your DNA or RNA. Okay, so difference between ribose sugar and deoxyribose sugar: ribose sugar, oxygen is present, oxygen is absent. Okay, more reactive, less stable. Due to the presence of oxygen in your ribose sugar, it will become more reactive and less stable. While the absence of oxygen in your deoxyribose, then it will become less reactive and more stable. That is the reason why your DNA is more stable and RNA is less stable. Okay. Now next, nucleoside and nucleotide. As I said in previous, nucleoside means pentose sugar plus nitrogenous base, nucleotide pentose sugar plus nitrogenous base plus phosphate group. Okay, now how does phosphate group and nitrogenous base attach to each other? Let's see. Okay, that nitrogenous bases which is present inside your nucleic acids, it will attach with each other with the help of that bond. I will tell you. Nitrogenous base, phosphate and pentose sugar, nitrogenous base and your phosphate. Okay, so when your nitrogenous base is connected by pentose sugar, so this bond is called an glycosidic bond. Okay, the bond which is present between nitrogenous base and pentose sugar in DNA and RNA is an glycosidic bond, while the bond which is present between pentose sugar and phosphate group, it is called phosphodiester bond. Okay, this bond which is present between pentose sugar and phosphate is your phosphodiester bond. Okay, clear? The bond between pentose sugar and nitrogenous base is an glycosidic bond, and the bond present between pentose sugar and phosphate group is phosphodiester bond. Okay, again in this diagram, again you can see this is your nitrogenous base, this is your pentose sugar, and the bond which is present between nitrogenous base and pentose sugar is called an glycosidic bond, and the bond which is present between phosphate group and your pentose sugar is called phosphodiester bond. Okay, so now we are going to discuss about DNA. Okay, so DNA is an acidic substance because of presence of phosphate. Okay, in nucleus was first identified by Frederick Manchester in 1969. Okay, DNA term was given by Zacchaeus. Okay, DNA is long polymer of deoxyribonucleotide. DNA is negatively charged because of phosphate. Okay, in DNA, pentose sugar is deoxyribose, and four type of nitrogenous bases are present: A, T, G, C: adenine, thymine, guanosine, cytosine. Okay, Wilkins and Franklin studied DNA molecule with the help of X-ray deflection data or X-ray crystallograph. Okay, means they put X-ray machine, and in the middle of that they put X-ray the DNA, and here we have in the middle they put here they have X-ray machine, here they have DNA. Okay, and here they put X-ray that X-ray film. When they bombard X-rays on this DNA, that printation of that DNA molecule occur on the X-ray film. That thing shows the presence of DNA is in spiral in the nature. Okay, helical in the nature. With the help of this study, Watson and Crick proposed double helix method. Okay, without this study of Wilkin and Franklin, we cannot get that DNA is double helical. Due to that X-ray diffraction method, they studied that DNA structure is double helical. Okay. Then Watson, Crick, and Wilkins are awarded by Nobel Prize in 1962, discovering that DNA is double helix in the model. Okay. Now next, according to this model, DNA is composed of two polypeptide polynucleotide chains. Both polynucleotide chains are complementary to each other, anti-parallel to each other. Means if a strand is running 5 to 3 in this direction, then in this direction it is 3 to 5. Okay, opposite to each other. Okay, it is always opposite to each other. If this strand is 5 to 3, then this strand is 3 to 5. Okay, next, in one strand of DNA, polarity is 5 to 3 that I said, and the other strand having 3 to 5, opposite to each other. Both strands of DNA held together with the help of hydrogen bond. Okay, these strands are held together by the help of hydrogen bonds. Okay, this is hydrogen bond. Okay, next, adenine bind with thymine, with two hydrogen bond. If this is adenine and this is thymine in DNA, then it is bonded with each other with the help of two hydrogen bond. And when guanine is bind with cytosine, then this bond is your triple bond. Okay. Next, in DNA molecule, one purine always pair with one pyrimidine. Always remember this, that purine is bond with pyrimidine and never pyrimidine bonds with pyrimidine or purine bonds with purine. Pyrimidine always bonds with purine. This thing is should be remember always, that purine always bonds with pyrimidine, never purine bonds with purine or pyrimidine bonds with purine. Okay. In DNA molecule, one pyrimidine always pair with pyrimidine. This generates appropriate uniform distance between the two strands of DNA helix, and that is the reason why we get double helix structure. Okay. In DNA plane, one base pair stack over the another double helix. This is the addition of hydrogen bond conference stability of helical structure of DNA. Means just like when we put, see this bond will always present upon of this. Okay. Means if this has a one bond, this is one bond, then other one will present like this. Okay, stack over the other, then other will like this, other will like this, other will like this. Okay, then this is called DNA plane of one base pair stack over the other one. Okay. Next, so this is your Chargaff rule. Okay, Chargaff rule tells that A plus G upon T plus C equals to 1. Okay, means the number of A equals to the number of T, G equals to C. These are not double bonds. Okay, people distinguish this equals to double bonds. Okay, it is not double bonds. It's saying that A equals to T or G equals to C. If in a DNA molecule we have 100 of A equals to T and G equals to C, in which 30 will be A, for example. Okay, 30 will be T. Okay, so it is equals to 60. Now remaining 40, 20 will be G and 20 will be C. 60 plus 40 equals to 100. Okay, so this is your Chargaff rule. Okay, Chargaff rule says that A equals to T, G equals to C. Okay. Next, melting point of DNA depends on GNC content. Okay. More GNC content means higher melting point. The DNA will melt on high point. Means if a DNA having more GNC in that DNA composition, then it can melt above 100 degrees Celsius. And if have more ANT, it will melt less than 100 degrees Celsius. Okay. Tm of prokaryotic DNA is more than the temperature, that melting point of prokaryotic DNA is more than the eukaryotic cell, because prokaryotic DNA have more GNC. Okay, the melting point of your prokaryotic DNA is more, while the melting point of eukaryotic DNA is less, because prokaryotic DNA have more GNC. Okay, while eukaryotic DNA having more ANT, and presence of more ANT makes your DNA more stable, and presence of more GNC makes your DNA less stable. Okay, the reason bonds. Okay, GNC having triple bond. Okay, and ANT having double bond. Okay, that bond, number of bond makes stability. Okay, so DNA absorbs UV radiation of 2600 Armstrong wavelength. Okay, then denatural and renaturation of DNA. If a normal DNA molecule is placed at high temperature 80 to 90 degrees Celsius, then both strands of DNA will separate to form each other due to the breakdown of hydrogen bond. It is called denaturation of DNA. Okay, when denatured DNA molecule is placed at normal temperature, then both strands of DNA get attached and recoiled to each other. This is called renaturation of DNA. Means if you remove high temperature, you put DNA from normal temperature, then your DNA again reform that hydrogen bomb and it will become normal. Okay, high temperature destroy hydrogen bond, DNA become denatured. Put that denatured DNA again normal temperature, it will regain its, it will reform its hydrogen bond and it will become normal. Okay. Next, configuration of DNA molecule. This is the most important part. Okay. See, this is helix of a DNA. Okay. Now from this point to this point, Okay, this is 1 ton, 1 pitch. Okay, now the length of this point to this point is 34 Armstrong and 3.4 nanometer. Okay, 1 ton and 1 pitch is the distance of 1 ton. Okay, means if you have a long DNA, the turning point from this point to this point is called 1 ton or 1 pitch, and the length of this 1 ton is 34 Armstrong and 3.4 nanometer. Okay, now in this 1 ton you have 10 nitrogenous base pair. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, 10 nitrogenous base pairs are present. 10 nitrogenous base pair. Okay, and 20 nitrogenous base. Okay, again I am saying 10 nitrogenous base pairs and 20 nitrogenous bases. This is one pair of shoes. Okay, in one pair of shoes you have two shoes. Okay, same thing is here. 10 nitrogenous base pair contain 20 nitrogenous bases. Okay, now the distance between from this nitrogenous base pair to this nitrogenous base pair. Okay, the distance between from this nitrogenous base pair to this nitrogenous base pair is 3.4 Armstrong or 0.34 nanometer. Okay, second value you get, that is the distance between one nitrogenous base pair to another nitrogenous base pair in 1 ton. It is 3.4 Armstrong or 0.34 nanometer. Okay, second value. Now third value is the distance diameter. Okay, from this point to this point. Okay, diameter. Okay, it is 20 Armstrong or 2.0 nanometer. Okay, from this point to this point the diameter of your DNA, okay, is 20 Armstrong and 2.0 nanometer. So three value we get. Okay, the dimension of DNA and dimension of DNA we have three values. First, 1 ton or 1 pitch equals to 334 Armstrong or 3.4 nanometer. Then the distance between two nitrogenous base pairs is 3.4 Armstrong or 0.34 nanometer. And the distance between one strand, ओके, इस स्ट्रैंड से और स्ट्रैंड के बीच का डिस्टेंस डायमीटर डीएनए का वो होता है 20 आर्मस्ट्रॉन्ग या फिर 2.0 नैनोमीटर. ओके, एंड इन 1 ton you have 10 nitrogenous base pair and 20 nitrogenous bases. Okay, so this is the configuration of your DNA. That thing is written here. See, this is one nitrogenous base pair, this is one nitrogenous base pair, and the distance is 0.3 nanometer or 3.4 Armstrong. And this is the 1 ton from this point to this point. See here also. From this point to this point, from this point to this point, this is 1 ton. Okay, so this is your 3.4 nanometer or 34 Armstrong. Okay, next, this is the diameter. Okay, from this point to this point means the hydrogen bond. Okay, this point to this point. Okay, this is 2 nanometer or 20 Armstrong. Okay, then we have different organism base pair composition. Then we have your lambda phage 174 phage bacteriophage, that is contain 5386 nitrogenous base pairs. Okay, nucleotides. Next we have bacteriophage lambda. Okay, which having 40 had 1500 base pairs. Okay, then tell me how many bases are there. Okay, 4850 base pairs are there. Then how many bases are there? You multiply 48502 into 2. Okay, then you get the number of bases. Okay, next is E. coli has 4.6 into 10 की power 6 base pairs. Okay, and human haploid cell having 3.3 into 10 की power 9 base pair, then diploid cell having 6.6 into 10 की power 9 base pair. Okay, this 3.3 into 10 की power 9 is base pair of haploid cell. Diploid cell having 6.6 into 10 की power 9. Okay. Next, packaging of DNA. Okay, how 2.2 meter long DNA is packed into 10 की power minus 6 size of nucleus. That thing is done with the help of packaging, ultra packaging. Okay. So in prokaryotes, the DNA is not scattered throughout the cells. DNA is negatively charged, so it is held with some positively charged protein to form nucleoid. Okay, which we call nucleosome. Okay. Packaging of DNA helix in eukaryotes. Okay, in eukaryotes there is a set of positively charged basic proteins called histones. Okay, these histones are present, accumulate the DNA by around them. Okay, histones are rich in positively charged basic amino acid residues like lysine and arginine. Due to that, they get positive charge. Okay, DNA negative charge, histone protein positive charge. Okay, acidic nature DNA का, basic nature histone protein का. Okay, negatively charged DNA is wrapped around the histone octane to give a nucleosome like structure. Okay, so now we are to see the structure of your nucleosome. Okay. This is your octamer of histone protein. Okay, 1, 2, 3, 4, 5, 6, 7, 8. Around this, your DNA round like this. Okay, and here we have one more histone protein which we call H1 histone. And in this octamer is made up of H2A, H2A, H2B, H2B, H4, H4, H3, H3. Okay, so this area which is surrounded by these stones having totally in nucleosome, one nucleosome we have 200 nitro base pairs. Okay, 200 base pairs are present. Out of the 200, 140 is present here, and remaining 60 base pair is present in this region, and this DNA is called linker DNA. Okay, you can see here. See, this part of DNA which is present wrapped around your histone protein having 140 nitrogenous base pairs, while this part linker DNA, it contains 60 base pairs. Okay. Next thing, these are the dimensions. Okay, we have two types of DNA inside: right-handed DNA, which is called B DNA, and left-handed DNA means Z DNA. Left-handed, right-handed means the twistness. Okay, when DNA twisted to the right hand, then it is called B DNA, and when DNA twisted to the left hand, then it is called Z DNA. Okay. Next, these are the calculation. These are the numerical values. BDNA of B form of DNA is right-handed. Z is left-handed. BDNA have 20 Armstrong diameter. ZDNA having 18 Armstrong diameter. Rise per ton of helix 36 Armstrong, 44 Armstrong. Okay. Base pair per helix 10 base pairs. It having 12 base pairs. Helix rise per base pair 3.6 Armstrong, it having 3.7 Armstrong. Okay, base tilt normal to the helix base 6 degree, 7 degree. Means when your DNA turn, so that degree of turn which is present in the DNA. In B DNA it is 6 degree and in Z DNA is 7 degree. Okay, glycosic bond configuration it is anti, means that opposite to each other. Okay, and anti for pyrimidine and zinc for purine, means the degree of turn in pyrimidine is anti, while in purine it is same. It is same to each other. Okay, this thing will not come in NEET. Only you have to remember this, and the thing is diameter and helical sense. Okay. Next is nucleosome continuous, constitute the repeating unit of chromatin. Chromatin is thread-like strand bodies. Nucleosome in chromatin is bead on stick-like structure. Okay, that structure this you like that you're looking here. It is bead on string-like structure. Okay. Next thing is chromatin is paired, then chromatin become chromatin fiber. Chromatin fiber coiled and condensed in the metaphase stage, and at last we get chromosome. That is a super coiled form of DNA. Okay, 2.2 meter long DNA is filled inside 10 to power minus 6 nucleus by the help of supercoiling. Okay, and that structure you will see potentially in metaphase stage. Okay. Next, packaging level on higher level. Okay, the base of the beads on string structure in chromatin is packed to form chromatin fiber that are further coiled to condense in metaphase stage and cell division forms chromosome. That is your chromosome formation, how your chromosome is formed. When chromatin more condensed, convert into more condensed form, then it will convert into chromosome. Okay, the packaging of higher level required addition of set proteins that is collectively referred as non-histone chromosome. This is the important point. Histone protein is present in nucleosome. Non-histone chromosomal protein is present in chromatin coiling. Okay, when the chromatin get coiled, it contains NHC, non-histone chromosomal protein. Okay, in a typical nucleus, some regions of chromatin are loosely packed, which is called euchromatin. Okay, euchromatin is closely packed, and heterochromatin is loosely packed. Okay, so chromatin that is more densely packed stain is called heterochromatin. Okay, and the loosely one is called euchromatin. Means if this is a structure, okay, and we are going like this. Okay, so this structure is your euchromatin. Okay, and this structure is your hetero. Okay, euchromatin loosely packed, heterochromatin compactly packed. Okay, the chromatin that is more densely packed and stand dark is called heterochromatin. Euchromatin is also said to be transcriptly active, while your heterochromatin is transcriptly non-active. Okay, next point. So the search of genetic material. Now we are going to study about different discoveries, different experiment related to DNA. Okay, so first is evidence from bacterial transformation. Okay, the bacterial transformation experiment. Simply, we have three types of bacteria. Okay, one is your R strain, second is S strain, third one is your S killed, and fourth one is S killed with R leaving. And when this strain is injected by Griffith in mice, mice survive. When this strain injected in mice by Griffith, then mice die. When this injected in mice, mice survive. And when we inject S killed R living inside the mice, your mice die. Okay. In this experiment, we use your that pneumococcus bacteria. Okay, streptococcus pneumonia bacteria, pneumococcus. Okay, this bacteria causes pneumonia in mice. Okay, so first he take R stain, rough stain. Okay, that stain does not having the virulency. Virulency is the power of a microorganism by which it will create disease inside the organism. Okay, so he used first R stain, that is rough stain. It does not have virulency. When Griffith inject that thing inside the mice, mice survive. Okay, second phase of the experiment, he take S stain, shiny stain, which is virulent. Okay, when he inject that virulent bacterium inside the mice, mice die due to pneumonia. Okay, third phase of the experiment, he take S bacteria with heat killed, means he killed the bacteria. Okay, and when he inject that bacteria inside the mice, mice survive. Okay, last phase of the experiment in which he take S killed and R living, and he sees that and different experiment and different result that mice died. Okay, he noted that something is there present inside the bacterium which transform R strain into S strain. Okay, R strain is not doing any killing, but when he put S strain in, he had heat killed S strain inside R strain mixture, he sees that mice died due to pneumonia. Okay, so he tells that there is something present inside the S strain bacterium which converting normal R strain into S strain. He do not know that thing is DNA. Okay, so next, then this is the experiment. Okay, In this, mouse died because it inject S strand. In this, mouse survived because it inject R. In this again, mouse survived because he injected heat killed S. And in the last, he injected heat killed S with R living. Okay. So next, now these three scientists come: Oswald, Avery, Colin McCall, and McCarthy. They will do biochemical research on that material and propose that biochemical that substance which doing this transformation is your DNA. Okay, let's see how they perform that experiment. So they take again mice. Okay, inject S killed plus R living with protease enzyme. Mice died. Okay, next, second phase, they take mice, inject again R killed, S killed, and R living plus RNase enzyme. Mice again killed. Okay. In the third phase, they again take mice. Okay. And S killed and R living with DNase enzyme. And mice survive. Okay, so the material which changing your S killed that R bacterium into S bacterium, that thing is DNA. Okay, by this experiment they proved that that material which changing R bacterium into S bacterium, that thing is DNA. And they perform by experimentally they perform that experiment which is done by Griffith more accurately. Okay, this is the experiment. Okay, next is your experiment done by Hershey and Chase, bacteriophage experiment. Okay, they take bacteriophage. One bacteriophage is labeled with S35. Okay, and one having P32. Both are radioactive isotopes of your sulfur and phosphorus. Okay, so this experiment show that what is where DNA is present, where DNA will goes. Okay, so when that bacteriophage outer wall contains sulfur, means that is protein. Okay, sulfur always present in protein while phosphorus is always present in DNA. Okay, so when this bacteriophage inject inside bacterium, its plasma is its protein coat capsid remain outside. Okay, so this step is done into three processes. First is infection, second one is mixing. Okay, and for third one is your centrifugation. Okay, so after that centrifugation they will see that one test tube in which they have sulfur labeled bacteria. That sulfur labeled bacteria, the sulfur labeled bacteriophage, it will on the surface. Okay, the radioactivity is seen on the surface. Okay, which is present in capsid. Okay, while in second test tube they will see that radioactivity is present at the bottom of the test tube, which is present inside the bacterium. So the DNA which is injected inside that bacteria content radioactive phosphorus is coming from bacteriophage. Okay, so this experiment says that the genetic material is your DNA, which is present inside the bacterium, which is incorporated by your bacteriophage. While the capsid, it is made up of protein, it contains sulfur, its molecular weight is less, so it is present on the top of the test tube. Okay, so Alfred Hershey and Master Chase and worked with T2 bacteriophage virus that infect E. coli bacteria to prove that DNA is a genetic material. Okay, it was already known that bacteriophage attacks to the bacteria to deliver its genetic material into the bacterial cell. The bacteria treat the viral genetic material as their own and thus produce more viral particles. Okay, Hershey and Chase worked to discover whether it was protein or DNA from the virus that behave as a genetic material. Okay, they have to find that the material which is working as a genetic material, it is DNA or protein. Then after, when they see that, they know that DNA contain phosphorus and protein contains sulfur. Okay, so on the top of the test tube, that is your supertectant, okay, which is protein, and that palate part of the test tube, that bottom part of the test tube, contain radio level phosphorus DNA inside the bacteria, that tells that the DNA, the genetic material which is present inside the bacteriophage, is DNA. Okay, now central dogma of molecular biology. Okay, central dogma, it is proposed by Francis Crick. It states that genetic material information flows from DNA to RNA and from RNA to protein. Okay, means the process of conversion of DNA into RNA is called transcription, and from RNA to protein is called translation, and from DNA to DNA it is called replication, and from RNA to DNA it is called reverse transcription. Okay, again, see here, DNA to RNA, that is transcription, RNA to protein, that is translation, and DNA to DNA, it is replication, and from RNA to DNA it is reverse transcription, that is central dogma. Reverse transcription, it is a special procedure which is present in retroviridry family only. Retrovirus, retrovirus is the only virus which can perform reverse transcription process, because they have enzyme reverse transcriptase. Example of retrovirus: AIDS, HIV virus. Okay, next, procedure of DNA replication. It is the experimental proof that DNA having self-replicating power, okay, or semi-conservative nature. It is done by your McCall and Stahl and McCarty. Okay, sorry, McCall and Stahl. These both perform that experiment by which you can say that DNA is semi-conservative in nature. Its half part has been conserved inside the body. Okay, okay, so replication is the copying of DNA from parent DNA. Watson and Crick proposed semi-conservative model of replication. Watson and Crick proposed semi-conservative theory, and the experimental proof is given by Melson and Stahl. Okay, again, semi-conservative theory of DNA replication, it is given by your Watson and Crick, and the experimental proof of semi-conservative nature of DNA, it is done, it is given by Mansell and Stahl. Okay, that is your N15 experiment, this experiment. Okay, in the generation 0 you will find that DNA contained a same type of that nitrogen, okay, which is your N15, N15 and N15. Okay, now on second generation 1 you changes the medium to N14. Now it will become hybrid DNA which contain N15 and N15 and 14 both. Okay, this light blue is your N15 and this dark blue is N14. Now again, when this DNA do replication, you will find that out of four bacterium in generation 2, two having N14 and 14 and two are hybrid. When generation 3, okay, after 20 minutes, and this experiment is running after 20 minutes, okay, this is starting point 20 minutes. After 20 minutes, you will see generation 2. Then after 40 minutes, you will see generation 3. And after 20 minutes, you will, after that 80 minutes, you will see generation 4. Okay. So in generation find 1, you have two hybrid DNA, in which one is N15 and one is N14. Medium is N14. Okay. Generation 2 tells you that this both are your N14, 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 and these both are hybrid. Okay, in generation C3 you will see that 4 will convert into 8, and in which 6 are N14 and 14 and remaining 2 are N15 and 14 hybrid. Then in generation 4 you have 16, okay, in which out of 80 16, okay, in which 16 you are 14 are having hybrid DNA and 2 are that sorry 16 DNA are there in generation 4. In 1958, Taylor and colleagues do an experiment on eukaryotic cell of your Vinkia faba, okay, your that pea plant, in which he do that thing using radioactive thymidine. In prokaryotic cell, we take that nitrogen isotope. Here we use radioactive thymidine, okay, and again same thing will happen, that this is the normal which contain that radioactive thymidine. Now when we replicate this one, and this one is hybrid, because the medium is your of normal, that normal one, okay. Again it will replicate, same result will come when which is present here, okay. Two hybrid and two are normal, same thing is here, okay. Two normal, two hybrid, two normal, two hybrid, okay. Same thing is there. Now the material is changed only, but result are same, okay. That hybrid remains there, okay. The parental strand which is coming in that hybrid, it is remains there, while that normal one increase in the number, okay. That is the semi conservative technique, semi conservative nature of DNA, okay. Next thing is DNA replication, okay. How your DNA formation take place from DNA, okay. So DNA replication start at the point called ori, okay, origin of replication. A unit of replication with one origin is called replicon, okay. Just like the unit of your kidney is nephron, okay. Unit of liver, hepatocyte cell. Unit of lungs is alveoli. Just like the unit of replication is called replicon, okay. During replication, the two strands of unwind and separate break by hydrogen bond in the presence of enzyme helicase, okay. Now from starting that there is some material, some tools which is required in replication. Now we are going to see this. First is your helicase enzyme, okay. Helicase enzyme is responsible for opening of hydrogen bonds, breaking of hydrogen bonds. Next enzyme is your topoisomerase in eukaryotes and DNA gyrase in prokaryotes. Okay. Third enzyme is your DNA polymerase, most important enzyme of this reaction. Okay. Next is your SIB. SIB. Okay. These all are different enzymes which is take part in the process of replication. Okay. If these enzymes are not there, then your replication process is not going to be proper. Okay. Helicase, if like this is a chain, this is a chain of DNA, okay. If this is a chain of DNA, okay, and this is your helicase enzyme, this helicase enzyme break the hydrogen bond and open the chain, okay. And behind this topoisomerase and SIB box, topoisomerase charge or take charge that this DNA strand does not rebond, okay. That hydrogen bond which is present between them, they do not again rebound. That is a function of topoisomerase, okay. They maintain the stretchiness of your DNA strand, okay. Stability of DNA strand, okay. DNA polymerase, it is the thing which start the process of DNA replication with the help of primase, okay. Primer, that RNA primer which should be added, okay. So these are your enzymes: helicase, cofactor of helicase, magnesium, SSB, single-stranded DNA binding protein, and topoisomerase in prokaryotes and DNA gyrase in eukaryotes. Okay, sorry, in prokaryote it is DNA gyrase, and in eukaryotes it is topoisomerase. Okay, and the direction of DNA replication is 5 to 3 direction. Okay, from 5 end to the 3 end that process runs. Okay, deoxyribonucleotide triphosphate, these are the source of energy for replication. Okay, so see how this work. This is the strand of DNA and it is opened by your helicase enzyme. Okay, and this is your fifth end to third end, and this is your fifth end to third end, opposite. Okay, now the strand which is formed here in this direction, it is 3 to 5, while this one will form in 5 to 3. Okay, now the main thing you have to remember, this strand is called template strand. Okay, and this strand is called non-template strand. Okay, this is your template strand and this is your non-template strand. That's how your formation of DNA, replication of DNA works. Okay, your primer will bind here. Okay, it will bind and form a starter, just like a curd. Milk need to convert into curd with the help of starter. Your primer will bind here, and when that primer is removed, your DNA polymerase will come and start to the formation of this gene. Okay, in which that energy is used with the help of your DATP, DGTP, DCTP, and DTP. Okay, this is your leading strand or this is your lagging strand. Okay, leading strand is always 5 to 3 in direction. Okay, means this is your template or leading strand and this is your lagging strand. Okay, it is 5 to 3 in direction. Okay, the working of strand is 5 to 3 in the direction, and the replication which is happening, the fork which is forming replication, it is 3 to 5 in direction. Parental strand direction is 5 to 3 and daughter strand direction is 3 to 5. In NCERT it is written in some tremendous form, means in the form confusing form, but clearly the parental strand is direction is 5 to 3 while the direction of daughter strand is 3 to 5. Okay, so now we have three types of DNA polymerase. Okay, DNA polymerase 1, DNA polymerase 2, DNA polymerase 3. Okay, DNA polymerase 1, yes, short chain gap filling. Okay, it fills the gap. Okay, that your lagging strand. Okay, that lagging strand is formed into the fragments. They are not formed in continuous manner. Okay, so it is having that discontinuous form. Okay, so that thing is short chain. Okay, next is DNA polymerase 2. It is least participating and reactive in replication. It is, yes, it is used in short chain formation. Okay, DNA polymerase 1 and DNA polymerase 2 always work on discontinuous strand or on lagging strand, while your DNA polymerase 3, it is the main enzyme and it acts on leading strand. Okay, means on this strand, okay, leading strand, your DNA polymerase 3 work, and this lagging strand, your DNA polymerase 1 and DNA polymerase 2 works. Okay, and the small fragments which made here, these are called Okazaki fragments. Okay, this guy is Reiji Okazaki, who discovered that Okazaki fragments. Okay, see this: first Okazaki fragment, second Okazaki fragment, third Okazaki fragment, fourth Okazaki fragment, and these fragments are filled up with the help of this DNA polymerase 1. Okay, DNA polymerase 1 filled that Okazaki fragment areas. Okay, means the part, see this empty space. Okay, this blue color empty space. Okay, this DNA segment part and this empty space, it is filled by your DNA polymerase 1. Okay, this is your replication fork diagram. Okay, so as I said, that leading strand, it always go from 5 direction to 3. Okay, and the daughter strand which is forming on that, it is 3 to 5. Okay, see, from 5 to 3. Okay, and this is 3 to 5. Okay, next, E. coli. Now that DNA replication in prokaryotes. Okay, In prokaryotes, you have circular DNA. Okay. In eukaryotes, you have linear DNA. In prokaryotes, you have circular DNA. So in circular DNA, this process occur in the form of orthodox manner. Okay. So in eukaryote, the replication of DNA take place at S phase of the cell cycle. Failure in cell division after DNA replication result in polyploidy. Okay. Polyploidy, it is a type of chromosomal mutation. Okay. Mutation is of two types, chromosomal and genetic. Okay, gene mutation. So it is a type of chromosomal mutation. Okay. Next, a summary of DNA replication. Again, we have first helicase unwind the parental double helix, then single-stranded binding protein stabilize the unbounded parental DNA. Okay, then say the SSB. Okay, then leading strand is synthesized in the continuous manner in 5 to 3 direction. Okay, by DNA polymerase. And the fourth, the lagging strand is synthesized discontinuous primase synthesize a short RNA primer which is extended by DNA polymerase to form an Okazaki fragment. These blue are your Okazaki fragment, and these red is your RNA primer. These will be removed by your DNA polymerase 2, and now DNA polymerase 1 will come and fill these empty spaces. Okay. Then after the RNA primer is replaced by DNA, the another DNA polymerase not shown, DNA ligase join the Okazaki fragment to the growing strand. Okay, and at last your DNA ligase will come and it joins your DNA, that remaining DNAs. Okay, now your RNA world. Okay, what is RNA world? So RNA world, the term was coined by 1986 by Walter Gibbert, which is we call now ribonucleic acid. Okay, RNA was the first genetic material. There is evidence to suggest that essential life processes such as metabolism, translation, splicing, etc. evolved around the RNA. Okay, these all processes, all the metabolic processes evolved around the RNA. If RNA not there, then not metabolic processes will happen. Okay, RNA use act as genetic material as well as catalyst. There is some important biochemical reaction in living system that are catalyzed by RNA catalyst and not by protein enzymes. Example, splicing. Okay, RNA being a catalyst was reactive and hence unstable. Therefore, DNA has evolved from RNA with chemical modification that makes it more stable. Okay, RNA is less stable because it contains oxygen. Okay, the ribose sugar content oxygen, OH group. Okay, that presence of OH group in your ribose sugar, okay, that pentose sugar makes your RNA unstable, while the absence of that OH group in the ribose sugar of DNA makes it stable. DNA being double stranded and having complementary state further resist change by the evolving process of repair. Okay, DNA is double stranded, RNA is single stranded. Okay, RNA is adapter structure molecule and in some cases catalytic, thus RNA is better material for transmission of information. Okay, it is single stranded, so it easily transfer information from one form to another form. DNA is double stranded, you have to open, then it will be transferred, so it is not used to transfer information from one cell to another cell. Okay, that's why we have mRNA, tRNA, rRNA. Okay, then RNA ribonucleic acid. Okay, structure of RNA is fundamentally same as DNA, but the difference is, first difference is on the place of thymine we have uracil, and second thing is the pentose sugar. In the ribonucleics, that the sugar which is present in your RNA content OH group, while in DNA that OH group is absent. Okay, that thing is written in the place of deoxyribose sugar DNA, ribose sugar is present in RNA. Okay, in the place of nitrogenous base thymine in DNA, uracil is present in RNA. RNA is made up of only one polynucleotide chain, while RNA is single stranded. Exception is there. There is one virus which is called reovirus. It contains double stranded RNA. Okay, normally RNA is single stranded, but reovirus is an exception. It contains double stranded RNA. Okay, next, types of DNA. Okay, sorry, types of RNA. We have three types. First is your rRNA, second is your tRNA, and third one is your rRNA. Okay, so genetically RNA or genomic RNA which is present in some viruses. Okay, like your reovirus, TMV, QB, bacteriophage. These are genetic material. Okay, second one is non-genetic material. Okay, that is non-genetic RNA, which is not going from one generation to another generation. It is only present inside that generation and uses as a metabolic activity, used for metabolic activities. That is called non-genetic RNA or non-genomic RNA. It is of three types: mRNA, tRNA, and rRNA. Okay, now we are going to discuss this thing. So first we have rRNA, which is also called ribosomal RNA. It is the most abundant rRNA present inside a cell. Okay, so it is 80% of the cell total rRNA. So total RNA. Okay, it is the most stable form of RNA. Okay, function, form, structure of ribosome, structural RNA, and some rRNA act as a catalyst or enzyme which is called ribozyme. Okay, normally we studied that enzyme, it is made up of protein, but ribosome is the only enzyme which is not formed from protein. It is made up of RNA, which is rRNA, catalytic RNA. Okay, next we have tRNA, transfer RNA. Okay, which have two structures. One is 3D structure and other one is 2D structure. 2D structure is look like clove leaf like. Okay, and 3D structure is inverted L like structure. Okay, inverted L like structure. So 2D structure, clove leaf like structure discovered by Holy, and 3D structure, which is the actual structure of your tRNA, which is inverted L, discovered by Kim. Okay, and now your tRNA have three arms: D loop, T loop, and anticodon loop. Okay, D loop, T loop, and anticodon loop. Anticodon loop is responsible for the coding of your genetic code. Okay, genetic code. Okay, it's opposite of this anticodon, which is present on your tRNA, and it is present on anticodon loop. Okay, these all T loops and other loops, it is also responsible for different works. Now we are going to see that. Okay, messenger RNA, mRNA, this is the least in number and the most unstable RNA. Okay, it is 1 to 5% of the total RNA present inside the cell. Okay, it is least stable RNA, it is template RNA, it is used in the process of your transcription. Okay, mRNA is produced, that mRNA gets spliced. Okay, and you get mature mRNA. Okay, this is your mRNA structure, in which we have pipe cap and poly A tail on third end. Okay, this is mature RNA. Okay, mRNA, messenger RNA, or matured RNA. Okay, next, the main process of genetics is transcription. Okay, means formation of RNA from DNA. So transcription process is a process of copying genetic information from one strand of DNA into RNA. Means you having a DNA. In that DNA part, an mRNA will be copied by the help of RNA polymerase. Okay, and this opening of this bonds between these strands, it is done by your helicase enzyme. Okay, so the DNA dependent RNA polymerase catalyze polymerization only in 5 to 3 direction. Okay, the processing, the working of your DNA dependent RNA, see there is three categories. First is DNA dependent DNA polymerase, which is work in your replication process. Second one is your DNA dependent RNA polymerase. It works in your transcription process. And third type is your RNA dependent DNA polymerase. That is your reverse transcriptase enzyme. Okay, it is depending on RNA to forming DNA. Okay, now your DNA is dependent RNA polymerase means DNA के ऊपर डिपेंडेंट होके तुम RNA बना रहे हो, तो that is your RNA polymerase. And when you are dependent on RNA and forming DNA, then it is your reverse transcriptase enzyme. Okay, see like this. DNA dependent DNA polymerase process replication. Okay, second one is your DNA dependent RNA polymerase process transcription, and third one is your RNA dependent DNA polymerase, that is your reverse transcription. Okay, DNA dependent DNA polymerase, that is your replication process. DNA dependent RNA polymerase, that is your transcription process. And RNA dependent DNA polymerase, that is your reverse transcription process. Okay, so enzyme which is present in your transcription process is DNA dependent RNA polymerase, means your RNA is dependent on DNA to perform process, to confirm the process. Then this is your transcription process. Okay. Next, it works on 3 to 5th end and acts as a template strand, and 5 to 3 act as a coding strand. Okay. Now this is the very interesting thing. In the process of transcription, you will say that names are opposite. Okay. This is the strand which direction is 3 to 5, and this is the strand which direction is 5 to 3, opposite. Okay. Now on this strand you have nitrogenous bases. Against A you have T, against G you have C, against C you have G, against T you have A, against C you have G, again A you have T, again G you have C, and again this C you have G. Okay, this is normal coding. Okay, now mRNA will come in the play. This will become U, this becomes C, this become G, this become A, this become G, this become A U, this become C, and this become G. Now see this coding and this coding. Okay, only the difference is of U and T. Okay, that's why this strand is called coding strand, and this strand is called non-coding strand. This is called nonsense strand. This is called sense strand. This is called template strand. This is called non-template strand. The reason why it is called coding strand, it is because the sequence which is written here, T, C, G, A, G, T, C, G, it is similar on your mRNA. The difference is only of U and T. Okay, U, C, G, A, U, G, U, C, G. Same thing is written here, only on the place of U, you have T. Rather than other sequences same. That's why this strand is called coding strand and this strand is called non-coding strand. This strand is called nonsense strand. This strand is called sense strand. This strand is called template strand. This sense is called non-template strand. Template, why it is opposite in template? Because on this strand we are doing the transcription process, that's why it is called template, and on this strand we are not doing any transcription process, that's why it is called non-template strand. Okay, so this is strand coding in the transcription process. Next, we have both strands are not copied during the transcription, only one strand is copied, which we call your non-coding strand or nonsense strand or template strand. The opposite one which is not get copied is called coding strand or sense strand or your non-template strand. Okay. Now there is 3 RNA polymerase which take action in the process of transcription. First is your RNA polymerase 1, which is made up of 20S RNA, 18S rRNA and 5' rRNA. Next, your RNA polymerase 2, which makes your hnRNA and mRNA, and RNA polymerase 3, which forms tRNA, 5S RNA and snRNA. So the maximum part of your ribosome, it is made by RNA polymerase 1. Okay, in eukaryotic cell you have ribosome. This is the larger subunit, okay, and this is the smaller subunit of your ribosome. This is 60S, this is 40S. We are talking about eukaryotic cell, okay, and this part is made up of 3 units: 28S rRNA, then your 5S rRNA and 5.8S rRNA. Okay, and this part it is made by your 18 rRNA. Okay, so 28S 18S, 18S, and 5.8S, 5.8S. It is produced by your RNA polymerase. This remaining 5S, it is produced by RNA polymerase 3. Okay, in eukaryotic and prokaryotic both. Okay. So prokaryotic one type, in prokaryotic only a single type of RNA polymerase is present, which we call normal RNA polymerase. Okay, this makes your tRNA, mRNA and rRNA. Okay, in prokaryotic cell, tRNA, mRNA and rRNA produces by your RNA polymerase single, but in eukaryotic cell, RNA polymerase 1 responsible of forming these three RNA, RNA polymerase 2 responsible of forming hnRNA and mRNA, while RNA polymerase 3 is responsible of forming tRNA, 5S rRNA and your snRNA. Okay. Functions of RNA polymerase: 5 to 3 polymerization, chain formation of RNA and breakdown of hydrogen bond, opening of DNA helix. In replication process, we studied that helicase is responsible for the opening of DNA helix, but in the transcription process, RNA polymerase is the responsible for opening of your DNA helix, RNA helix. Oh, sorry, DNA helix. Okay. RNA polymerase in eukaryotes, as I said, RNA polymerase first, it transcript rRNA 28S, 18S, 5.S, same thing which is written here, it is here in the form of diagram. Okay. Next is your transcription unit. Okay, what are the parts of transcription unit? Okay, so this area is called promoter region, this area is called transcriptional unit, and this area is called your terminator. Okay, again, this is your promoter starting point, this is your structural gene where your coding thing take place, and this is the terminator area where the transcription process stops. Okay, so your transcription three regions are there: promoter region, starting point, then structural gene, the area where the transcription process take place, and the terminator area where the process of transcription stops. Okay. So promoter region, the promoter and terminator flanks in the structure gene in the transcription unit C. If this is a strand of DNA, then this part is your promoter. This area to this area is your structural gene where transcription taking place. And from this point to this point is your terminator where transcription stops. Okay, that thing is written here: promoter region, then your terminator, and at last the types of genes. Okay, we have monocystonic gene and we have polycystonic gene. Monocystonic gene, it is called split gene, which is present in eukaryotic cell, and this is your polycystonic gene, which contain exon and intron both, and it is present in prokaryotic cells. Okay, this is your monocystonic gene, means exon, intron, exon, intron, exon, intron, exon is present. Okay, and this is your polycystonic gene, in which gene 1, gene 2, gene 3 is present regularly in the regular interval. The main thing you have to remember in this: monocystone is present in eukaryote, polycystone is present in prokaryote. Okay, so splicing is done in, so splicing procedure done in this monocystonic. Okay, means in eukaryotic genes. Okay, in eukaryotic DNA we do splicing process. In prokaryotic we do not require, because there is no intron and exon present in your prokaryotic genes. Okay, they are directly connected to each other. In splicing we remove this intron part and attach these red exon parts. Okay, so splicing procedure, it is done in eukaryotic cell or monocystonic structural gene, while the splicing process not take place in polycystonic structural gene, which is present in prokaryotic cell. Okay. Next thing is your mechanism of transcription. Okay, same thing that I tell you before, that it will start it at the promoter region where you have sigma factor will get attached with the promoter region. Then the that sigma factor runs with RNA polymerase and open your DNA helix and move forward. Now this process is called elongation, when it move forward and start forming RNA chain. Okay. Now at the termination region you have rho factor, which attach with RNA polymerase and stop the process of transcription. Okay. Next is your post transcription scenes. Okay, after transcription, what happens? Okay, that thing is called split gene. Okay, means splitting, cutting the gene. Okay, it is discovered by Sharp and Robert, and they awarded Nobel Prize in 1993 for this split gene theory. Okay, so see what happened in split gene. This is your mRNA, but it is not mature now. Okay, it contains both intron and exon. Okay, for making this thing active for translation process, we have to remove intron, this part. Okay, this part should be removed, then this mRNA become active, and this removal of this intron part is the process is called splicing. Okay, and it is done by a complex which is called splice spliceosome complex. See, this is your spliceosome complex. Okay, this cut your intron. It sits on intron part and cut that intron part. Okay, now before that thing, we have to do some extra things. Okay, from making that mRNA mature, first we have to apply cap and tail on that mRNA. Okay, so capping is done by your on the fifth end, and your tailing is done on third end. Capping we use your methyl guanosine triphosphate cap. Okay, 7 methyl triphosphate cap. Okay, it is seated on your fifth end. While in tailing we add polyadenylation. Okay, means a 400 to 300 a is added on the 3 end, which we call poly a tail. Okay, ऐसे ए करके पूरी चेन बन जाती है. Okay, that thing is called poly a tail. Okay, while on the cap we put 7 methylated guanosine triphosphate. Okay, and after that we put spliceosome, and that spliceosome cut this thing from the middle. Okay, and at last then ligase will come and join that part, you get mature mRNA. Okay, next is your genetic code. Okay, so it is a sequence of nucleotide and mRNA that contain information for protein synthesis. Okay, it is sequence of 3 bases determining a single amino acid called codon. AUG, UAC, GUA, CUG, ACG, UAC, GUA, CGA, UAA. These all are genetic codes. Okay, in this mRNA you have 1, 2, 3, 4, 5, 6, 7, 8, 9 genetic codes. Okay, this is your initiation codon, this is your termination codon. Okay, or end codon. Okay, so these are the 3 scientists. Okay, Francis Gamow, Hargobind Khurana, Marshall Nirek, and Sarva Okara. Okay, these search different theories, different experiment to tell that your genetic code is in triplet. Okay, so Savaro-Okara enzyme, which is called polynucleotide phosphatase, is used to polymerize RNA with defined sequence that template independent manner. Okay, independently they will tell you what is the sequence of mRNA, what is the sequence of codons on mRNA. Okay, sequence of codons present on mRNA. Okay, this is your Savaro-Okara. इस बंदे के नाम पे एंजाइम आ गया जिसको बोलते हैं Savaro-Okara enzyme. Okay. Next, this is the experiment done by your Marshall Lindbergh and Mithai. Okay, इसका experiment था ये. Okay, they developed cell-free system for protein synthesis. Finally, help the code to discipline. They use synthesized homopolymer of RNA like poly U, poly C, poly A, means they form a chain of U, a chain of C, a chain of A. By the help of Savaro-Okara enzyme, this enzyme is being used. It make polymerization of these nucleotides. Okay, this enzyme help in the polymerization of RNA with defined sequence in template independent manner, enzymatic synthesis of RNA. These RNA will allow to translate forming protein in cell-free medium. Okay, means when they do this experiment, they will see that three coats comes together and form a protein. Okay, three nitrogenous bases come and form a one genetic code by the help of this experiment. Okay, next is your Har Gobind Khurana. The chemical method developed by Har Gobind Khurana was instrumental in synthesis of RNA molecule with defined combination of bases, homopolymer and copolymer. Means he formed he forms homopolymer just like this. Okay, and copolymer in which he differentiate the nitrogenous bases. Means on the place of AAA he used GAC, AUG, CUG, CAC. Okay, GAC, all different things. Okay, now we have 20 types of amino acid involved in translation. Okay, so these are the 20 amino acid which is present in the process of translation. Out of that, 10 are essential amino acid and 10 are non-essential amino acids. Okay, so this is a trick to remember your amino acids. Okay, G gas space CT. Okay, glutamine, glutamic acid, glycine, alanine, aspartic acid, asparagine. This is your GA, G cube, A cube. Glutamine, glutamic acid, glycine. Alanine, aspartic acid, aspartagine. Okay, then S is your serine, P is your proline, C is cysteine, and T is tyrosine. Okay, again G cube, A cube, S, P, C, T. Okay, gas space CT. G is glutamine, second G is glutamic acid, third G is your glycerine. First A is your alanine, second A is aspartic acid, and third A is asparagine. Then S is serine, and the C is cytosine, and T is your tyrosine. Okay. Second, the trick for remembering your non-essential, okay, that is your PVT, Tim Hall, private Tim Hall, in which P is phenylalanine, V is valine, T is threonine, this T is tryptophan, I is isoleucine, M is methionine, H for histidine, A for arginine, L for leucine, second L for lysine. Okay, so this is the trick to remember non-essential amino acid. These are essential, this is non-essential. Essential are those which is not produced inside your body, non-essential are those which is can produce inside your body. Okay, next is your the genetic code dictionary. Okay, you only have to remember one thing for this. Okay, that thing is this. U C A G. Only remember this thing. Okay, again U C A G. Okay, and here U C A G, and same thing in the next block U C A G, next in second, third U C A G, and last again U C A G. Okay, and you can make this table. You only remember UCAG. Okay, next thing you have to remember then which block belongs to which amino acid. Again I have a trick for that. First line, okay, this line, in this you have phenylalanine, leucine, isoleucine, methionine, and valine. Trick is pehle live in my middle village. Okay, pehle live in middle village. Okay, या फिर pehle lives in my village. Okay, pehle is equal to phenylalanine, live leucine, in isoleucine, middle methionine, and village valine. This is for first row. Okay, now second row. Okay, second row, sir proposed 3 alia. Okay, sir serine proposed proline 3 threonine and alia alanine. Okay, again this is the second row. Okay, sir proposed 3 alia, serine, proline, threonine, and alanine. Okay, second row, that is tire stop his glue as lie as gulab. Okay, his glue as lie as a gulab. Okay, in which tyr is your tyrosine, these two are stop codon, that was stop, then his is stamine, then gn is your glutamine. Okay, s is aspargine, then lysine, then aspartic acid, and last is your glutamic acid. Okay, in this trick you will see that in aspartic acid you have 2 times A, and in gulab you have G and A, glutamic acid, aspartic acid. Okay, last trick is for cis, stop, tripping, argue, say argue, glycine. Okay, cis is equal to cysteine, stop codon, then trip is equal to tryptophan, then arginine, then serine for say, then again arginine, then glycine. Okay, cis, stop, tripping, argue, say, argue. Cis, stop, tripping, argue, say, argue, glycine. Okay, in which cis is your cysteine, stop is your stop codon, trip is your tryptophan, argue is your arginine, say is serine, and then arginine again, and the glycine. Okay, that's how you can remember this genetic code table. Okay. Next, the silent features of genetic code. Okay, what are the silent features? First, codon is triplet. They always come in triplet, means three nitrogenous bases form your genetic code. Second, genetic code is unambiguous, specific, one codon specific only for one amino acid. Okay, that thing, another genetic code does not code for different amino acid. Okay, then genetic code is degenerate in nature. Okay, all amino acid except tryptophan and methionine, because they have only one genetic code. Degeneracy means one amino acid can have different genetic codes. Okay, just like your leucine have six different genetic code. Okay, isoleucine have three genetic codes. Valine have four genetic codes. This is a degeneracy of genetic code. Means a single amino acid can have four genetic code. But GUU is only code for valine. It is not going to code alanine. This feature of genetic code is called unambiguous in nature. Okay, and second nature of your genetic code is degeneracy, that one amino acid can be coded by four different genetic codes. Okay, leucine can be coded by 6, isoleucine coded by 3, valine coded by 4, threonine can be coded by 4, alanine coded by 4. Okay, exception is there that methionine is have only one genetic code and your tryptophan having only one genetic code. Okay. Next, the genetic code in the mRNA is read continuous as there is no comma, pause or overlapping. Okay, means AUG, after that GAC, then after that CCA, after that UAC. That is the reading of sequence of mRNA. Okay, then genetic code is nearly universal. Okay, it is universal. It is present. It is code. Means if you AUG is coding for methionine, then it will code methionine AUG in my body too. In the body of cow, AUG codes for methionine. In the body of bacterium, AUG code for methionine. In the body of kangaroo, AUG codes for methionine. Okay, so it is universal, but there is some exceptions. That is, some exception of rule is present in the mitochondrial codon in some protozoans. Okay, the codon which is present in mitochondria code differently, and some protozoans, ciliated protozoans, they have different type of codon sequences. Okay, so codon AUG is a dual function. It codes for methionine amino acid and also act as an initiation codon. Okay, two functions AUG have. First, it is an initiation codon. It starts the process of translation. If a mRNA does not contain AUG, translation process does not start. Okay, because AUG is the starting codon. Okay, and it codes for methionine. Okay, and rarely GUG or valine codes act as an initiator. Rarely, in some cases, GUG also act as a starting codon. Rarely, some means out of 100, only 1% cases there where GUG act as a starting codon. Okay. Next thing is codon is triplet made up of three adjacent nitrogenous bases. Triplet code specific placement of one amino acid in a polypeptide chain. 61 codons are there. 61 codes for 20. Totally in that chart you have 64 codons. Out of 64 codons, only 61 do coding. Remaining 3 do not code, that are your stop codons. Okay, the genetic code in the mRNA is read continuous and there are no comma, pause or overlapping in between the triplet. Again, the same thing is here. Okay, insertion and deletion of one or two bases changes the reading frame from the point, and insertion or deletion, it is called frame shift insertion or deletion mutation. okay, which we studied in mutation part of that chapter. Okay, again same thing. Then hypothesis. Okay, it was proposed by Crick, normally anticodon. This is your Bobel hypothesis. Okay, Bobel कहाँ चला गया इसका? That is Wobble hypothesis. Okay, it was proposed by Crick. Normally, an anticodon records only one codon, but sometimes if it records two codon, then it called wobbling. Okay, one codon is normal coding. When it codes for other codon, it will start doing wobbling. Okay, see, for example, GAG anticodon and CUC, it's your codon. When it's do coding, it is normal coding. But on the place of C, U will come and it will start doing coding. Then it will called vowel coding or vowel pairing. That thing is called vowel hypothesis. Okay. The reason of wobbling is it always on third position of codon. Okay, first, second, third. The third position, it is responsible for vowel pairing, and in this the bond which is formed between G and U, it is unstable. It will degenerate soon. Okay. Next is your translation process. Okay, formation of protein from mRNA is called translation. Okay, so it takes place in ribosome. Ribosome consists of structure of RNA and about 80 types of proteins. Okay, ribosome also acts as catalyst. 23 as RNA in the bacteria and the enzyme ribozyme. These are the reason why your ribosome acts as a catalyst. Okay, for the formation of peptide bond, peptidal transfer enzyme in large subunit of ribosome that act as a help in the formation of different enzymes and act as a catalyst for your ribosome. Okay, so steps of translation is your charging of tRNA, initiation, elongation, and termination. Okay, so first we have to see the machinery, the machines, the items which is present in the process of that translation. Okay, so first is the ribosome. Okay, ribosome is the most important thing, the site where the process of translation take place. Okay, second one is amino acid. Okay, that amino acid which is going to be bind with tRNA. Third one is mRNA, due to which that coding will take place. Okay, tRNA, the vector molecule which contain anticodons, which read the codon which is present on your mRNA, and amino acid tRNA synthase, which is responsible for charging of amino acid. Okay, so first step is your activation of amino acid. Okay, in which your amino acid get activated. So this is done by with the help of ATP and enzyme amino acyl tRNA synthase enzyme, which forms amino acyl AMP enzyme complex and inorganic phosphates. Now your mRNA become your amino acid become charged. Okay, now you have the time for charging of tRNA, means loading of tRNA. That activated amino acid should be loaded on tRNA. That process is called charging of tRNA or loading of tRNA or amino acylation of tRNA. Okay, this is your normal tRNA which is not charged yet. Okay, and here you have amino acid tRNA synthase enzyme with activated amino acid. Now this activated amino acid will put on your third end of your tRNA, and now your tRNA become activated tRNA or charged tRNA or loaded tRNA. Okay. Next process is your initiation of polypeptide chain. Okay, means synthesizing of polypeptide chain. It again consists three steps: initiation, elongation, and termination. Okay, initiation, it is done with the help of AUG. Okay, the methionine containing charged tRNA attached with the start codon and mRNA with the anticodon at the P site. There is three sites: E site, exit site; P site, peptidal site; A site, amino site. Okay, amino acyl site. Okay, so in this site your tRNA get bind. Okay, and wait for next codon, next anticodon, next tRNA to get attached here. Okay, due to which, see, this is your AUG, USC, and it contains methionine. Okay, and now your second tRNA will come and bind on A site. Okay, when that second, here, देखो, ये second, okay, A site, on the A site, the second amino acid will come, charged amino acid will bind here. Okay, and it makes a bond with your methionine. Okay, which is your peptide bond, and it is produced by transferase enzyme. Okay, transferase enzyme is responsible for formation of this bond between methionine and your threonine. Okay, and it is on your A side. Now, when this bond formation take place, this tRNA detach. Okay, this tRNA from this side get detached and your ribosome move ahead. Okay, now this side will become P site, this become E site, and this become A site. Okay, and this is the elongation, how this elongation will happen. Okay, now in next histamine will come, it will bind. Now in the next will it will become E site, it will become P site, it will become A site. Okay, that's how the step runs. Okay, and this is the elongation of that process, translate the part of translation process which we call elongation. Okay, and last is your termination. Okay, it is done by with the help of termination codon, which is present at the end of your mRNA, which is your UAA stop codon. Okay, it will stop the process of that process of that protein synthesis, the polypeptide chain synthesis, and it is alone not do this thing. Okay, with RF1, RF2, RF3. The full form of RF is releasing factor. Okay, and here in the initiation phase, we have IF1, IF2, and IF3. That is your initiation factors. Okay, so that's how your polypeptide chain form with the help of your releasing factor and termination codon or stop codon. Okay, so protein synthesis stop when stop codon UAA, UGA, UAG of mRNA reaches at the A site. These stop codons are not recognized by any tRNA because we do not have amino acid for these codons. Okay, we do not have anti-codon for these three codons. That's why it is your stop codons. Okay, the P site is hydrolyzed and completely polypeptide chain is released with the help of releasing factor, that is RF1, RF2, RF3. Okay, it will bind here at your stop codon area and your polypeptide chain is released. Okay, and it will go for protein synthesis inside the cell. Okay, next is your regulation of gene expression. Okay, so the mechanism which stimulates the expression of certain gene inside the cell is called gene expression. Okay, or gene regulation. It is possible only if organism have mechanism of regulating gene active by allowing some of the function and other to restrain their activity through switching on and switching off a system. Okay, see there is two type of switch inside your gene: switch on or switch off. Switch on take place when your gene have to prepare protein, and when your gene does not have to prepare any protein, it will become switch off. Okay, this means the genes are turned on or off as per requirements. Okay, so the molecules of metabolite that come in this type of these genes are termed as inducer, and the phenomena is called induction. Okay, just like in your lactoferrin. Okay, inducer is your lactose. Okay, so types of gene, we have two types of gene: non-constitutive genes which called luxury gene, and constitutive gene which is called housekeeping gene. So these genes are not always remain on or off. Okay, second, their product are not constant and need for cellular activity, means the product which is produced during by the help of these luxury genes are not regularly required in your body. Okay, for this gene regulation is required. Okay, means switch on switch off you have to required for this. When the protein required in your body, it get on. When the protein not required in your body, it get off. That is your luxury gene. Next one is your housekeeping gene. This remains always on. Okay, the product of constant needed for your cellular activity, the product which is always need inside your cell, it's produced by housekeeping genes. Okay, these genes are not need to regulation because that product you always need, so there is no on off switch, it remains only on. Example of this gene is the gene of glycolysis cycle, Krebs cycles. Okay, the enzyme which is produced inside your glycolysis and Krebs cycle produced by these housekeeping genes. Okay, next, gene regulation. Okay, gene regulation include a wide range of mechanism that is used by cell to increase and decrease the production of special gene products. Okay, means on and off you are doing different procedure, you are doing oning and offing of the switch, and when you do on means you require the material, when you off that thing means you do not require that material inside your body. Since transcription and translation are energetically very expensive process, these have to be tightly regulated. ATP, GTP, CTP, TTP, all these things are required in your translation and transcription. So it is never remain on. Okay, so you have to control that thing, and for that you have gene regulation. Okay, so gene regulation is the metabolic, physiological, environmental condition that regulate the expression of particular gene. Okay, when your body requires the gene, then that operon get on, and when your body do not require that gene, it will become off. Okay, the development and differentiation of embryo into adult organism also result of the coordinated regulation or expression of several set of genes. Okay, that growth hormone, thyroid hormones, okay, insulin, that glucagon, that aldosterone, adrenaline, all these hormones coming out from your body at a specific time. Okay, so that required on and off. Okay, if just for example growth hormone, take a growth hormone example, if that switch remains on every time, then your body become large. Okay, and that they convert into a one disease which we call gigantism. Okay, and if that producing gene remain off every time, then it will cause your dwarfism. Okay, so gene expression result in formation of polypeptide. It can regulate it at several levels. These are the levels. ट्रांसक्रिप्शन लेवल पे अपन यूज करते हैं इसको, प्रोसेसिंग लेवल पे यूज करते हैं, ट्रांसपोर्ट ऑफ एमआरएनए फ्रॉम मॉलिक्यूल टू साइटोप्लाज्म, मीन फ्रॉम न्यूक्लियस टू राइबोसोम, देन वी यूज दिस थिंग, एंड ट्रांसलेशन लेवल फॉर्मेशन ऑफ पॉलीपेप्टाइड. At these levels we use gene regulation inside the eukaryotic cell. Okay, next, gene regulation in prokaryotes. Okay, in prokaryotes it is contained in the form of operon. Okay, we say that gene regulation take place in prokaryotic cell with the help of operons. Okay, so the predominant site of control gene expression is transcription initial levels. The mechanism of gene regulation in prokaryotic was given by Francis Jacob and Jacob Monod. Okay, Jacob and Monod were the first to elucidate the transcriptionally regulated gene system, which we call lac operon. Example. Okay, normally we call them operon. Okay, they work on E. coli and give operon model. This is your operon model given by Jacob and Monod. Okay, operon in bacteria, more than one gene is arranged together and regulate the unit called operons. Okay, your operator gene, regulator gene, inducer gene, all these genes sit together and control the expression of particular enzymes, particular gene, then it is called operon. So in operon you have four items, four components. First is structural gene, which is responsible for the formation of protein. Okay, then regulator gene, who control the regulation. Okay, operator gene, which is responsible for the operation of RNA polymerase, and promoter gene, which is the binding site of your RNA polymerase. Okay, so lac operon is in our course. There is different operon like val operon, his operon, ara operon, trip operon is for tryptophan, ara operon for alanine, his operon for histamine, and val operon for valine. Okay, in our course we have lac operon, lactose operon. Okay, so lac operon is operon control lactose metabolism in bacterium like E. coli. A regulatory or inhibitory gene codes for repressor protein. Three structural genes, Z gene, Y gene, and A gene. Z gene for beta galactosidase, Y gene is for permease enzyme, and A gene for transacetylase enzyme. Okay, an operator region and a promoter region. Okay, gene and operon functioning together in same and related to metabolic pathways. They always functions together. Okay, so this is your lac operon. See, this is your lac I gene. Okay, regulatory gene which produce repressor protein. By the transcription translation of lac I gene, we produce repressor. This repressor binds with operator when there is no lactose inside the body of bacterium. Okay, and due to the binding of this repressor to operator, your RNA polymerase cannot go ahead and bind to your structural gene to perform transcription and translation process. Okay, so now when lactose is available to bacterium, that lactose go and bind to the operator, and the way for your RNA polymerase is opened, and your RNA polymerase reaches to the structural gene and start producing beta galactosidase, permease, transacetylase. ओके, ये एक तरह का स्पीड ब्रेकर होता है जो एक्टिव रिप्रेसर होता है. इट इज अ टाइप ऑफ स्पीड ब्रेकर विच इज प्रेजेंट हियर, विच इज avoiding the movement of RNA polymerase from here to here. Okay, see mechanism. This is repressor. Okay, this repressor go and bind with your operator region. Okay, due to the binding of this repressor to operator, your RNA polymerase which is present on promoter region, it does not move ahead. Okay, and it not move ahead, there is no processing, no working of structural gene, there is no formation of beta galactosidase, your acetyl that acetylase or transacetylase or permease enzyme. Okay, now your of that lactose will come in the play. Okay, lactose will come and bind on the operator, bind with that active that repressor gene. Okay, due to that binding with your repressor gene, now this repressor will not go and bind to the operator. Okay, and the way for RNA polymerase is open, and RNA polymerase go here and start doing transcription. From that, translation will perform and you get beta galactosidase, permease and transacetylase enzyme. That is your uh, gene regulation. Okay, last second topic of this chapter is DNA fingerprinting or DNA profiling. Okay, so this technique is to identify the similarities and differences of DNA fragments of two individuals, developed by Alfred Jeffrey in 1985. Okay, this is your Alex Jeffrey. Okay, so principle: DNA of human are almost 99.9% same, but varies a small amount of 0.1% is decimal. Means the DNA which is present in my body and in your body is 0.1% different. Remaining 99.9% DNA in you and my body is similar. Okay, so human genome is 3.3 into 10 to the power 9 in base pair. Only 0.1% is different in that. Means 3.3 into 6 10 to the power 6 base pair are different in me and you. Okay, this difference are mainly at repetitive DNA or satellite DNA, which we call this site. Okay, so this is person 1, it having 8 repeats. Person 2, it having 9 repeats. Person 3, it having 10 repeats. Every person on this earth having different repetition of satellite DNA. That is the 0.1% difference between person A to person B. Okay, so what is the satellite DNA now? Okay, so during the density gradient centrifugation, maximum repeated DNA appear as a satellite. So this DNA referred as satellite DNA. Okay, see, this is a uniform DNA. Okay, in which the DNA is stable. Now we will do centrifugation, and the satellite bands which is present on the top of the test tube is called satellite bands, and the remaining one is called major peak. Okay, so this DNAs do not code for any protein. Okay, it is not doing any coding, not doing any translation. It makes up large portion of human genome. Human genome का large portion made by your satellite DNA, that is 3.3 into 10 to the power 6 base pair. Okay, it shows high degree of polymorphization variation in population. Means a population of different individual contain different type of satellite DNA. Okay, it also show high degree of polymorphization in homologous chromosome of an individual. In homologous pair, one chromosome come from father and other come from mother. Okay, means a single homologous chromosome contain different satellite DNA. Okay, so it is form the basis of DNA fingerprinting. The basis of DNA fingerprinting is this satellite DNA. Okay, because that satellite DNA is even different in homologous chromosome. Okay, so depending on the base competition, A T rich or G C rich, we differentiate this thing. Okay, so by this we get two type of satellite DNA. One is micro and second one is milli. Okay, so this is your microsatellite short tandem repeat STR, which is also called to microsatellite, and this is mini satellite, which is called VNTR. Okay, so VNTR variable number of tandem repeats, and your that STR short tandem repeats. VNTR variable number of tandem repeats, and STR is short tandem repeats. Okay, so satellite DNA is classified as microsatellite, mini satellite, etc. based on the base composition. If it is AT rich or GC rich, length of segment and number of repetitive units. Okay, a DNA sequence which is randomly repeated in many copy number is called variable number of tandem repeat or VNTR. VNTR belongs to mini satellite. Okay, mini satellite is equals to VNTR and microsatellite is equals to short tandem repeat STR. Okay, so microsatellite it is made up of 1 to 6 base pair repetition means 1 2 3 4 5 6 ऐसे base repair होते जाते हैं, इसमें repeat होते जाते हैं. Okay, in mini satellite what happen, 11 to 16 base pair repeat. Okay, it is also called VNTR, it is also called SSR, simple sequence repeats, or your STR. Okay, short tandem repeats. Okay, size of NTR varies in size from 0.1 to 20 kb, means it will contain 20,000 nitrogenous base pair or 100,000 base pairs. Okay, then end of NTR palindromic sequence is present. End of the palindrome, the end of your satellite DNA of mini satellite, we have palindromic sequences present. Okay, the repetition number of VNTR is vary in homologous chromosome also. As I said, it is different in mother, that chromosome coming from mother and coming from father, it is different. Okay. Next, the process of DNA fingerprinting. Okay, the steps for DNA fingerprinting. First, we have to isolate the DNA. Second, digestion of DNA by restriction endonuclease. Third, separation of DNA fragment by gel electrophoresis process. Okay, that again negative positive thing. Okay, then transferring DNA fragment to synthetic membrane, that is your nylon sheath. Okay, and cellulose sheath. Then hybridization using by radioactive level VNTR probes, mean you are hybridizing, you making a hybrid DNA, adding that radioactive DNA into a normal DNA. Okay, and at last you have the detection of hybrid DNA by autoradiography, means bombarding X-ray on hybrid DNA. Now that hybrid DNA is imprint on X-ray film. Okay, see like this. This is your isolation of DNA. Okay, DNA sample, from that you isolate the DNA. Okay, now you will do cutting of that DNA by the help of restriction enzymes. Okay, after that you will do gel electrophoresis. Okay, in which you will tag short and long that large and small fragments into the side, that negative charge side having large molecules and the positive side have smaller molecules. Then you put a nitrocellulose sheet on this. Okay, and this process is called southern blotting. Okay, now electrophoresis, and you put this nitrocellulose sheet on that, and this process is called southern blotting. Okay, now that in this pigmenting thing will be imprinted on your nitrocellulose sheet, means you are copying, you are taking that impression. Okay, impression of this thing on nitrocellulose sheet. Okay, after that you will do fall that X-ray on them, and you will find that this type of pattern will appear. Okay, when you put X-ray on this. Okay, so the DNA fingerprinting application is forensic tool to solve parental rape and murder cases. Okay, for diagnosis of genetic diseases, to determine phylogenetic status of animal, and to determine population and genetic diversity. Okay, that's, and then that is a case of to determine the child father, father 1, father 2, father 3, the most similar thing which will give to father 1. Okay, this child the DNA match with the father 1, then it is your father 1. Okay, is the father of this child. Next is the case, crime scene का case है ये. Okay, in which you have this C, it is the sample which is collected from the murder site. Okay, and this is the DNA of two suspects. Okay, in this can be both, can these both can be murderer. Then you have to match the C DNA strip with these both, and the matching one, this blue match with this light blue, this light blue matches with this light blue, this dark blue matches with this dark blue, this dark blue matches with this dark blue, this red with this red, and this red with this red, means that C match with the B, means that is the murderer. Okay, B is the murderer of C. Okay, next last topic of this chapter is Human Genome Project, HGP, a mega project. Okay, so all thought being individual of same species, every human being has different identities, different in their perfection makeup. Okay, genetic makeup is different. So we have to study that different makeups, and we store that information in computer by the help of Human Genome Project. It was thus an ambitious and scientists from all over the world sequence human genome. Okay, human genome is being In 1990, this mega project launched by USA. Okay, and in coming new years, UK and different countries will join this thing. Okay, so welcome trust, a trust of UK joined this project which is launched by USA, and this project become more mega. Okay, that's why it is called mega project. See, initially this project is launched by USA. Okay, and now after some years there is one trust which is called Welcome Trust of UK joined this project, and after some more years more countries come and join this project. That's why it is called mega project. Okay, the Human Genome Project was a 13-year project which was completed in 2003. It is started in 1991. Near 1991, 1992 it is started. Okay, that's why it is also a mega project, because it takes 13 years to complete. Means a genome, a human genome content, how many gene has been discovered, has been studied in 13 years of time. Okay. Next thing, two different approach were taken to analyzing human genome. Okay. First is identify all the gene only coding region of the DNA that expressed as RNA, referred to express sequence text EST. And second different approach is that sequencing the whole set of genome that contain all the coding and non-coding sequence. Means the coding sequence, the non-coding sequence, the sequence which is performed to made amino acid has been read it, and non-coding sequence which is not perform any coding has been read it. So all the sequencing which is present inside your DNA get studied. That is the two approaches done in Human Genome Project. So first is express sequence text EST, focus on identify all the genes that are expressed as RNA. Means the RNA expression, that mRNA, messenger RNA, tRNA, rRNA, that all the expressions which we get due to RNA has been studied. That is your express sequence text. Okay, and second one is sequence annotation, that is your coding and non-coding sequencing study. Okay, so sequencing whole set of genome containing all the coding and non-coding sequence, and later assigning different regions in the sequence with the function. See, these are the different colors. The different color represent different sequencing, different regions. Okay. Next is goal of Human Genome Project. What are the goals of Human Genome Project? Why we will doing this Human Genome Project? So first is identifying all the estimated genes present in human DNA. There is different genes. 30,000 genes are present in human DNA. So we have to study that 30,000 genes. That is the goal of Human Genome Project. Sequencing of 3 billion chemical base pair present in human DNA. Okay, 3 billion chemical bases means that A, G, T, C that is present inside your DNA, that concentration is 3 billion. Okay, store this information in database in the computers. Okay, because this thing is not in should be stored in paper, because it is that much large amount of information we cannot put inside the paper. So it is computerized, present software, in the form of software, in the form of database it will store. Okay, improve tool for data analysis. Data analysis means we are doing analyzing data. Okay, for different diseases, different functioning of genes has been studied by the help of this thing. Okay, next, transfer related technologies to other sectors. Okay, your army, okay, medicine sector, army sector, finance sector, you can send the information from them. Army sector, when you are sending that information to army, you will can assign what type of genomic sequence that particular human content. Medicine, for all the genetic disorders, different disorders you are studying. Okay, address the ethical, legal and social issues that may arise from this project. Okay, these some bad countries, the terrorist countries, can also use this thing, so it should be properly secured. That's why your UNO nations have the right to use Human Genome Project database. Not every country should use have the right to use Human Genome Project database. Now that Pakistan does not go and study the Human Genome Project database, if they start doing, they will start something other type of wars. Okay, so Human Genome Project procedure. Okay, how we will do this processing? First, isolate DNA from the cell, then convert into random fragments, small fragments like this. Okay, then clone in host, bacterial or yeast. Okay, BAC or YAC, bacterial artificial chromosome, or your yeast artificial chromosome, BAC, YAC. Okay, now sequencing of fragment using automated DNA sequencer. This is the machine. Okay, software machine which do this thing. Okay, arrange the sequence based on overlapping regions. Okay, means you have to current laga. Okay, these are the overlapping sequence, means you have to overlap this thing upon each other. Okay, next arrange the sequence based on overlapping regions, then alignment of sequence using computer program. Okay, now you have to arrange this in a sequence by the help of program. Different program softwares are there to arrange this thing in a particular sequence, and at last you get a perfect idea, a database which is stored inside the computer. Okay, and this is the full form of BSc, bacterial artificial chromosome, and yeast artificial chromosome. Okay, this is the procedure, this is the diagram from your NCERT, this is the well informed information diagram which is show you how human genome project works. Okay, now next is your Frederick Sanger. This guy developed sequencing for amino acid also. This guy also do coding for protein. In the previous part we were studying about DNA coding. This guy do this thing for proteins also. Okay, DNA is converting to random fragment as there is technical limitation in sequencing very long pieces of DNA, so he start doing sequencing for amino acids. Okay, HG was closely associated with bioinformatics. When computer science merged with biology, it's called bioinformatics. Bioinformatics application of computer science and information technology to the field of biology and medicine, means when biology comes with computer science we call it bioinformatics. Okay. So silent features of human genome project. First, human genome contains 3164 million nucleotide bases, means that 3 million that previously I told. 3 billion. Total number of gene about 30,000. Previously I said that 30,000 gene are present in the DNA of human. Okay. Average gene consists 3,000 bases, but size varies. Largest known gene of human body is dystrophin gene, which contain 2,948 DNA base pairs. Okay, contain 2.4 million bases. Okay, dystrophin gene which is present on X chromosome. This dystrophin gene is responsible for muscular dystrophy. Okay, 99.9 nucleotide base are same in all people, only 0.1 is different, that I said you in DNA fingerprinting. Function of over 50% is discovered gene, now 15% is still remaining. Only 50% gene function we know now, till now. Okay, chromosome 1 has the most gene 2968, while Y chromosome has the fewest 231 in number. Okay, less than 2% of genome codes for protein. Okay, less than 2% of genome code for protein, remaining 98% does not code for protein synthesis. Only 2% part of your whole genome code for protein synthesis, remaining 98% is non-coding gene. Okay. Last, very large portion of human genome is made up of repetitive part, that is your 3.3 into 10 to power 6. Okay, and about 1.4 million location of single base DNA differences. Okay, these are called SNP, single nucleotide polymorphism, or SNPs, like this. Okay, this help to find chromosomal location for disease associated sequence and tracing human history. Okay, human history means our ancestral history, it will discovered by SNPs. Okay, SNP is single nucleotide polymorphism, means a single nucleotide get polymorphized, means having different modes. Okay, so this chapter molecular basis of inheritance completed. Thank you for watching. Now in 10 minutes we are going for second chapter, and thank you for all the viewers who are viewing now, all the 1000 viewers who are watching that live. Thank you for viewing. Okay, in 10 minutes we are coming back. छोटा चैप्टर उठा लेता हूँ कोई. ये हो गया, ये हो गया, मॉलिक्यूलर बेसिक्स ऑफ तो फॉलोवर्स भी बढ़ गए मुझे ऐसा लगता है। हाँ 309 हो गए, 262 थे ना शायद। तो मैसेज कौन कर, मुझे मैसेज ही नहीं दिख रहा किसी का, इसमें रिकॉर्ड तो नहीं हो रहा। हाँ, so now next chapter of 12th NCERT, reproductive health. This chapter basically present in your 12th NCERT because to give you idea about the reproduction. Okay, the health related to the reproduction has been given to you. That's why this chapter is present in this your 12th NCERT. Okay, this chapter tells you what you have to do relate the reproduction related problems which is present inside our society. In Indian society, sex is related to taboo. Okay, so this chapter is given in 12th NCERT to release that misconceptions, misrelationships to sex. Okay, so that is reproductive health. Okay, so what is reproductive health? Okay, World Health Organization WHO defines reproductive health as a total well-being in all aspects of reproduction, physical, emotional, behavior, and social. Means your body should be physically, emotionally, and behaviorally and socially active. Then you are called reproductively healthy person. Okay, means you can produce your progeny easily when your body physically, emotionally, behaviorally and socially active. Okay, this is initiated that program. Okay, in 1951 family planning program is initiated in India, in which we get idea about the that how to plan a family, and this is the first country in world which initiate family planning program. Okay, wider reproduction related areas are operational under the reproductive and child health care program. Wide regions of India has been come under this thing. Okay, wide reproduction related areas are in operation under a reproductive and child health care programs. That wide areas means the many parts of India come under this reproductive health program system, family planning program system. Okay, so such program deals with the following points. Okay, first, give awareness about the reproduction related aspect for creating reproductive healthy society. Give idea what is reproduction, the better ways of reproduction, the misconception which is present in the society of India related to the reproduction, related to the sex. Okay. Second, educate people about birth control, care of pregnant mother, postnatal care, and the child. Improvement of breastfeeding, equal opportunity for male and female child has been given in this program in India. Okay, awareness problem due to the population explosion, social evils like sex abuse and sex related crimes should be go down when you tell all these things to the population of India. Okay, so what is the aim and need of sex education in school? First, to provide right information about sex related aspects in the health to avoid sex related myth and misconception. क्या हुआ? हाँ. Little pause is there. Now we are doing a performing a nice little experiment related to betadine and iodide as in a redox reaction. Okay, in which do a magic trick in which we convert a colorful beaker, brown color beaker into a colorless liquid. Means a brown liquid convert into a colorless liquid. This is a small experiment we are going to done now. We are performing now. पानी water. So now this we have a glassware in which we put water. Okay, now this is betadine solution. Okay, made up of iodine. Now we will put this thing in this. Okay, now you are seeing that the color of the solution is brown. Okay, थोड़ा माउल स्ट्रिपी होने वाला है. Okay, this is the tablet of vitamin C, ascorbic acid. Now I put this thing and mix. Now you will see that the brown color is disappeared and it will become colorless. Slowly, slowly this thing will happen. See, the color is fading, start to fading. Okay, more it is going to fade. Slowly, 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 slowly. Now you will see that thing is colorless. So the science behind this experiment is this is betadine solution. Okay, this content iodine I2. Okay, and this is your water in which we put this iodine solution with this tablet. This is your ascorbic acid vitamin C. Ascorbic acid react with betadine and reduce it. Okay, reduce it and do that, due to which what will happen, that ascorbic acid eat the iodine and convert iodine into iodide. Okay, I2 convert into I minus. Okay, that is your iodide. That is the simple experiment will be which we do. Okay, thank you for watching this experiment. That's magical experiment. Okay, again back to the chapter reproductive health. Okay, so the aims and need of sex education in school. First, to provide right information about sex related aspects that help to avoid related myth and misconceptions, the taboo which Indian community relate to the sex. Okay, second, to give proper information about reproductive organs, adolescence and related changes, safe and hygienic sexual practices, and tell about the different sexually transmitted diseases. Okay, so next is population explosion or birth control. How to control the birth? What is the reasons behind the population explosion in a particular community, in particular population? Okay, so the world population, which was around 2 billion in 900, rocketed to 6 billion by 2000 and 7.2 billion in 2011. That is the range, that is the power how your population rocketed in the world. Okay, a similar trend was observed in India too. Our population, which was appropriate 350 million at the time of independence, reaches to the close mark of billion mark by 2000 and crossed 1.2 billion in May 2011. See, in 1947, during the time of independence, our population is 350 million, and by 2000 it will become 1 billion. Okay, that's why at the time of 2999, Indira Gandhi launched some scheme. That scheme is called Nasbandi, in which they will caught male from different area and takes to the sarkar, that the government dispensaries, and do their Nasbandi, that is your tubectomy. Okay, next, a rapid decline in death rate, maternal mortality rate and infant mortality rate, as well as increased number of people in reproductive age, probably reason of these. Death rate means the percent death. Okay, it is decreased. Maternal mortality rate, okay, means mother. Okay, and infant mortality rate, infant, the newborn mortality rate. All these rate when goes down and increase in the age of reproductive age people rate, that age, the section increase in a particular area, then the population get explode. Okay, through our reproductive child health programs, though we could bring our population growth rate, it was only marginable. We have different population control program, but that thing is not proper for our country because of the education is not proper in India. The sex education is not proper in India. Okay, such is alarming growth rate could lead to the absolute scarcity of even basic requirements like food, shelter, and clothing. As you know, when population rises, the food scarcity will happen, shelter scarcity will happen, clothing scarcity will happen. Despite significant progress made in those areas, therefore the government was forced to take up serious measures to check this population growth rate. Okay, and for this we have the reasons for population explosion. What are the different regions of population explosion? The first region is developing health provinces. See, developing health provinces is good. Developing health provinces means developing healthcare system, proper healthcare system. But if a country having very good health care system, then the death rate get decreased. Okay, and if they are not controlling their birth rate, then automatically there is explosion of population take place. Second, engagement in technology, more basic developments in art, your assisted reproductive technologies, okay, that will come into the play, that also increases the birth rate, and due to which population explosion take place. Comparative superior lifestyle have explosive impact on the growth. The more healthier you eat, the more healthier you become, and then more reproductively healthier you become. Okay, that again increases the birth rate and decrease the death rate and cause of population explosion. Okay, next, descending mortality rate. Okay, means death rate is decreased due to again the reason of population explosion. Death less, but more, automatically there is explosion of population take place. Decreasing maternal mortality rate and descending infant mortality, mother mortality rate and your infant mortality goes down, automatically your population explosion take place. Means infant is decreased, mean production of infant is decreased. Okay, it will cause more production of babies, that cause increase in the population of the particular area. Okay, means death of more infant is decreased, automatically the number of, means when there is death of infant is taking place less, means more child are producing, more babies are there in the population, so automatically it will the reason of population explosion. Okay, spike in the number of individual reproductive age, the more reproductive age people, they will do perform more sex, they will perform more progenies, automatically increases the population explosion. Okay. Now, what are the consequences of population explosion? What will happen if population explosion take place very rapidly? First is your unemployment. Okay, people are more, job is less. Automatically, there is unemployment taken place in particular area. Second one is poverty. Unemployment more, automatically poverty is more. You know, in India, 50%, more than 50% people belongs to below poverty line. Okay, you see the movie that Shivaji the Boss, in that Rajini can't tell about that. In India, we have more than 50% people which are below poverty BPL line, and other one that good rich peoples. Okay. Second, third one is high immortality rate and child mortality rate. Okay, means that the baby death rate is very less. Okay, next is your increased crime rate. Okay, if people become more unemployment, automatically they start to do crime, and then crime rate increase. Okay, fifth, distributed social fabric, means different cases of rape, different cases of murder will happen inside that society where population is exploded. Okay, then you have burden on natural resources, more people is there, more vehicles they will use, more food they want, in that they will burn more fossil fuel, that will make burden on natural resources of that particular area. Okay, deforestation, if as you know, when you are more in the population, you need more houses, then you start to cut down the trees, that causes deforestation. Increase waste generation, the more people there, the more waste they produce. Okay, the most they will go for toilet, the more they will use polythene, the more they use different polluting materials, so that will increase the waste generation in particular region. And at last, the most important thing, when the population explosion take place, that is pollution. Okay, pollution that is also harming particular environment of a particular region. Okay, so control measure, motivate small family by using contraceptive methods. Aware people about a slogan which is called "Hum do, Hamare do." Okay, we two or our twos. Many couples have adopted one child norm also, in which they only want one children. That is enough. Okay, saturation rising marriageable age, female 18 year and males 20 year. Now in India it is rise to male 25 year and female 21 years. Okay, that is the main movie in which they will tell that two child is sufficient, not more than two child is good for a family. Okay, now we have contraceptive methods. Okay, ideal contraceptive property, the properties of an ideal contraceptive. First is user friendly, it does not do any allergic reaction, any type of discomfort to the user. Second, easily available, it should be available everywhere, even it is not available only in medical shop, it should be available in confectionery shops, normal malls, normal markets. Okay, next, effective, should be effective, should avoid fertilization process, reversible, it can easily remove from the body parts. Okay, no or least side effect. Side effect is zero, or if having it is very less. Okay, should not interference with sexual drive or sexual act. Okay, it should not interfere during the process of sex, during the process of intercourse. Okay, the natural beautiness of that process should be maintained during the process. Okay, then this contraceptive is an proper contraceptive, ideal contraceptive. Okay, so these are the different contraceptive methods which we are going to study: natural or traditional method, barrier method, intrauterine devices, oral contraceptive, injectable, surgical methods. Okay, so first we have natural traditional method. Okay, we have three: periodic abstinence, coitus interruptus, lactational ammonia. Okay, so periodic abstinence means avoid coitus from the day 10 to 17 of the menstrual cycle, the day at which ovulation take place. Okay, ovulation of egg take place. At that day, when you avoid the sex, automatically there is zero chance of fertilization take place. Second, coitus interruptus means withdrawal of penis. Okay, withdrawal penis from the vagina just before the ejaculation to avoid insemination, that injection of semen inside the female reproductive tract. Third one is your lactational ammonia. It is the absence of menstrual cycle and evolution due to the intense lactation after the parturition. Means when you start doing breastfeeding, when you start giving milk, colostrum to your newborn baby, the level of estrogen is decreased inside your body, due to which you will do not face ovulation of menstrual cycle, does not happen during that phase. Okay, and if it is remains more inside, then there is no chance of conceiving second baby. Okay, next is your barrier method. Okay, to prevent the physical meeting of sperm and ovum, example condom, diaphragm, cervical caps, and bolts. Okay, condom. Why condom are very popular? Okay, there is three reasons. First, it protects the user from STD and AIDS. Okay, it is the most important feature of condom, that it protects user from STD and AIDS. Second, it is disposable, one use karo, फेंक दो. Okay, it can be self inserted and thereby give privacy to the user. No one going to insert it in your body, you have to insert this thing by your own. Okay, condom. What is condom? Made up of rubber and latex sheet. Okay, condom for male cover the penis, condom for female cover the vagina and cervix. Okay, condom are used just before the coitus. They prevent the entry of semen into the female reproductive rate. Nirod, it is a popular condom for male. It is given by government in the government dispensary. You can take this thing freely in government dispensaries. Okay, next is diaphragm, cervical cap, and valve. Made up of rubber and are inserted in the female reproductive tract to cover the cervix during the coitus. Diaphragm, cervical cap, valve, it is for female only, not for male. Okay, they block the entry of sperm through the cervical. They are reusable. Okay, ये reusable होते हैं. You can reuse again that thing. Spermicidal creams and jelly and foams are also used along these barriers to increase their contraceptive efficiency. Means you will insert different spermicidal creams to increase the efficiency of these contraceptive methods. Okay, next one is your IUD, which is the most common contraceptive method in India. Okay, IUD, it is of three types: non-medicated, copper releasing, hormone releasing. Non-medicated means there is no medicine, no hormone, nothing is present there. Okay, example of non-medicated is lippy loops. Okay, they retard the sperm motility and also have spermicidal effect. Example, lippy loops. It is lippy loop, the name given is lippy loop because it's loop like appearance. Okay, and this thread is for the removing that thing again. Okay, when you have to remove that thing from the female reproductive tract, these threads are there. Okay, next thing is copper releasing IOD. Copper ion suppress motility and fertility capacity of sperm. Example, CUT, CU7, multi-load 375, in which multi-load 375 is the most successful that copper releasing IUD. Okay, next is hormone releasing IOD. These are responsible for releasing hormones like progesterone and less amount of estrogen. Okay, they make the uterus unsuitable for implantation and the cervix versatile for sperm. Example, progesterone and LNG20. See what will that hormone releasing IOD works. It will put it here inside the uterus. Now it will release progesterone. Thus, progesterone when go inside your uterus, it will induce muscular contraction, due to which your implantation process does not occur. It makes your uterus very stiff. That why your implantation process does not take place inside your uterus. Okay. So this is birth control intrauterine device IUD. It will inserted by the help of nurse or a medical staff. Okay. Next is your oral contraceptive or oral administration of progesterone and progesterone estrogen combination inside in the form of pills. Okay. Pills are taken daily for 21 days, starting with the first 5 days of menstrual cycle. After a gap of 7 days, it should be repeated in the same pattern till the female desire to prevent conception. Okay. So this is your contraceptive pill strip in which from 1 till 21 these all are tablets which contain progesterone and estrogen, and this from 28 to 20, sorry, from 20 to 28, this is iron containing pills. Okay, it contain irons. Okay, it is for iron. Okay, that loss of blood during the menstrual cycle, in this time iron concentration is also decreased inside the female body. So this 22 to 28, these pills are iron containing pills, while this from 1 to 21, these pills contain progesterone and estrogen hormone. Okay. Next is your contraceptive method, which is injectable. Okay. Now you are going to inject that hormone inside your skin in the form of patches, or these are the strips. Okay, implant which you implanted here. Okay, it will implant it under the skin, and it slowly release the progesterone and estrogen hormone. These strips release that thing rapidly. Okay, and this thing release that thing slowly inside your body. Okay, progesterone or progesterone estrogen combination of IUDs are used in emergency contraceptive within 17 hours of coitus. That is your I pill. Okay, I pill which is given under 70-20 hours of coitus. Okay, it avoids pregnancy due to the rape or causal intercourse. And if you want to terminate pregnancy, then these are the two drugs: mifepristone and misoprostol. These are anti-pregnancy drugs. It will do abortion, directly abortion, abortion of the baby. Okay. Next, surgical method or sterilization. Okay, this is two type: vasectomy, tubectomy. Tubectomy done in male, vasectomy done in male, and tubectomy done in female. Vasectomy means cutting and tying of your vas deferens, and tubectomy means cutting of fallopian tube and tying that fallopian tube. Okay, so this is your vasectomy, cutting and tying of vas deferens, and this is your tubectomy in cutting fallopian tube and tying that fallopian tube. Now that egg which is coming from that ovary does not reaches to your ampulosthemic junction, and from here the sperm does not reaches from testes to your penis area. Okay, and this is the permanent solution of that. If you do not want the child, this is the permanent solution. But in males, within 1 2 week, this thing will remain. Means the sperm release inside, remain inside the male reproductive system, because this area of prostate gland store the sperm. Okay, after 1 2 ejaculation of semen, then the sperm count becomes 0. Okay, and from the day of that tubectomy, woman is become sterile. Okay, it will not have the power to do reproduction. Okay. Next, side effect of hormonal contraceptives. Okay, mid-cycle spotting, blood spotting will happen. Okay, reduced sexual drive, headache, nausea. They will start doing vomiting due to the excessive load of that medicine inside the body. Vaginal thrush or vaginal yeast infection due to that abnormal concentration of hormone inside the reproductive tract. It will cause infections. Okay, mood swings will happen inside the females, and breath symptoms, breast cancer can also occur inside the female if that side effect of that drug will happen. Okay. Next is your medicinal termination of pregnancy. Okay, it is the intentional or voluntary termination of pregnancy before full term is called MTP or induced abortion. Means abortion is MTP, MTP is abortion. Okay, 45 to 50 million MTPs are performed in a year all over the world. Okay, 45 to 50 million MTP means 45 to 50 million abortion is done in a year in the world. Okay, MTP helps decrease the population. Obviously, you do abortion, you are not getting a child, automatically population goes down. Okay, many countries have not legalized MTP due to the emotional, ethical, and religious and social issues. In India, MTP legalized in 1971. ओके इंडिया में एमटीपी लेगलाइज हुई थी 1971 में, उससे पहले एमटीपी इलीगल थी इन इंडिया. ओके. नेक्स्ट, medical termination of pregnancy. What is the importance of MTP? First, to avoid unwanted pregnancy due to the causal intercourse or failure of contraceptive using during coitus or rape. Okay, it is essential in the case where continuance of pregnancy could be harmful to the mother or the fetus or both. See, sometimes sometimes ectopic pregnancy takes place. Means implantation should be done in your uterus, but sometimes that thing happen in fallopian tube or in the coelom cavity of your reproductive tract. So that thing is not good for the developing fetus and mother also. At that time, MTP is also performed. Okay, so MTP is safe during the first trimester, means up to the 12th week. Till 12th week of pregnancy, MTP is safe. If you done MTP after 12 week, means after first trimester, it is not good for baby or for that mother, especially for mother. Okay. Then problem related to the MTP. First, majority of MTP are performed illegal. Okay, in many states, just like states in India like Haryana, Rajasthan, they do not want girl child, so they perform MTP. They kill the child inside the womb of the mother, or if that child take birth, then they will make her die. Okay, misuse of amniosynthesis test for fetal sex determination. If the fetus is female, it is followed by MTP. Such practice are dangerous for the young mother and fetus both. Okay, amniosynthesis, in this some amniotic fluid of the fetus is taken to analyze the fetal cell and dissolve substances. It is used to test the presence of genetic disorders, survivability of fetus, etc. The good use of amniosynthesis is to determine genetic disorder. Genetic disorder only, not disorder of heart, kidney, liver. Okay, only genetic disorder, just like infantile syndrome, Down syndrome, Hutchinson syndrome, color blindness. Okay, hemophilia, sickle cell anemia. All the genetic disorder can easily read it by amniosynthesis. But the bad use of amniosynthesis is the determination of sex of the fetus. Okay, so Government of India enhanced the Medical Termination of Pregnancy Act 2017 to reduce illegal abortion and consequent maternal mortality and mobility. Okay, due to this MTP, some mothers will also die with the fetus. So in 2017, Indian government released a Medical Termination of Pregnancy Act, in which you have to take a legalized permission from government hospital or a government legalized gynecologist. Then you will can perform MTP. Okay, according to this act, a pregnancy may be terminated within the first 12 weeks on an opinion of a registered medical practitioner, means a government doctor which is a gynecologist. Okay, if the pregnancy is between 12 to 24 weeks, two registered mediated practitioner must be opinion, means we have to take from two different gynecologists to perform that MTP or not. Okay, this is the procedure in which you will do amniosynthesis. This is ultrasound machine which send some sonar type location on the child, which will turn back and you will see the fetus on a computer screen. Okay, and this is a particular fine needle which is injected inside the womb of the mother, and which we suck the amniotic fluid, and this amniotic fluid will seen under the microscope, and we will study what type of that genetic disorder that baby have. Okay, now next is sexually transmitted diseases, STD. Okay, we studied that thing in human health and disease. Again, it is here in this chapter also. Disease transmitted through sexually intercourse as called sexually transmitted disease, infectious disease, venereal disease, or reproductive tract infection, RTI. Okay, example: gonorrhea caused by bacterium, syphilis caused by bacterium, genital herpes caused by virus, chlamydia caused by your protozoan, genital wart caused by virus, trichomoniasis caused by again protozoan, hepatitis B caused by virus, HIV caused by AIDS caused by HIV. Okay, these are the images of these all diseases. This is syphilis, this is gonorrhea, this is genital herpes, and this is genital warts. Okay, so this is these are the names of the diseases, and these are the organism, and these are the what are the signs of that disease, particular disease. So these five are curable sexually transmitted disease, means you can cure them. You have the medicine, you have the cure of these diseases. First we have gonorrhea caused by bacterium Neisseria gonorrhoeae, and its painful urination, pain around the urethra. These are the symptoms of this disease. Second we have syphilis, bacterium Treponema pallidum. Okay, it is an spirochete bacterium. Itchy, fluidy discharge, pain in urogenital tract. Okay, next is chlamydiasis. This is caused by bacterium chlamydia. Okay, painful urination, painful urination and pain during the intercourse, muscular discharge from mucus discharge from penis and vagina. Okay, next one is genital wart caused by virus, human papilloma virus. Okay, warts over external genitalia means podesis type of appearance will occur on your external genitalia, vagina and cervix. Next is trichomoniasis caused by protozoan Trichomonasis vaginalis. Okay, in female, it especially infects female. It is especially infect in female inflammation, itching, and whity froth in vaginal discharge. Males remain asymptomatic. I say again, trichomoniasis always happen in female. It never happen in male. Okay, male remain unsymptomatic but occupationally painful swelling occur in the seminal vesicle and prostate gland. Okay, this will affect only female. In male, there is only some swelling take place, not that much dangerous to male. Okay, trichomoniasis. Next, we have incurable sexually transmitted disease. We do not have cure of these diseases yet. Okay, first the most common sexually transmitted disease, AIDS, caused by HIV, fever, weakness, infection, cancer. Cancer is your sarcoma, Kaposi sarcoma. Okay, Kaposi sarcoma is the cancer which happen in your AIDS. Okay, next is genital herpes caused by herpes simplex virus type 1 and type 2 both. Okay, in this reddish ulcer oval external vaginal and vaginal discharges happen. See this. That red color of this color will happen on the external genitalia of male. Okay, next one is hepatitis B, which is caused by hepatitis B virus, fatigue, persistent low grade fever, jaundice. That is the main sign of hepatitis B, that is jaundice, because it affects your liver also. Okay, rashes and abdominal pain, because it is happening in the abdominal region, it is affecting mainly liver. That's why you have abdominal pain. Okay, now how that sexually transmitted disease is spread. First, unprotected sex or sex with multiple partners, then sharing needle with infected person. Okay, then transmission from infected mother, as these viruses can easily cross placenta. Okay, and infection from blood users. Okay, if you are injected infected blood, automatically you get these diseases. Okay, now these are the early symptoms of all these disease: itching, fluid discharge, light pain, swelling, etc. in genital region. This lead to complications such as pelvic inflammation disease, PID, abortion, stillbirths, ectopic pregnancy, infertility, and cancer of reproductive tract. Prostate cancer is most common in male. Okay, next, prevention. How can you prevent these STD? Avoid sex with unknown partners or multiple partners. Then always use condom during coitus. In case of doubt, go to qualified doctor for early detection and get complete treatment. If you have any doubt that your partner have STD or you get that STD, then go to doctor. वरना तुम लोग गए. Okay, now last part of this chapter is infertility. Okay, how to fight again infertility and what is the infertility. Okay, so it is the inability to conceive or produce children after 2 years of unprotected sex. Means you are doing sex without using any contraceptive, and again after that you are not getting any baby, any that fertilization or anything, then this condition is called infertility. It can happen in male, it can happen in women, and it can happen in both. Okay, the reason for this may be physical, congenital disease, drug or immunological, or even psychological by your brain. If you are thinking that I cannot do sex, I have that not having proper partner or having does not have the power to do sex, then you cannot produce a child also. Okay, this disorder can be treated by infertility clinics using assisted reproductive technology, which we call ART. Okay, so ART having these different types. Gamete infrafallopian transfer, gift, in which we transfer gamete inside the fallopian tube. Then intracytoplasmic sperm injection, artificial insemination, surrogacy. Okay, and in vitro fertilization, we have two types: ZIF and UTI. Okay, so first we have in vitro fertilization, IVF. It has two types: zygote intrafallopian transfer or intrauterine transfer. Okay, then gamete intrafallopian transfer, intracytoplasmic sperm injection, and artificial that insemination. We are going to study that thing. Okay, so first we have in vitro fertilization. Okay, it is of two types: zygote intrafallopian transfer, in which you introduce zygote which is less than 16 cell stage. Okay, and intrauterine transfer, which is more than that thing. Okay, so zygote intrafallopian transfer, GIF, transfer of zygote or early embryo within up to the 8 blastoma, mean 8 cell stage, not 16, not, it is 8 cell stage. Okay, into fallopian tube. ZIFT is transferring of 8 cell zygote, that cell states inside your fallopian tube, that is zipped. Okay, and intrauterine transfer is the transfer of embryo with more than 8 cells, this is 16, okay, into the uterus. Okay, more than 8 means 16. Okay, this is introduced inside the uterus. Intrauterine transfer, it is for uterus. Zygote intrafallopian transfer, it is for fallopian tube. Okay, children get confused between zift and gift. See, zygote intrafallopian transfer means you have a zygote, means your egg and sperm get fertilized, that is zygote, and it converts, come into in vitro fertilization. Next we have gift. Okay, gift, gamete intrafallopian transfer. Okay, transfer of an ovum from a donor into fallopian tube of another female who cannot produce ovum, means the ovary is not functioning of that female in which we introduce that ovum from some donor, that's why it is called gift. Okay, you are giving gift to an infertile lady. Okay, but can provide suitable environment for fertilization and development, means uterus is working but your ovaries is not working, then you will perform gift. Uterus is the site of that implantation, it is working, the fertilization site is working, but the main thing, the ova producing site, ovary is not functioning, then you will perform gift. Okay, next is intracytoplasmic sperm injection. Okay, a laboratory procedure in which single sperm from male partner is injected directly into egg from female partner. After the fertilization, the embryo is implanted into the woman uterus. This is the whole procedure. First we see the most perfect sperm by the help of microscope, then we take micro injection and inject that sperm inside the ova. Okay, after that we will see that fertilization take place. Okay, then the cell will convert into the two cell stage, after that it will convert into the zygote. Now that the zygote is get implanted into the female reproductive tract inside the uterus. Okay, next is your artificial insemination AI technique. Okay, in this, semen is collected from husband or donor is artificially introduced into the vagina of uterus of female. The male partner whose penis cannot stand that much tall is used this process. Okay, artificial insemination into the uterus is known as intrauterine insemination. Okay, this technique is useful for the male partner having inability to inseminate inside the female or low sperm count. Okay, next, last that infertility solution is surrogacy. Okay, here a woman surrogate mother bears a child for a couple unable to produce children because the wife is infertile and unable to carry. It is not having the power to produce ova. It has not the power to make that ova. That if some other techniques just like gift we will do, their uterus is not compatible to make that zygote, to give the zygote a home. So in that case we use surrogacy method. Okay, we have live example of this, just like Shah Rukh Khan and that Gori. Okay, Gori Khan. Okay, that Gori Khan does not, in that case that Gori Khan does not want to disfigure its body, that's why he used surrogacy method. Okay, the surrogate is the impregnation through the artificial insemination or implantation of embryo produced by IVF technology. Okay, problems of art, it requires high precision handling and specialized professional and expensive instrumentation. Okay, it will be done with the help of highly professional function peoples. Not a normal guy cannot perform this procedure. And second, it uses very high level technology, high level machines, very costly machines. Okay, emotionally, religious and social problems are related to that. Surrogacy is the most common that problem which related to that art techniques. Okay, last but not the least, legal adoption is the one of the best method for couple looking for parenthood. Okay, and if you do not want children, then you will adopt dogs also or cat also. Okay, animals are the best partner for your life than human. Okay, so legal adoption is also one thing which you can do to adopt children. Okay, so that is uh, reproductive health. Okay, in 10 after 10-15 minutes we are going to start principle of genetics, second largest chapter of biology. Hello. So now principle of inheritance and variation. Okay, it is the most important chapter of 12th class and the longest chapter of 12th class after molecular basis of inheritance. Okay, so first from starting this chapter we have to discuss some terms. Okay, so first term is genetics. Okay, it is a study of inheritance and heredity and variation of character or a study of gene or chromosome. This thing is called genetics. Okay, inheritance, transmission of characters from parents to the progeny, it is called heredity. Okay, that sorry inheritance, and it is the basis of heredity. Okay, next is variation. Difference between parents and progeny is called variation. In this image, you can see that is the actor of Kerala, okay, and that is the father of that guy, okay, both are different from each other, okay, so this is the variations. Next is the character, a heritable feature among the parents of offspring, example eye color, that is called character. Next is trait. Various forms of characters are called traits. Means if this is a character, the character of this pen is black. Okay, then in let's see, this is the character that this is pen. Okay, now the color of this pen black, blue, green. This can be the trait of this pen. Okay, character having different traits, but different traits have only a single character. Okay, next is the factor or gene. It is the unit of hereditary segment of DNA which is responsible for inheritance and appearance of character. These factors are referred as gene by Jonasson. The first scientist come in this chapter is Jonasson, who give the term gene. Okay, after that Mendel used that term and give that element and factor. Okay, factor and element, this is given by your Mendel, and Jonasson give that thing named gene. Okay, next thing is your allele. This is alternate form of gene which is located on same position loci on homologous chromosome called alleles. Okay, see, this is allele 1, this is allele 2, a capital small a. Okay, this is your homozygous AA, this is homozygous small a, and this is heterozygous in which you have a capital A and small a. Okay, so homozygous, it is pure. Okay, means presence of two similar alleles of genes of a diploid organism. For example, capital T, small t, small r, or capital R. These are called homozygous or pure condition. Then you have heterozygous or impure, means mix up. Capital T, capital R, capital W, capital Y, small y. That conditions are called heterozygous conditions. Okay. Next is hemizygous. Okay, in which diploid organism presence of single allele of gene human male are hemizygous for sex-linked genes located on sex chromosome XY conditions. Okay, in which X is expressing its character, Y having different characters. Okay, next is phenotype. This is the physical, morphological character of particular organism. Then it is called phenotype. Next is genotype. It is the genetic constitution, genetic makeup of the organism. The genes, the present inside their chromosome, inside their genes. This is called genotype, genetic makeup. Okay, genotype and phenotype terms was coined by Jonasson again. So word gene is coined by Jonasson, and word that phenotype and genotype, it is coined by Jonasson. Okay, next is phenocopy. Two different genotype develop similar phenotype under different environment condition. Then it is called phenocopy of each other. Means it is a copy of each other's. Okay. Next, dominant allele, that allele which express itself in every generation. Okay, example TT, that is homozygous dominant, and capital T small t, that is heterozygous condition. Okay, next one is recessive allele, it does not express itself, it does not show itself in the progeny, that is small t, small w, small c, small b, small b. Okay, then it is called recessive alleles. Okay, next, hybrid vigor or heterosis. Superiority of hybrid offspring over its parent is called hybrid vigor, and it's developed due to heterozygosity. Okay, hybrid means that hybrid thing, the hybrid nature and XY, that sorry, that capital T small t, capital W small d compressed, that is heterozygosity. Okay. Then inbreeding depression. Okay. Hybrid vigor can be lost by inbreeding depression or self pollination, mean pollination between that sex between same parents. Okay. Same family parents or same organism, then it will causes inbreeding depression. Then you have father of genetics, Gregor Mendel. Okay, he is the papa of genetics. He is the mafia of genetics. He is the Pushpa of genetics. Okay, without this guy, we cannot study genetics. Okay, that's why he is known as father of genetics. Okay, now we have some information about Mendel Chacha. Okay, first experiment performed by Mendel on genetics and description of mechanism of hereditary process and formulation of principle of known as Mendelism. Whole theory of genetics is given by Mendel, and that we call Mendelism. Okay. Next, Mendel postulate various experiment laws in relation to genetics. That is law of dominance, law of independent assortment, law of segregation. These all are the laws given by our Mendel Chacha. Okay. So Gregor Johann Mendel, 1822 to 1884, was the Australian monk. He is not a scientist. He is not anything. He is a normal monk. He do prayer in that church. Okay. He conducted many experiments with garden pea plant and considered as the father of genetics. Okay. The result of his experiment were published in 1865. Okay. After the death of Mendel, the three scientists come in the play: Carl Collins, Hugo de Vries, and Erwin von Tesemek. Okay, these three guys developed and modified the theories of Mendel's, and now we get modified theories of Mendel, which we study in this chapter: law of independent assortment, law of segregation, and law of dominance. These been expanded and modified by these three scientists. Okay, this is Carl Correns from Germany, Hugo de Vries from Holland, and Erich von Tesek from Australia. Your Mendel is also from Austria. Okay, so Correns converted two postulates of Mendel into two laws of heredity Mendelism, that is law of segregation and law of independent assortment. Okay, law of segregation and law of independent assortment is postulated by Mendel, but that laws is modified by Carl Correns. Okay. Next, we have reasons of Mendel's success. What are the reasons behind Mendel's success? How that Mendel guy get that much success in their life? First, Mendel studied the inheritance of one or two character at a time. At a single time, he studied only one or two character. He no go behind 3, 2, 5, 3, 6, 7, 8, that much of character. He studied only two character at a time. Okay, Mendel quantitatively analyzed the inheritance of qualitative characters. He goes with quality, not with quantity. Okay, he always go with quality, never go with quantity. Okay. Next, he maintained the statistical record for all experiment. Mean he used mathematics, statistics. Okay, mean, mode, medium to define its probability. Okay. Next, his experiment had large sampling size, which gave greater credibility to data that he collected. Okay, he used large sample, means he used large number of pea plants to accumulate its data. Okay, and the most important point of success of Mendel is selection of material, that is pea plant. Okay, Pisum sativum, that garden pea, normal garden pea, he selected that thing. Okay, the reason of that thing is garden pea plant is suitable for study, which have following advantages. First, pea plant is annual plant with short life cycle of 2 to 3 months, and large number of offspring can be produced and analyzed with short period of time. First reason of the success. Second, it has many contrasting traits. Okay, we studied 7 characters and 14 traits in this chapter of pea plant. Okay, naturally pea plant carry out self pollination. Okay, it carried out self pollination, so you do not have to do anything with that plant. Okay, reproduction it can do itself, self pollination it will done. Okay, cross pollination can be performed in artificial. Cross pollination it cannot do by its own, so we perform that cross pollination by our own. Okay, and pea plant is easy to cultivate. Pea seeds are large in seed. You can easily see the seeds of pea plant that you eat in matar paneer sabji. Okay. Next, Mendel studied 7 characters or 7 pair of contrasting. 7 pairs means 14 traits, and 7 characters. These are the 7 characters and these are the traits of these 7 characters. Stem height, flower position, flower color, pod shape, pod color, seed shape, seed color. These all are 7 characters which Mendel studied on pea plant. And these are the traits of these 7 characters. Stem height, tall and dwarf. Tall is dominant trait, recessive is dwarf trait. Okay, dwarf is the recessive trait of height of the plant. Okay, second flower position, exile is a dominant, terminal is recessive. Okay, flower color violet, that is dominant, recessive is white. Okay, pod shape inflated, that is dominant, constricted, that is recessive. Then pod color green is dominant, recessive is yellow. Then seed shape round is dominant, wrinkled is recessive. And at last seed color yellow is dominant, recessive is green. And you can see these all things in the image: tall, axial, violet, full, green, round, yellow. These all are dominant traits. Okay, dwarf, terminal position of flower, white color of flower, constricted pod shape, yellowish color of the pod, wrinkle shape of seed, green color of pod. These all are recessive traits of pea plant. Okay. Next, now Mendel experiment technique. How do he do experiment? Okay, that or cross pollination. Okay. So first he select a pure plant. Okay, pure plant means homozygous condition. Capital T, small t. Okay, that selection of pure plant means, I will write here, capital T or small t. Okay. Next, Mendel selected 14 true breeding varieties of garden pea plants. Second, true breeding varieties in the plant varieties that have undergoes continuous self pollination and so stable trait of inheritance expression from several generations. Means he do self pollination and after self pollination he find that same result is coming. Okay, and we got this thing. Okay. Next, Mendel developed true breeding varieties of garden pea plant by continuous self-pollination and selection. Okay. Then hybridization between pure plants means capital T into small t and we get same result, that is T small t. Okay. So in this hybridization process we have five stages. Okay. First is emasculation. Removal of anther from bisexual flower and making that bisexual flower unisexual flower. That is emasculation. Next is bagging with paper bag so as to prevent undesirable cross pollination. The cross pollination we do not want is being prevented by the help of bagging. Okay. Next, tagging. Emasculated and bagged flower are tagged by the writing date and time on the every step. See, Mendel do precise thing. He do everything with proper way and proper arrangement and proper you can say in a proper means we can say that thing is in statics in a proper number. Okay, so he do tagging, he tag all the flower, at what time he do that cross pollination, at what time he do bagging of that flower, at what time he do emasculation of that flower. Okay, last is dusting. Pollen grain from the selected male plant and dusted over the stigma of emasculated flower with the help of fine brush. Okay, and at last he collect the seed of F1 hybrid, that is your capital T and small t. That is your F1 hybrid. Okay, and at last selfing of F1 hybrid to obtain F2 generation means capital T into capital T small t. Okay, and we get this thing. One homozygous dominant, two heterozygous dominant, and one homozygous recessive. That is the result of selfing of F1 hybrid and getting F2 generation. Okay. Now next, monohybrid cross, inheritance of one gene. Okay, that cross I will done previously. Okay, a cross done to study inheritance of one character, one pair of contrasting trait at a time. The single time you are studying a single character. You are studying that height of that stem or color of the flower or that position of flower at a single time. Then it is come under inheritance of one gene. Okay. First of all, Mendel selected tall and dwarf plant. Okay. So this is your capital T and this is small t, you do that thing and you get first pairial progeny F small t and hybrid and heterozygous tall plant. Okay. Now there is one scientist, Punit. Okay. Punit Punnett square, he provided that it is the graphical representation to calculate the probability of all possible genotype of offspring in a genetic cross. Means this square in which you get all the possibilities of the cross between two different heterozygous conditions. Okay, so there the production of gamete by the F-part plant formation of zygote in F2 generation can be easily shown in the diagrammatic view, which we call Punnett square. Punnett scientist is belong from England. Okay, next male gametes lie horizontally on the top of the row. Okay, this is your male. Okay, and female gamete lies vertically in the left of the column. These both are the female gametes. Okay, phenotypic ratio is 3 is to 1. These three are tall and this one is dwarf, then you get 3 is to 1 ratio. And genotypic ratio is 1 capital T, 2 capital T small t, and 1 small t small t. That is 1 is to 2 is to 1 ratio. That is the genotypic ratio. And phenotypic ratio is 3 is to 1. Okay. Now next is law of dominance and law of segregation, which comes from your monohybrid cross. Okay, law of dominance. If a cross is conducted between two character of parent, then the character that appears in F1 generation is called dominant. Means when you are doing cross between a tall and a dwarf, the result of that thing is tall. Okay, so this thing is called law of dominance. Okay, and the suppressed one will appear in F2 generation is called recessive. Okay, in F2 generation you will see that small t coming in the Punnett square. Okay, so this is the law of dominance in which you will get a dominant character, a dominant phenotype and genotype in F1 generation always. Then it is the law of dominance. Okay, next is law of segregation. There is exception of law of dominance, that is law of that incomplete dominance and codominance. Okay, now law of segregation. This law is based on the fact that alleles do not show any blending. No, never. They never show blending. Means according to Mendel, if you breed white color with red color, it will not show pink color. Okay, three should be red dominant, one should be white recessive. According to Mendel, will study incomplete and codominance, which is the exception of law of segregation. Okay, so recovered success in F2 generation through one of these is not seen in the F1 stage. This law based on the basis of F2 generation. Okay, so this is the law of segregation in which at the end you will see there is no blending will happen. Okay, next is four line method of gamete formation. Okay, this is very simple. I can draw this here. You can see if there is a that phenotypic correction of that genotypic for that genotype we having capital A, small a, capital B, small b. Then find how many gamete this parent can produce. Okay, simple. Put a capital A small a. Then make two arrows, write capital B ahead of this. Okay, and small b here, then capital A above there and small there. You get capital A, capital B, first gamete, second gamete, capital A, small b, third small a, capital sorry, capital small a, capital B, and small a, small b. Four gametes is produced by this. If it become tri A, B, B, C, small c, then the number of gametes become 8. If it is tetra, then it will become 16. If it is penta, then it will become 32. Okay. Next, Next, we have dihybrid cross. Okay, inheritance of two genes. Okay, a cross done to study inheritance of two characters or two pair of contrasting traits. Mendel wanted to observe the effect of inheritance of one character on other character. Okay, he performed dihybrid cross to see that what is the influence of one trait over the second trait. So he collect two traits. Okay, one is round shape of seed and second one color of the seed. Okay, which we write like this. Capital R, capital R, capital Y, capital Y. Okay, and wrinkled green, small r, small r, small y, small y. Okay, now we will do the selfing of this and we get the result capital R, small r, capital Y, small y. Okay, that is your F1 generation of dihybrid cross. Now we will do again that then we do selfing of this. Okay, now we are going to selfing that. Selfing between the F1 generation capital R, capital Y, small y into capital R, capital Y, small y. At last we get a 16 box Punnett square in which the gametes will be produced. Okay, capital R, capital R, capital Y, capital Y, capital R, capital R, capital Y, small y, capital R, small r, capital Y, capital Y, capital R, small r, capital Y, capital Y. This how this whole Punnett square will form. Okay, the next slide have that thing. Okay, this. Okay, capital R, capital Y, capital I, that is round yellow. Capital R, capital R, capital Y, small y, round yellow. Capital R, capital small r, capital Y, round yellow. Capital R, capital Y, small y, round yellow. Okay, this line is full of round yellow. This round yellow, this round yellow, then this is wrinkled yellow because small r there, small r there, that is again wrinkled yellow, small r there, and this is wrinkled green, small r and small y is there. Okay, this is the Punnett square of F2 generation. Okay, next the ratios 9 is to 3 is to 3 is to 1 is the phenotypic ratio of two generation in dihybrid cross. Again, most important point, 9 is to 3 is to 3 is to 1 is the phenotypic ratio of F2 generation in dihybrid cross. Okay, next we have genotypic ratio, that is 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1. That is the genotypic ratio of F2 generation in dihybrid cross. Again, I am saying 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1. That is the genotypic ratio of F2 generation in dihybrid cross. Okay, so by dihybrid cross we got one principle again, that is a third principle of genetics, Mendelism, is law of independent assortment. Okay, conclusion of dihybrid cross is your law of independent assortment. This law states that when two pair of traits, two different characters are combined in hybrid, segregation of one pair of characters independent of other pair of character, means both characters are not dependent on each other, they are independent of each other. Okay, it is based on F2 generation of dihybrid because law of dominance, law of segregation is based on F, that monohybrid cross, while your law of independent assortment, it is related with your dihybrid cross. Exception is linkage. The exception of law of dominance, law of segregation is your law of codominance and law of incomplete dominance. Okay, so this is again trihybrid cross. Okay, now we are taking three characters. Okay, and in this ratio we get 27 is to 9 is to 9 is to 3 is to 9 is to 3 is to 3 is to 1. That is the phenotypic ratio. In CBSE or your state board exam, this thing will not going to be asked because this is very complicated thing. But in NEET, they can ask the phenotypic ratio of F2 generation in tri-hybrid cross. That is 27 is to 9 is to 9 is to 3 is to 9 is to 3 is to 3 is to 1. Okay. Now next is your back cross. Okay, back cross is the cross between hybrid and its parents. Okay, it is of two types. Back cross, it is of two types: test cross and out cross. Test cross means cross between hybrid offspring with recessive parents. Okay, and out cross is hybrid into dominant parent. Test cross is done to find the genotype of the parent, genotype of the progeny, genotype of the parent. Okay, here test cross have to find the out the genotype of unknown dominant individual. Either it can be small t capital T or it can be capital T capital T. Okay, and the ratio for test cross is always 1 is to 1. In monohybrid 1 is to 1, in dihybrid 1 is to 1 is to 1 is to 1, but its ratio always 1 is to 1. Okay. See, monohybrid cross, test cross 1 is to 1, dihybrid cross, test cross ratio 1 is to 1 is to 1 is to 1. And if we go in tri-hybrid cross, then test cross ratio is 1 is to 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 1, 8 times 1. Okay. Now next is non-Mendelian inheritance. Now we are not going to follow the rules of Mendel. Okay, the laws of Mendel is not followed in non-Mendelian genetics. Okay, so beyond dominant and recessive alleles, there is something which come under non-Mendelian genetics. Okay, the examples of that is incomplete dominance, multiple allele, co-dominance, and pleiotropic gene. And that's your intergenic interaction. It is of two types: intragenic interaction and intergenic interaction. Intragenic means two alleles of gene present on same locus of homologous chromosome produce modified phenotypes. Means it is present on same locus. Locus is same and they are giving different result. Okay, in Mendelism we studied that they are present on same locus and they are giving same result every time. Means 3 will be tall, 1 will be drop, but this thing will not show in your intragenic interaction. Then we have intergenic interaction in which the genes are present on different chromosome. Chromosome number is changed, chromosomes are changed and produce a change phenotypic effect. Example, epistasis gene and complementary gene. Okay, so first we have incomplete dominance. Okay, this law does not follow law of dominance. Okay, exception Mendel's law of dominance. Okay, in this interaction, dominant allele is not fully dominant over the recessive allele, so heterozygous condition and intermediate phenotype is appear. Just like if F1 in F that if the parent is capital T and it's giving green color, sorry, red color, and small t, it is giving white color, then the F1 generation does not show you red color, it will show pink color. Okay, so example is flower color in Mirabilis jalapa. This is the flower of Mirabilis jalapa, 4 o'clock plant. Okay, it is called 4 o'clock plant because in the evening of 4 p.m. the flower blooms in this plant. Okay, this is the cross between this plant. Okay, this is your dominant, homozygous dominant, this is homozygous recessive. When you cross this thing, you produce F1 generation which is heterozygous, but its color is not resembling both the parents. Okay, now when do you do selfing of these F1 generation, you will find that there is three different coloring is coming in your F2 generation. One is your capital T, that is your pink color. Then capital T, small t, that is your intermediate pink color, light pink color. Okay, and that small t, that is your white color. The genotypic ratio in this is 1 is to 2 is to 1. Okay. Genotypic ratio is 1 is to 2 is to 1 and phenotypic ratio is also 1 is to 2 is to 1. Okay, same is not coming which we studied in Mendelism. Mendelism phenotypic ratio is 3 is to 1 of that monohybrid cross, but in this it is not coming, it is coming 1 is to 2 is to 1. Okay, next example is flower color in Amphidium majus. Okay, it is your snapdragon plant, okay, or dog plant. This is your snapdragon plant, and this is also called skull plant. When the flower of snapdragon plant get dried, it will look like human skull. Okay, so again same thing will happen in this also, because it is also an example of incomplete dominance. Okay, and third example is size of starch grain in pea plant. Okay, in which you will get intermediate size. Okay, not small, not big, you will get intermediate size. Again the ratio comes 1 is to 2 is to 1, that is phenotypic, and genotypic is also 1 is to 2 is to 1, that is your codominance. Okay, next one is your sorry, incomplete dominance. Now next is your codominance. Okay, now the characters, both the characters will come into the F1 progeny. Okay, not mixing up take place, but the progeny will show both the characters. Okay, the progeny will show both the characters of homozygous dominant and homozygous recessive. Okay, see, this is the example. Okay, this is black bear, this is white bear, and all progenies look like panda. It is an not real example, but is an example by which you can easily understand codominance. Okay, black bear crossed with white bear, all progenies will show black and white color appearance. Okay, second example, the roan coat in cow. Okay, that roan color. Okay, it will contain both blue and black color inside the cow. Okay, so example, coat color in cattle, when a black parent is crossed with white parent, a roan color F1 progeny is produced. Note, F2 generation is obtained in animal by submating. Just like self-mating, self-pollination done in plants, in animals we do submating. Okay, now these two F1 generation of same heterozygosity has been crossed, and we get four different animals, which having one having white coat, two having roan coat, and one having your black coat. Okay, against phenotypic ratio 1 is to 2 is to 1, genotypic ratio 1 is to 2 is to 1. Okay, next example is multiple alleles. Okay, so multiple alleles, more than two alternative form of same gene is called multiple alleles. Means two genes are there and they are more form of alternate forms. Then it is called multiple alleles. Multiple alleles are formed due to mutation. Okay, example AB blood group, not ABO. It's AB blood group. Only the AB blood group is the example of multiple alleles. See, there is three or more alleles of the same gene. Okay, 1, 2, 3. 1, 2, 3. Okay, three genes are there which are controlling a single character. Then it is called multiple alleles. Okay, multiple alleles are located on the same locus of homologous chromosome, and diploid individual contain two alleles of gamete containing one allele of a gene. Okay, see, this is a diploid organism containing one allele of the same gene. Okay, sorry, two alleles of the same gene. Next one, example of multiple alleles, as I said, ABO blood group. Okay, in which ABO blood group controlled by gene I. The gene I has a three alleles, IA, IB, and I. IA is dominant, IB is dominant, and I is recessive. The plasma membrane of red blood cell have sugar poly members that protrude from the surface and kind of sugar control, that is gene I. Possible phenotypes is A, B, A, B, and O. Possible number of genotypes, can you tell? That is 7. Okay, in this table you can see. Okay, next is alleles in coat color of rabbit. Okay, we have 4 alleles. Okay, 4 different alleles will control the coat color in rabbit. That is wild type, brown fur, then chinchilla, black tip, white fur, then Himalayan, white fur with black paws and nose and tail, and albino, whole white. Okay, next is your pleiotropic gene. Okay, the single gene control many phenotype characters called pleiotropic gene. A single gene controlling many characters, then it is called pleiotropic gene. Okay, a single gene controlling many traits, then it is called pleiotropic gene. Okay, example B gene, it is dominance and incomplete dominance. Okay, seed shape round BB, small b, capital B, and your BB, that is B gene, which come under dominance. Okay, and incomplete dominance, again that B gene controlling the size of starch grain. Then this is the example of your pleiotropic gene. Okay, in human, many diseases are caused due to the inference of pleiotropic gene like phenylketonuria, sickle cell anemia, which is an autosomal linked recessive disorder. Okay, then your sickle cell disease is a hereditary disease and most of the often among the people of the African ancestry caused by mutation. Alarm क्यों बज रहा है? Okay, sickle cell disease is an heritable disease seen most often among the people of African ancestry caused by the mutation in one of the gene that encode the hemoglobin protein. The disease inherited is an autosomal recessive trait. Okay, this is normal. Okay, normal human, normal RBC. Okay, this is missense mutation, a type of point mutation in which valine come on the place of glutamic acid. Normally we have glutamic acid on the sixth position of beta chain, but in missense it's valine come in the play. Okay, so in complete dominance your phenotypic ratio get 1 to 1, genotypic ratio 1 to 1, codominance phenotypic ratio 1 to 1, genotypic ratio 1 to 1. Okay, so this is end of the first part. Now second part of this chapter. First part of this chapter is completed. Now second part of the genetics. Okay, in which we study linkage and all other characters, that multiple allele, that your complementary gene and your or more genetics. Okay, so non-allic interaction in which two different alleles come in the play. Okay, so first we have complementary gene. Okay, when two non-allic genes are not able to express the dominant trait when present separately, then it is called complementary gene. Means it is capital A, this is capital B, both are dominant. Okay, if alone capital A is there, then it will not going to express inside the phenotype. Okay, if the capital B is there, then it will express itself inside the progeny. Then it is called complementary gene. At least one dominant allele of both the gene must be present to express the dominant trait. Thus, both the genes complement each other to express the dominant trait. Example, that color of flower in Lantharis odensis seed P. Okay, you can see here where alone a capital letter is there, then you get white color, recessive trait. Okay, capital C is there, alone a single C is there, capital. Okay, here you can see alone a single capital, that trait is there. Okay, then alone it is alone P capital is there, C is not capital here. Again that small p is there, capital P is there, alone capital P, again alone capital C.
दो लाइन कर दो लाइन
Okay. Gene which inhibit or suppress the character, it is called epistatic gene, and the gene whose expression is suppressed is called hypostatic gene. Okay. See, we have example of summer squash. Okay. See, in this, when you see the white color, okay, it is affected one. Okay. In this, B gene control all the things. Okay. If B gene is in double, okay, or if your B gene with capital A, then it will show your white color. Okay, capital B should be there, then it will show white color. If there is no capital B, then no white color. Okay, B gene is controlling the color in summer squash. Okay, white, 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 white. On all the cases, you will see capital B is there. Then you have small b, small b, small b, and capital A, capital A. Then it's your yellow color. Okay, and at last, the most recessive one, that is your small a, small b, that gives you green color. Okay, and ratio comes phenotypic 12 is to 3 is to 1. Okay, and genotypic ratio 1 is to 2 is to 2, 4 is to 1, 2 is to 1, 2 is to 1, same that coming in your complementary gene. Okay, next is your lethal gene. Okay, lethal gene, also known as lethal allele or gene that causes the death of an organism that carries the them. Okay, either it is dominant or it is recessive. So death can occur at any point in the organism development, but it's most common during the early stages, means in the embryonic stage. Okay, lethal gene can be recessive, can be dominant, can be conditional, can be semi-lethal or synthetic depending on the gene involved. Okay, recessive lethal gene kill only when they are in homozygous state. Dominant lethal gene, both homozygous dominant and heterozygous individual will die. Okay, so this is the example of recessive lethal gene, and this is the example of dominant lethal gene. This is your snapdragon example, and this is the coat color in mice. Okay. Next is your polygenic inheritance, quantitative inheritance. Okay, or population genetics come under some power, come under the population part of the human. Okay, so polygenic traits are generally controlled by two or more gene, and each gene show quantitative effect. Okay, it affects cost with the quantity, not with quality. Again, it is your quantity, not quality. Okay, so beside the involvement of multiple gene, polygenic inheritors also take into account the influence of environment. Human skin color is a classical example of polygenic inheritance. Okay, so the type of phenotype in this is 2n plus 1, type of genotype 3 ki power n, zygotic combination 4 ki power n, and here n is equal to number of polygene, the number of genes which is used in this. Okay, so we have first example, kernel color of wheat. Okay, the outer covering of wheat seed. Okay, that outer husk part of the wheat is called kern. Okay, kernel. So regulated by two polygene A and B. Okay, so the more capital, the more homozygous dominant, all the four are dominant, then it is red. If three are dominant out of four, then it is light red. If two are dominant out of four, then it is intermediate red. And if one only dominant allele is there, then it is very light red. And if there is no dominant allele there, then white. Okay, and the type of genotype will produce 3 ki power 2, because two alleles are there, A and B, then we got type of genotype 9. Okay, then we have zygotic combination, that is the Puneet square, for that we get 16. Okay, 4 ki power 2. Okay, and at last parental combination, that is 2 by 16, because 2 are parental in that Puneet square, that is this capital A, capital B, and small b, and divide by that 16 into 100, we get 12.5% parental percentage in the plant, in the that progeny. Okay, so we have that Puneet square for okay. So color of the skin, it is an again second example of polygenic inheritance. The inheritance of color of skin in human was studied by Davenport. Okay, human skin color is regulated by three polygenes, that is your A gene, B gene, and C gene. Okay, so in this, when this negro black, capital A, capital A, capital B, capital C, capital B, capital B, capital C crossed with small a, small b, capital small c, then we get an intermediate color which is called mulatto brown color. Okay, so in which we have the phenotype is this capital A, capital B, small b, capital C, small c. Okay, and at last we have this thing. Okay, when we have all the 6 alleles capital, then it is negro black. Out of 6, 5 are dominant alleles, then very dark brown. Out of 6, 4 are dominant alleles, then dark brown. Out of 6, 3 are dominant alleles, then it's mulatto color. Out of 6, if 2 are dominant, then light brown color. And out of 6, if 1 is dominant, then very light brown. and all are small or are recessive alleles, then it is white color. Okay. Then type of genotype 3 ki power n, that 3 ki power 3 equals to 27, and the Punnett square for this zygotic combination is 64 box Punnett square will be formed. Okay. And the percentage of parental character is, it is your sorry, it is not parental plant, it is parental skin, then 2 divided by 64 into 100 equals to 3.125. Okay. This is the tone. Okay, skin tone. See, in this, 5 are recessive, 1 is dominant, then brighter skin tone. Again 3 become 3 become dominant, 3 become recessive, then little bit darker tone. Then 4 become dominant, 2 become lighter, that recessive, then darker tone. Or 5 become dominant and 1 remain recessive, then more darker tone. Okay. And this is the Punnett square. For that, 64 box Punnett square will be formed. Okay. Next is your cytoplasmic inheritance discovered by Carl Correns. Okay. It will seen in snapped that plant of Mirabilis jalapa, the stem color. Okay. Plastid color. Okay. So the gene which is present in the cytoplasm are called cytogenes or plasmogene or extranuclear gene, which is present inside your mitochondria and your plastids. Okay. Total cytogen present in cytoplasm is called plasmon. Okay. Means total means the gene which is present in the means in the mitochondria, inside the plasmid and inside the nucleus. It is called plasmon. Okay. A gene which is located inside the nucleus is called karyogene. The gene which is present inside your nucleus is karyogene. Okay. Inheritance of cytogene occur only through female, because when do fertilization take place, that only the nucleus part of the sperm get inside the ova, remaining part, that mitochondria which is present inside your body, now it is of your mother, not from your father. Okay. Cytoplasmic inheritance involve organelles like chloroplast and mitochondria. It is called organelle inheritance. Okay. This is an example of organelle inheritance, plastid inheritance in Mirabilis jalapa. Okay. See, this is variegated plant. Okay. In which we have mix, and this is the green plant. Okay. Now this variegated plant will produce 3 types of egg. Okay. One is green, one is variegated, and one is white. Okay. So when you cross this green with a normal, with a pollen, it will produce green color. It, when you cross this variegated egg with pollen, it will produce variegated. When you cross white with pollen, it will produce white. Okay. So it is totally depend on egg, not on sperm. Okay. Next one is chromosomal theory of inheritance. So this theory is basically based on meiosis. Okay, meiosis 1. In meiosis 1, we have prophase 1. Prophase 1 have 5 stages. First is your that lipotine, second is zygotin, then then your pectine, then your diplotin, at last you have diakinesis. In your zygotin, there is formation of synovial complex takes place. In your that pectine, crossing over take place, and in your that is diplotin, your chasmata formation take place, and while in your diakinesis, dissolution of chasmata take place. And after this, in metaphase, that all the condensed chromosome place in the that plate, that metaphyseal plate, and after that, on that anaphase phase, that is, it will convert into that in anaphase, you will show that segregation of chromosome take place. They will go to the different poles of the cell. Okay. This will going to be studied in the chromosomal theory of inheritance. Okay. So why did Mendel work remain unrecognized? Okay. It is connected with that unrecognized work of Mendel. First, communication was not easy at that time. Telephone is not discovered. Okay. His mathematical approach was new and unacceptable. Okay. The concept of gene or stable discrete unit could not explain the continuity of variation. It could not give physical proof of existence of factor. These are the drawbacks of Mendel. Okay. And due to which the drawbacks has been read by these 3 guys, Hugo de Veris, Carl Correns, and Rich von Tessman, and they will modify the theories of postulates of Mendel, that law of dominance, law of independent assortment, and law of segregation. Okay. So the last word of Mendel is Mense Zeit word second kommen. My time will yet to come. Okay. In 1900, Dewey Devereaux, Score and Wallace Waxman independently rediscovered Mendel results. Okay, and they will publish that thing in their book. Okay, so chromosomal theory of inheritance proposed by Wallet Certain and Theory Boveri. Okay, these are the two guys, Certain and Boveri. He gave you chromosomal theory of inheritance. They said that the pairing and separation of pair of chromosome lead to segregation of pair of factor they carried. Means the segregation, the factor, the gene which is carried inside the chromosome, they get segregate from each other at the during of your anaphase 1 of meiosis 1. Okay, sudden united chromosomal segregation with Mendel principle and called it chromosomal theory of inheritance. Okay, certain united your chromosomal theory with the Mendelism, and at last you get chromosomal theory of inheritance. Okay, so Wharton and certain Boveri noted that behavior of chromosome was parallel to the behavior gene. Means gene equals to chromosome. Both are same. Both characters are same. See in this you can easily study that thing. Gene occur in pair, chromosome occur in pair, segregate at the time of gamete formation, segregate at the time of gamete formation, and such that only one of each pair is transmitted to the gamete. Again, each pair is transmitted to the gamete. Okay, only from one pair, only one thing will go inside the gamete. Okay, independent pairs segregate independently of each other. They are not independent. They do not dependent on each other. They are independent of each other. One pair segregate independently of other again. Okay, so that Sutton and Boveri argued that pairing and separation of pair of chromosome would lead to the segregation of pair and factor they carried. Sutton and united the knowledge of chromosomal segregation with Mendelism principle and called it chromosomal theory of inheritance. Okay, next, now Morgan come in the play, the second father of genetics. Okay, your Hunt Morgan. Okay, Thomas Hunt Morgan. Okay, he modified and do experimental verification of chromosomal theory of inheritance on your Drosophila melanogaster. Okay, so Drosophila melanogaster, which were found very suitable for such studies because first, they could be grown on simple synthetic medium in laboratory on your ripened banana, on anything it will can easily grow. They complete their life cycle in about 2 weeks. A single mating could produce large number of progeny. There was a clean differentiation of sex, the male and female. You can easily differentiate, easily distinguish. This has many types of hereditary variation that can seen with low power microscope. Even by your naked eyes, you can see the variation, the traits, the character present in Drosophila. Okay, and that's how we get the theory of linkage by your Thomas Hunt Morgan. Okay, so linkage and recombination. Okay, exception to Mendel law of independent assortment. According to Mendel, what he says that genes are independent of each other, factors are independent of each other, they are segregate independently, they are not dependent on each other, but linkage dissolve this thing, linkage does not follow this thing. Okay, the physical association between gene present on chromosome and their tendency to inherit together is called linkage. Means if this is one gene, if this is one gene and this is one gene, they are near to each other, they both get to inherit. If that distance is long, they not get to be inherit. If they are easy, they are near to each other, then it will get inherit. That is law of linkage. Okay, again I am saying the physical association between gene present on a chromosome and their tendency to be inherit together is called linkage. Okay, collective inheritance of character is due to the linkage. Okay, linkage first time observed by Batson and Panith. Okay, linkage term is detailed study done by T.H. Morgan. He performed experiment on Drosophila melanogaster fruit fly and give the theory of linkage. Okay, this is your fruit fly. Okay, and this is how your linked gene is being seen. A is linked with B, C is linked with C. That A is not linked with C, or B is not linked with C, or B is not linked with C. A linked with B, C linked with C. Oh, sorry, C linked with D and A linked with B, because the distance between A gene and B gene is very less. That's why they are linked gene, and the distance between C and D is very less. That's why they are linked gene. A and D is not linked gene. B and C is not linked gene. Okay, so this is linkage. When genes are near to each other, they perform linkage. They simultaneously go in next generation. Okay, so theory of linkage, according to this theory, genes show linkage are called linked genes. Okay, just like this A and B. Okay, these are linked genes. Okay, next, linked genes are non-allelic. They are non-allelic. They are not present on same chromosome. Okay, linked genes are present on same chromosome, but their action is not same. Okay, linked gene can be separated by crossing over. Crossing over take place, linkage absence. Take linkage over when crossing over take place. Okay, crossing over, exchange of chromosomal segment between non-chromatids or homologous chromosome. Okay, when there is crossing over take place between homologous chromosome, then your linkage is breakdown. It will convert into recombination. Okay, see, this is linked gene. Okay, A and B, small a small b. Okay, now crossing over will take linkage break. Okay, this a small a is the capital A small a is there. Now on the place of capital B, small b is here, and on the place of small b, b is here. Crossing over, okay, exchange of this thing will take place. This thing is crossing over. So there is simple logic. Strength of linkage is inversely proportional to crossing over and inversely proportional to distance between linked gene. Means more the distance, more chance of crossing over, less chance of linkage. That crossing over chance is more, then linkage chance is less. Okay, next is the factor affecting crossing over. What are the factors which affect the crossing over? First, less distance. Less distance between the genes, then crossing over not happen, then linkage happen. Okay, that distance less, crossing over less, low recombination, linkage high, tightly linked gene. Okay, distance increases, crossing over decreases, then high recombination rate and linkage automatically goes down. Then it is called loosely linked genes. Okay, so factor that affect crossing over and linkage. First, distance. If distance increase, crossing over increase and linkage decrease. Euchromatin, if it is more, then crossing over increase, and if it is more, then the linkage decrease. Heterochromatin, if it is more, then crossing over decrease and linkage increase. Age, if it's more, then crossing over decreases and linkage increases. Okay, that's why we call that you should marry in the age of between 28 to 34, because after that there is chance of developing of linkage, some bad character inside the progeny also. Okay, next, linkage groups. Okay, all the genes which are located on one pair of homologous chromosome form a linkage group. Genes which are located on homologous chromosome are inherit together, so we consider them as a one linkage group. Number of linkage group equals to the number of homologous chromosome pair or haploid number of chromosome pair. Let's see like this. If there is A gene and there is small a gene, if there is B gene or small b gene, small c gene or capital C or small g, capital D or small d, capital E or small e, then these all things simultaneously go into the segregation. Okay, and these are called linkage group. Okay, so we have different linkage groups. P, that 2 and 14, haploid and number of pairs 7. So linkage group 7. Okay, maize 2 and character 20 and 10. So pair 10, linkage group 10. Means the haploid number is your equals to the linkage group. Okay, haploid and linkage group 7. Okay, haploid 10, linkage group 10. In onion, haploid 8, linkage group 8. In Drosophila, haploid N, linkage group 4. In human, haploid 23, linkage group 23. Okay, so haploid number of your set of chromosome equals to the linkage group. Okay. Next, arrangement of linked gene on chromosome. It will of two type. Either it is crisscross, okay, or it is trans or repulsion. Okay. See, if capital A small b on same side and capital A capital B on same side and small a small b on same side, then it is called criss. Okay, and when your capital A and capital B opposite, okay, diagonally, and capital A small b again they are diagonally, they are intersecting each other, then it is your trans or repulsion. Okay, again I am saying that both the capital things are on same side and small things on same side, then it is your cis or coupling. Okay, and when your crisscross type of appearance take place, then it is repulsion or trans. Okay, next type of linkage. Okay, we have two types of linkage: complete linkage and incomplete linkage. Okay, it is very rare. Okay, 100% linkage, 0% crossing over, new recombination 0, parental combination 100%. Example: few gene of male Drosophila and female silkworm. Okay, see, crossing over take place, same thing will appear, same parent. Okay, gametes is same just like parents. New gamete 0%. Okay, next thing is incomplete linkage. It is the most common type of linkage. In this linkage, crossing over occur between the gene. The maximum frequency of recombination is 50%. It not never cross 50%. It always remain 50 or less than 50. If it is more than 50%, 51, then it will become recombination. It is not incomplete linkage. Okay, see, in this 50 are parental and 50 are new. 50 and this is your complete. Okay, complete linkage, this one. Okay, in which all are parental. Okay, complete versus incomplete. Okay, this is your complete, in which the F1 generation is just like your parent, and if and F2, if then you will see your 50% recombination is there and 50% is look like your parent. Okay, next is genetic map, linkage map or chromosomal map. Okay, it is a straight line in which different genes are linearly arranged in a specific sequence and separated by some distance. Distance is directly proportional to the percent of the recombination. More the distance, more recombination will occur. 1% recombination is equal to 1 map unit or 1 centimorgan. Okay, it is based on the result of test cross. Maximum recombination up to 50%. 50% cross, then there is no recombination, then it will convert into linkage. Okay, next, recombination frequency or crossover value you can find easily by number of recombination of offspring upon total number of offspring into 100. Okay, Alder Safred prepared first chromosomal map of Drosophila by using combination frequency. Combination map provide information about sequence of different linked genes and chromosomes and distance between two linked genes. By the help of chromosomal map, you will find what is the distance between A gene, B gene, B gene to C gene, C gene to D gene. Okay, this is the cross in which we have first character of body color between brown and yellow. Okay, brown is your wild color and yellow is the mutant current hybrid. Okay, second character eye color in which red is the wild and white is the mutant, which is your hybrid. Okay, now in cross B we have first character of eye color which is red, wild, white, mutant. In the previous you will see mutant is yellow and brown is the wild. In this second cross you will see the eye color red is the wild and white is the mutant. Okay, character for wing size, normal one is wild M and the miniature means small size, it will mutant. Okay, okay, in F2 generation you will see 98.7% parental character will show and 1.3% character show recombination, while in cross B the cross between eye color and wing size, because these are separated, see, these both is near to each other. Okay, in this diagram you can see that body color and eye color, this both characters. Okay, these are very close to each other, so linkage is 98.7% and recombination is 1.3%, while in the second cross you will see that eye color and wing size, there is eye color and there is wing color distance, see the distance between eye color and wing size, then you will see linkage decreases 62.8% and recombination becomes 37.2%. Okay, means distance increase, linkage decrease, recombination increase, distance decrease, linkage increase, recombination decrease. Okay, this is the map of your Drosophila which is present in your NCERT. This is simply telling that thing. Okay, the distance will go more, then the percentage of linkage decreases, recombination increases. Okay, so major finding of Morgan's experiment is the two genes did not segregate independently and F2 ratio deviated from 9 is to 3 is to 3 is to 1. That is your Mendelian ratio of dihybrid curve. This will never come in Morgan's linkage experiment. Okay, gene were located on X chromosome. Again, this is the character which that finding Morgan gave you. Okay, third finding, when two genes were situated on same chromosome, proportion of parental gene combination was much higher than non-parental type. Means because of that linkage, parental character more come in the progeny, while non-parental character will not come in the progeny because of linkage. This is due to linkage. Okay, gene of white eye and yellow body color were tightly linked to each other. That's why recombination is 1 is to 1.3. Recombination is 1.3 because they are tightly linked, they are very close to each other. Okay, next, tightly linked genes show low recombination and loosely linked genes show high recombination. This is the example. Okay, this is loosely linked gene, recombination 37.2%. This is very tightly linked gene, recombination 1.3%. Means linkage more, recombination less. Distance increases, more that linkage is decreased, recombination increase. In comparison to this value, you will see that linkage is 62.8% and in this it is 98.7%. Okay, so these are the major findings of your Morgan. Okay, now next is your sex linkage. Okay, it is related to the sex of the organism. Okay, so genes that are located on sex chromosome. In previous we are studying about autosomes. Okay, now we are talking about sex chromosomes. Okay, so sex linked genes and their inheritance is called sex linked inheritance. That was also discovered by your Morgan. He studied eye color of Drosophila and performed reciprocal and test cross and found that genes of eye color is located on X chromosome. Okay, example first, eye color in Drosophila. If red eye color gene is represented with S plus and white eye color is represented W, then this basis different types of genotypes are found in Drosophila, just like X plus plus X plus plus, that is homozygous red, S plus XW, that is heterozygous red eye, XW homozygous white recessive, dominant heterozygous. In it is in female. Okay, in male, X plus Y hemizygous red eye, XWY hemizygous white eye, both condition become hemizygous. In this you have homozygous, heterozygous, homozygous, but in male it is become hemizygous and your hemizygous red eye and hemizygous white eye. It is clear from above genotype that female is either homozygous or heterozygous for eye color, but for eye color male is always hemizygous. Okay, male is always hemizygous in Drosophila for eye character, while your female is can be homozygous and can be heterozygous. Okay, so this is like now this is sex linkage in human being. Okay, this is your X gene and this is your Y gene in male. Okay, so non-homologous region of X chromosome, this is the non-homologous region which is not similar to this Y gene regions. Okay, and this is the biparental same region ABC. You will find that this is a homologous region of XY chromosome, XY linked gene or biparental. Okay, next is your Y linkage. Okay, so gene present on non-homologous region of Y chromosome means this region. Okay, this IJK. Okay, this is non-homologous region of Y chromosome. It is uniparental. Okay, present only in male holoandric gene means the genes which is present only in male in human, it is called holoandric gene. Example SRY gene, sex determining region of Y chromosome, or TDF, testis determining factor, synthesis of protein of TDF. Okay, it is only present on Y chromosome, non-homologous part. Then hypertrichosis, excessive hair on ear pinna, this is again present on Y chromosome, non-homologous region. Okay, gene present on non-homologous chromosome of X chromosome, it is again biparental. Okay, X-linked recessive, the diseases which cause when your that X chromosome become recessive, that is glucose 6 phosphate dehydrogenase deficiency, then DMD, Dutchman, muscular dystrophy, diabetes insipidus, hemophilia, color blindness, these all are X-linked recessive, while X-linked dominant, when your both X gene comes together, then it is pseudorickets and defective animal of teeth. Okay, next, the first is your hemophilia. Okay, Hemophilia is a rare inheritable blinding disorder that prevents blood from clotting properly. It is caused by low level of clotting factor in the blood, which are protein that help stop bleeding. People with hemophilia are more likely to bleed after injuries. Okay, because hemophilia is a bleeding disorder. Okay, in which your blood cannot coagulate properly, due to which blood start to do bleeding. So it is X-linked recessive disorder. Okay, blood clotting time in normal person it is 1 to 8 minutes, while hemophilic person it is 30 minutes to 24 hours. 1 day is it required to clot. Okay, so that person can die easily with blood loss. Okay, so hemophilia again same thing which we study in Drosophila. X positive wild allele normal, XX mutant allele hemophilic. Okay, so X plus normal, X plus XX normal, but carrier XHXH hemophilic. Okay, XY normal, X plus Y normal, and XHY hemophilic means male for hemophilia is either normal or either hemophilic, while female for hemophilia can be normal but carrier and can be hemophilic. Okay, all three conditions are present in females in hemophilia. Okay, so types of hemophilia, we have three types: hemophilia A, royal disease; hemophilia B, Christmas disease; and hemophilia C. In hemophilia A, we have the absence of clotting factor 8, okay, which is called anti-hemophilic globulin HAHG. Okay, in hemophilia we have Christmas disease because it is caused Christmas disease because ninth clotting factor, which is your Christmas factor, it is absent inside your blood. Okay, and hemophilia C, it is due to the absence of factor 11 inside your blood, which is your plasma thromboplastin and anticoagulant. Okay, this is that same thing. Okay, next is your color blindness. Okay, this is again X-linked sex linked recessive disorder. Okay, color blinded person is unable to differentiate between some basic color like red and green color. The person who having color blindness, they cannot see red color object. They will look red color object in different color, either it can look gray or some other color it will see. Or same thing will happen to the green color blindness person. They cannot see green color, any shade of green color they cannot see by their eye. Okay, there is ishara chart. Okay, this is ishara chart in which you can determine the color blindness person. You can identify either the person is color blind or not. Okay, if you are normal, you can see that this 74 is written with green color, and if you are a color blind person, they can you not determine. If I ask from which color this 74 is written, you cannot say that it is of green color because you are green color blind. Okay, same here, this 12 is written with red. Okay, if you are red color blind, you cannot determine this color easily. Okay, so it is again show same thing: female normal but carrier and color blind, and your male is normal and color blind, no middle thing, because you are hemizygous. Male are hemizygous, female can be heterozygous and can be homozygous. These both are homozygous and this one is your heterozygous, and male is hemizygous in both condition. Okay, next type of inheritance on sex linked characters. So we have criss-cross inheritance. It is again given by Morgan. It is of two types: diagenic and diandric. Diagenic means from father to daughter to grandson. Okay, and diandric from mother to son to granddaughter. Just for example, your diabetes mellitus. Okay, it will present in your grandfather, but it is not present in your father, but it can come in your. Okay, that thing is diandric or diagenic type of crisscross pattern. Okay, Knox is non-crisscross. This is simple. Okay, mother to daughter, daughter to granddaughter. Okay, female to female, then it is called hologenic, and from father to son, then son to grandson, then it is called holoandric. Okay, next is your sex limited characters, means both sex having that thing, but only it is showing only in one sex. Okay, then it is called sex limited character. Okay, just like testosterone, it is also present in female, but in female that gene is not get expressed, it only expressed inside the male body. Okay, so these characters are present in one sex, absent in another sex, but their genes are present in both the sex. Okay, that sex, the gene for testosterone activation, it is present in both male and female, but in female that gene is never get activated. Either you have a genetic disorder, then it will get activated, but normally in female testosterone gene is not get activated, in male it get activated. So that type of characters is called sex limited character, which is coming only in one sex, not in second sex. Okay. Example, gene of beard moustache express their effect only in the presence of male hormone testosterone. In female, it is not going to be expressed. Okay. Last one is your sex influence character, means it is present in both, it is expressing in both, but when it's come in dominant, then it will going to be expressed. Okay. Example, pattern baldness. Okay, gene of pattern baldness is dominant B. Okay, so in that, if a male having phenotypes of small b small p, then it have normal hair. Same will happen in female, it having normal hair. Okay, if male have capital B small b, then it will become bald, ganja. Okay, and if in female it become normal hair. Okay, if capital B both are present, then male is bald and female is also bald. So it is called sex influence character. It is influencing both the sex. Okay, this is the image of this thing. Okay, small b, both are not bald. Okay, both have hair patterns. Small b, capital B, heterozygous condition. Male is bald, female is not bald. Homozygous dominant character phenotype, capital B, female and male both are bald. Okay, in this condition. Okay, now last part of this PPT is your sex determination. Okay, so sex determination is the process of sex determination is the process of sex differentiation by which whether a particular individual will develop into male and female. Okay, you can find either that particular organism is male or female, then it is called sex determination. Sex determination is of many types: allosomic sex determination, haploidy, diploidy, environmental sex determination, genic balance determination. Okay, so first we have progamic sex determination, in which sex is determined before the fertilization. Fertilization not take place, but still you know the sex of the organism. Example, drawn in honeybee. Okay, second, syngamic sex determination during fertilization. During the fertilization is not completed, but it is on the way. Then you get the sex, you get the know the sex of the zygote, then it is called syngamic. Example, most of the plant and animals. And last we have epigamic, in which sex is determined after the fertilization, like crocodile and turtle. In this organism, sex is determined by the environment. If that egg get hotter environment, then it convert into male, and if get colder environment, then it will convert into female. Okay. So first we have allosomic determination, which is the most common type of sex determination which is present in our body, male and female human also. XY determination. Okay. It, on the base of this homogametic parents, we have an heterogametic. Homogametic is your XX and heterogametic is your XY, means your male. Okay. This is the karyotype of male because here XY is there. Okay. So why first we have XXXY type. Okay, it is your lysogenous type because it given lysogenous because it is first the research and was searched on lysogenous insect by Stephen. Okay, lysogenous insects are those insects which are present in larva stage. Okay, they develop their development in the form of larva. Then it is called lysogenous or XXXY type of development. Okay. Then this is the normal, your XY, this male will produce two gametes, X or Y, and female will produce same type of gamete, XX. If this X fuses this X, then female will form. This Y fuses with this X, then male is formed. So in sex in humans, it is determined by male, not female. Okay. Next, three mechanisms are there: XXXO mechanism in grasshopper, XXXY mechanism in Drosophila in human, and ZZZW mechanism in birds. Okay. So ZW female, ZZ male. Okay. Now there's one interesting thing that in human, in mammals, XX is equal to female, XY equals to male. But in birds, this thing ulta. Okay, opposite. ZW become female means heterozygous become female and homozygous become male. Okay. So this is your male hen, which we call cock, and this is your hen. Okay, female. Okay, female murgi. Okay. Next is your grasshopper. Okay, in this we call Spitenoid type. Okay, XX female and XO male. Okay, in this you will see that female having X and X homogametic. Okay, male having X and O heterogametic. This is O. This is not zero. This is not nothing. This is not haploid. This is diploid thing. Okay, in some cases, in some people, this that confuse this thing with that honeybee ploidy. Okay, haploidy, but it's not haploid. It is something is there, but we do not give this a name. So it is called XXXO type of sex determination, which is present in grasshopper. Okay, this is we studied that hand one. Okay, next is your haploid diploid mechanism sex determination in honeybee. It is simple that by that label diagram you can easily understand that thing. Okay, in honeybee female haploidy 32. By meiosis that will produce 2 n. Okay, and 16 and 16. This n will undergo parthenogenesis and produce male haploid drone. Okay, now this haploid drone male undergoes mitosis and produce n16. As you know, mitosis start product is equals to end product. Okay, now this n16 of this female fuses with this n16 and produce a 2n female, which as this female feed on royal jelly it will convert into queen, and if this female feed on bee bread it will convert into worker sterile honey bee. Okay, again I am explaining this thing. If female, okay, 2 and 32 ploidy, it will do meiosis, it will convert, it will divide into two, 1 and 16 and 1 and 16. This one directly go into the parthenogenesis and convert into male drone. Okay, now this n16 fuse with this n16 in form of female, which is 2 and 32 number of set of chromosomes. Okay, now if this female feeds on royal jelly, then it convert into queen bee, which is fertile, it can reproduce. But if this female eats bee bread, then it will convert into sterile worker bee. Okay, this is the sex determination in honeybee. Okay, then dance of honeybee. There is two dance of honeybee. First is round dance. Mean if the honeybee does this type of dance in a particular axis, then it is the round dance. If the food is 130 meter near to the beehive, then your bee perform round dance. Okay, and second one is your waggle dance. It will do like infinity sign type of dance when the food is far away from 130 meter from the beehive. And this have two patterns. One is upside waggle dance and downward waggle dance. Upward side means that flower or the nectar side is towards the sun, then it will do upside waggle dance, and if the flower does not showing it face to sun, then it will do downward waggle dance. Okay, this you studied now exosystem in grasshopper, ZW system in birds, and haploid diploid system in your honeybee. Okay, environmental sex determination. Okay, in this environmental condition fix the sex of the organism. Example, we have crocodile. Okay, if crocodile egg get high temperature, then it will convert into male, and if that crocodile egg get low temperature, then it will convert into female. So in crocodile or turtle cases, the determination of sex, it is basis on environment. Okay, if environment is hot, then it is male. If environment is cold, then it is female. Okay, next is your genic balance theory. Okay, given by C.B. Bridge. Proposed genic balance theory for sex determination, in which sex index ratio come in the play. Number of X chromosome upon number of set of autosomes. Okay, in Drosophila, gene of femaleness is located on X chromosome and gene of maleness is located on autosome, as you know, normal. Okay, gene of male fertility is located on Y chromosome, and Drosophila Y chromosome play additional role in spermatogenesis. So Y chromosome is essential for the production of fertile male, fertile progeny. So this is the ratios. Okay, X by A equals to 1, then it is female. If X by A equals to 0.5, then it is male. Okay, 2A plus XX, 2A plus XY, then it is your fertile male. Okay, if X by A value is 1.5, then it is super male or meta male or sterile, 2A plus triple X. Okay, if X by A is less than 0.5, then it is super male or meta male, that is your 3A plus XY, there is sterile, this is trisomy. Okay, then X divided by A, and it is between 0.5 or 1, then it is intersex, 3a into XX, and this is the karyotype for that. Okay, in which we have 6 autosome and 2 sex chromosomes. See, 1 2 3 4 5 6, 1 2 3 4 5 6, 1 2 3 4 5 6 7 8 9, because it is 3a, that's why it is here. Okay, and again this is again 3a, 1 2 3 1 2 3 1 2 3. Okay, this is 3 3 3, this is 3 3 3. Okay, 3a plus xy, this is your super male. Okay, and this is your 3a plus xs, this is your intersex. Okay, this is the in the table form. You can see the x-ray ratio 0.5 male, 1.0 female, 1.5 meta female, 0.6 intersex, and 0.3 is your meta male. Okay, so this is the second part of your that principle of inheritance is done. Now the last part of this chapter is remaining, that is mutation. Okay, which लास्ट पार्ट रह गया वो कर ले Okay, so the remaining part of this chapter, that mutation, okay, and remaining chapters, that ecology unit and sexual reproduction in flowering plant and sexual reproduction in human, it will be conducted on Wednesday. Thank you for watching. Like, share, and subscribe.